Hello, all. A very good morning to everyone. It's my absolute pleasure to welcome all of you to the Clinical Interpretation of Whole Genome Sequences course. Today's program is not just another webinar, it is a knowledge intensive, immersive experience designed for clinicians, researchers, genetic counselors who are passionate about advancing precision medicine and genomic healthcare. Whole genome sequencing is no longer a futuristic concept. It's here, informing diagnostics, guiding treatments, and personalizing care. But sequencing a genome is only half the journey. Interpreting the data to generate clinically meaningful insights is where the real challenge lies. And that's exactly what we aim to demystify today. We have some of the most distinguished minds in clinical genomics with us here today. Dr. Vinod Skaria, Dr. Bani Jolly, Dr. Sridhar Sivasubbu, Dr. Jyoti Bajpai Dikshit, and Ms. Ramya Venkatesha, each bringing in their unique perspectives and practical experience to help bridge the gap between raw genomic data and its clinical application. Whether you're joining us from a hospital, a lab, a university, or your home, know that you're a part of a growing community that believes genomics can radically improve patient care. Please stay engaged, ask questions, and use this opportunity to sharpen your ability to interpret WGS data in a clinical setting. If there was ever a time to build the skill, it's now. The live session links are available across LinkedIn, YouTube, and StreamYard. Please choose the platform of your choice that works best for you and refresh the shared document for access to updated links. With that, I wish you an insightful and an enriching day ahead. Let's begin with the first session for the day. The first session for the day is on what, why, and when of genomic sequencing. The first session is by our revered Dr. Vinod Skaria. Dr. Vinod Skaria is a globally recognized leader in genomics and bioinformatics with over 300 scientific publications, currently serving as the Chief Data Officer at Karkinos Healthcare, Bangalore. He is also an adjunct professor at IIT Kanpur and a distinguished visitor at Ashoka University. A clinician turned computational biologist, Dr. Skaria has led pioneering efforts in rare disease genomics, population scale sequencing, COVID-19 genomic surveillance, and open data science in India. He co founded Guardian, India's largest rare disease genomics network, along with some of India's large scale genome sequencing projects like Indigen and COVID-Seq. He continues to shape the landscape of precision medicine through his research, mentorship, and policy engagements. Welcome, sir. Welcome to the stage.
Thank you very much, Harish, and it's a pleasure to be in here. Over the next 30 odd minutes, I think I'll give a brief overview about genomics and its applications in a very short presentation entitled What, When, and Why of Whole Genome Sequencing. So I'll also delve a bit deeper in why we think this is important to do at this point in time. More importantly, because the technology is changing very rapidly and also is becoming very affordable. And before I start my talk, let me get back a bit into history, and this history is around 50 years old. And I was a medical student at the time point in the history of this particular technology, and probably many of you would be at the same stage for genomics. Now, this contraption, this very small contraption, 50 years ago, was invented by Godfrey Hounsfield, who was knighted later. And he was not a medical professional, he was an engineer by profession. He worked in the music industry. And what he discovered was that a significant number of patients die due to medical procedures because the only way a patient could be looked at or a doctor can peek into the human body of a patient was to essentially open up the patient using surgical techniques. Now, he got fed up with this, and he created this contraption for the first time, which allows clinicians to peek into the human body without having to surgically open up the human body. And this is the invention of the CT scan. And 50 years later, CT scans have become the mainstay of diagnostics in many specialties. And in fact, many of us would have already gone through a CT scanner in our lifetimes. Now, I put this up largely because of the fact that not just being able to peek into the human body, today we have the ability to peek into every one of our 37 trillion cells and understand the blueprint, and that essentially is the whole genome sequencing. And we hope that in not 50 years, but in a few years from now, this is going to become a mainstay in clinical medicine. It is already 20 odd years old. Today, we can sequence and analyze whole genomes as fast as a CT scanner and produces the same almost the same amount of data as a CT scanner. And I think, therefore, we are ripe to be implementing genomics in clinical medicine. Now, India was not far behind in implementing genomics. We were the seventh country to be able to sequence, analyze, and interpret a whole human genome. And this was announced in the parliament way back in December 2009. Now, you might ask this question, does really genomics impact patients? I will probably explain three patients, or rather two patients, to sort of drive home this point. Now, the first story is of Ashi, and of course, not the right name of the patient. Ashi came to All India Institute of Medical Sciences as a nine-month-old baby. The parents had multiple miscarriages in the past. Ashi had microcephaly, small eyes, lethargy, elevated lactate and pyruvate, and with a provisional diagnosis of a mitochondrial disorder or a neurometabolic disorder. Now, she was in the NICU for many days without a diagnosis. And that was when we were trying out looking at genetic testing as part of the Guardian program for undiagnosed genetic conditions. And being a mitochondrial genetic disorder, the first attempt was to sequence the mitochondrial genome, which we did. At that point in time, we were one of the only institutions in the country doing a whole mitochondrial sequencing. We did sequence the mitochondria. We did not get any pathogenic genetic mutation. Then, of course, the next line of thought would be to sequence on a much broader panel. So we did a whole exome sequencing. We did it in quite quick succession, but nevertheless, we did not get any pathogenic genetic variant. Now, this is very common to most patients who come across genetic clinics. They go through triage testing. They lose considerable amount of time, energy, effort in trying to do this testing. And more often, they get up with a negative report. And that is the end of it. Typically, people do not pursue much further. Incidentally, at that point in time, we were doing whole genome sequencing. And this is, mind you, almost 7 years old. And the whole genome sequencing identified something very interesting. The interesting part of it was that the child had a compound heterozygous deletion encompassing 2 genes. And we also found that the deletions were overlapping, and each of these deletions came from either of its parent and encompasses 3 genes. But nevertheless, there is no evidence to suggest that the genes indeed cause the disease. So we would classify them as a variant of uncertain significance, or the VUS. Now, if you come across a VUS, there are 3 broad ways where you can change the classification of a VUS. One is to do segregation in the family pedigree. The second is to look at the population prevalence of the particular genetic variant. And third is to do functional studies. And we did all 3 of it. We did an extended family segregation for this particular variant. We understood that each of the parents were having a heterozygous genetic deletion. Population prevalence was thankfully made available through the Indigent program. And we also did functional validations to prove that the genetic variation indeed caused the disease. And quickly putting forward, what you see here is the father and the mother genomes, and the proband, that is Ashi, is a compound heterozygous deletion that looks like a homozygous deletion in the common regions. But of course, as you see here, there are regions which has low coverage, which overlaps with the paternal genotypes. Now, of course, the next obvious question, as she did not survive, the parents want to do prenatal testing for the next pregnancy, and the prenatal testing was done, probably the first prenatal genome done in this country, and that the fetus also had the compound heterozygous deletion. Now, of course, with multiple pregnancies later, Ashi has three sisters, three healthy sisters born out of the identification of the genetic variant and, of course, born out of prenatal testing appropriately. Now, that largely says that whole genome sequencing can put an end to the diagnostic odyssey because it can uncover genetic variations which were previously not routinely picked up on a whole exome sequencing, or for that matter, any genetic testing previously. I'll come to one more case, and this is much very recently, and this patient referred to us for a second opinion with suspected autoinflammatory disease. An autoinflammatory disease is one of the primary immunodeficiency conditions. Now, closely looking at the exome, what we realized is that the patient's exome did not have reads mapping some regions. Now, this is very, very common in exome sequencing. If you have ever looked at exome sequencing, you will see that there are regions in the exome sequencing where the probes might not have worked, and therefore there would be no reads and therefore no genetic variants called on this region. And fortunately, in this particular case, the parents' genotype was also available, and the parents' sequencing seemed to have worked quite very well, and you see coverage in the exonic regions. Now, what is this? This could be just a technical artifact because this is in exome sequencing. And second, this could also be a deletion that is probably missed out on exome sequencing because large deletions are missed out on exome sequencing. And what we could do is to do a whole genome sequencing, and the whole genome sequencing can put an end to this dilemma. As you see here, there's a patient's chromosomes and you have the parent's chromosomes. You have loss of coverage all across that region, which sort of suggests that this is a homozygous deletion. And this patient has a diagnosis today as Majeed syndrome because of the deletion of LPIN2 gene. And that puts an end to the diagnostic odyssey for the patient. The patient can be taken up for very specific management. And more importantly, the patients and the families could be counseled appropriately regarding the next pregnancy. So whole genome sequencing, therefore, becomes a very useful, important tool to put an end to the diagnostic odyssey. Now, this is nothing new. In fact, there is a lot of literature on how whole genome sequencing is a much better tool than conventional standard of care genetic diagnostics. This is published in 2018 from Canada on a large cohort of individuals, suggesting that WGS has 41% additional diagnostic positivity compared to standard of care. Now, we have similar data from India. This is for cardiac channelopathies. This is from Dr. Sridhar Sivasubbu's lab, worked on by Vignesh, who was a graduate student. What he did was essentially look at whole exome sequencing negative samples for cardiac channelopathies, and whole exome sequencing negative samples or with no diagnostic yield were put up for a whole genome sequencing. And what he suggests is that a whole genome sequencing provides an additional almost 12% diagnostic yield in cardiac channelopathies compared to whole exome negative, compared to whole exome sequencing. Now, that is really, really significant because a significant number of genetic diseases, while they are currently accessing whole exome sequencing, might have been missed out because of the genetic variations, which are typically not captured on whole exome sequencing. The other application is to, for example, look at intellectual disability. And in fact, intellectual disability, the genetic causes come in multiple sizes and forms. It could be from chromosomal abnormalities, it could be copy number variations, it could be point mutations, it could sometimes even be repeat expansions and subtelomeric rearrangements. Now, there are different kinds of tests today that are being used to look at each of these kinds of genetic diseases. Chromosomal abnormalities typically on chromosomal karyotyping or FISH, chromosomal copy number variations on a chromosomal microarray on RACGH, point mutations on a panel or exome sequencing, and subtelomeric repeats on long read sequencing, so on and so forth. Now, in fact, we have been following last week, the American Academy of Pediatrics has changed the guideline, and today whole genome sequencing is included in the tier 1 testing for global developmental delay and intellectual disability. That's largely because of the fact that many of these patients go through multiple tests and lose considerable amount of time before they end up in a diagnosis. And even that, they're not really followed up when they turn up negative on one or the other genetic testing. And whole genome sequencing, therefore, becomes a very attractive proposition because it covers all the kinds of genetic changes or variants that we currently know about. Now, on one end of the spectrum, whether you look at chromosomal abnormalities, structural variants, repeat expansions, small insertion deletions, and similar variations, it's all covered by whole genome sequencing in a single test. That means that instead of going through a triage testing or a sequential testing, where you do one or more of these tests, each of them having its own turnaround times, its own complications, you could now do away with it with a single whole genome sequencing, making it much faster, cost effective, and timely. Today, you can actually get a routine whole genome sequencing in less than 14 days. And of course, there are rapid options that you will hear in the talk today, and how we can really reduce it to clinical turnaround times in approximately 48 hours. So whole genome sequencing, therefore, can put an end to the diagnostic odyssey of patients and families, and more importantly, can avoid spending additional time and effort in doing multiple different tests, because every late diagnosis is a lost opportunity in treatment, because at least some of these genetic conditions today are treatable if they can be diagnosed very early on. Now, the third application of whole genome sequencing is to understand what is really prevalent. Now, this is a really big problem in clinical medicine, especially for rare genetic diseases, because if you put in a cardiologist, a neurologist, and a pediatrician or a nephrologist into a room, they will keep fighting on what is really prevalent in the population. And national rare disease policies cannot be built unless you understand what is really prevalent. And prevalence estimates today largely come from clinical settings. And clinical settings, the prevalence is largely because of they tend to diagnose that condition much better. And if you tend to diagnose and treat a condition much better, then you tend to also see a lot more patients of that particular condition. So if you have a cardiologist who can understand cardiomyopathy much better, then practically all cardiomyopathy patients tend to go to that particular clinician and get care there. And therefore, there would be an inflated prevalence estimate for cardiomyopathy in that clinical center. And most of the other people who do not diagnose cardiomyopathy would never, therefore, record a cardiomyopathy, and therefore that would be lost in numbers. Now, to me, the only way to look at an absolute prevalence of a genetic disorder is to be able to sequence apparently normal people. The idea is very simple. You have two pairs of each of the chromosome, and you might be a carrier in one of the pair, because you have a genetic mutation in one of the pair of the chromosome. And if you can estimate the carrier frequency, and you pretty much understand the marriage patterns in a particular community, very specifically in India, because people in the communities tend to marry within the same community, either by caste or creed or religion. And given these numbers, you should be able to estimate what is the prevalence of a genetic disorder. And if that is very different from what clinicians see, that means that there's a gap. And this gap largely can be filled in by providing them the right diagnostic and enabling them to diagnose the patients much better. Now, to do it, we need to have a population genome program. And thankfully, in 2019, with Dr. Sridhar, my colleague and collaborator, will also be speaking in this course, we created Indigen, one of the first population scale pilot genome programs in India. And the idea was to essentially look at prevalent conditions which can advise policy. And for example, let us look at treatable intellectual disabilities. Treatable intellectual disabilities encompass around 80 odd genetic disorders. There is newborn screening programs, but the newborn screening programs are based on prevalent disorders elsewhere in the world. Now, we could look at the population scale data for around 1,000 odd individuals from cosmopolitan Indians to understand what is really prevalent in India in terms of treatability. And what we found is that the most prevalent treatable genetic disorders are not the ones that for which newborn programs exist. For example, Wilson's disease, phenylketonuria, biotinase deficiency are almost 76 times more common in Indian population compared to the Western population. We don't have a newborn screening program for any one of these except phenylketonuria. Certainly, some states have introduced phenylketonuria in the newborn diagnosis program. For example, Wilson's disease, the carrier frequency in the population is nearing almost 1%. That means 1 in every 40,000 odd children would be born with Wilson's disease. In India, we have 25 million births every year, and then you can pretty much understand what is the number and burden of Wilson's disease today we are missing. And this is a treatable genetic condition, not with a 70 lakh rupees drug, but probably a 3,000 rupees drug every month. Now, of course, while we speak about genomics, what we need to keep understanding is that the cost of genomic sequencing is dwindling very rapidly. And this would largely mean that we are at the verge of WGS being used as a discovery or a research application to a routine clinical test. Just because it is being affordable, it is also being accessible, and more importantly, it provides a wealth of information that can be used in clinical settings. I talked about wealth of clinical information that can be correlated for rare genetic diseases. In the next set of slides, I will also prelude onto how this could be used in apparently normal individuals. Now, for that, we need to understand how clinical medicine works today. Now, clinical medicine works today because on the basic premise that you can train doctors to understand clinical phenotypes. The patient would come to a doctor. A patient would come to a doctor because he or she has a problem in the first place, or a disease, or an illness, or an ailment in the first place. And therefore, the doctor elicits the history, signs, and symptoms, arrives at the diagnosis, offers some labs to confirm the diagnosis, and genetics is today offered at the end of the spectrum. That means once a diagnosis is made, genetics is today used to confirm the diagnosis. And that really is the problem. Because there are very, very few people who would now follow this entire funnel of operations to be able to arrive at a diagnosis. And a molecular diagnosis, therefore, is offered to only a very small, minute fraction of patients who would otherwise would have benefited from genetics. Now, assume that genomic sequencing is cheap, affordable, and accessible, and assume that people could get their genome sequences, and which is possible today. You could now look at the genome to understand genetic variants which will cause genetic diseases by virtue of their pathogenicity, and there is databases and evidence on how to classify variants as pathogenic. And using this information, you can preemptively treat the individual and prevent the individual from developing the disease. And I think that is a real paradigm shift that is going to happen. Because in the earlier approach, the patient has to develop the disease before the patient needs to go to a doctor. And only a very few lucky ones get to the right doctor to get the right diagnosis and the right treatment. But now, for example, if you could predict a genetic disease even before the disease has manifested, now you provide an opportunity to treat individuals. Now, if you think that this is something rocket science, this is not really rocket science, because that's how the clinical medicine works. Now, all of clinical medicine starts from the individual arriving at a clinical center with the start of symptoms and signs. And anything before this is what we call as screening. And anything after this is what called as diagnosis, prognosis, treatment, and monitoring. Now, the real idea is, can we really push this needle to the left? Or in other words, can we diagnose individuals early on at a molecular level, at an accurate, precise modality, and be able to prevent the onset of the disease? Now, we have numbers to suggest why this is more important. If you sequence 100 odd individuals in the population, approximately 5% of individuals today would have an incidental and medically actionable genetic variant. That means that 5% of individuals, or 1 in 20 individuals in the population, has a molecular defect in their genome. And with this molecular defect, he or she will manifest a condition, but the condition today is treatable or can be preemptively managed. Now, to put numbers, half of this is of cardiovascular disease and half of this is related to oncology. In fact, around 2% of people would have a hereditary cancer predisposition syndrome, BRCA1, BRCA2, colorectal cancer, so on and so forth. And another 2% would have for cardiovascular diseases, 3 disorders, cardiac channelopathies, cardiomyopathy, and familial hypercholesterolemia. And all of these are treatable, manageable conditions or amenable to early diagnosis. Now, that is a significant number today on today's evidence, and this evidence is increasing ever rapidly. And in fact, if you have been following the ACMG secondary findings list, have been improved with another 3 more genes last week. Now, early diagnosis and treatment is not something new. People have been doing, and this is called newborn screening. But newborn screening is largely based on metabolite testing. Now, we have an opportunity to convert this into a genomic testing, and we will have a full session today on what is genomic newborn screening. Now, the idea is very simple. Can we change from a 37 condition newborn screening, which is one of the most broad tests that is done anywhere in the world, in the US, with a diagnostic positivity of approximately 1 in 300 odd individuals, to convert it into a much broader test using whole genome sequencing with a diagnostic positivity of approximately 1 in 182 newborns, with many of them being treatable genetic conditions. And more importantly, genomes are for a lifetime, because your germline genome is constant from the day you are born to the day you are dead. And at different points in time, you can ask very different questions. When you're a newborn, you would want to look for treatable causes. You would want to look for cystic fibrosis, primary immunodeficiency disorders, neuromuscular disorders, metabolic disorders, which can be treated. Much later on, you could look for cardiac genetic disorders, muscular atrophies, neuromuscular disorders, hearing loss. Some of them are treatable. Some of them are increasingly being treatable because drugs are in clinical trials. And early adulthood, you could look for example, maturity onset diabetes of the young. Most of the patients of MODY are misdiagnosed, underdiagnosed, and lost to the system. You could look at family hypercholesterolemia and cardiac channelopathies and be able to treat individuals accordingly. And of course, when you're planning to get married, you could use this genomic sequencing for carrier screening and reproductive decision support. And much later on, you could look for oncology and neurodegenerative disorders. And more importantly, not just the diagnosis of the disease, genome sequencing can enable precision therapy through pharmacogenomics. You have an entire session on pharmacogenomics and how to go about a whole genome pharmacogenomics. I'll stop there, but let me summarize by saying that genome sequencing is becoming affordable for routine clinical implementation, and it is all genetic tests put into one and probably more in terms of the diagnosis, and it can also enable precision treatment through pharmacogenomics. Genomic sequencing at an individual level for a patient can enable disease diagnosis, and at a population can implement and enable public health decision making. Genetic diseases are prevalent in the population, many of them being undiagnosed and underdiagnosed, and almost 1 in 10 individuals do have a genetic disease, half of it which is treatable, or approximately 1 in 20 individuals do have a treatable genetic condition, which you can step into a doctor's office, and the guidelines today available on how to enable early diagnosis, early management, and some of it early treatment. And therefore, whole genome sequencing is not just for patients, it is also for apparently normal, healthy individuals, including probably you and me. And genomic evidence and genomic sequences for a lifetime, the evidence might change over the years. Thank you very much, and I will take questions. If you have questions, you could put it in the chat box, and I'll be happy to address them. Yeah, so there are some questions that have come in. I will quickly take them.
Yes.
The question from Dr. Tanudatta is, are we able to completely understand the non-coding variants? The answer is no. For most of the non-coding variants, we don't even know they exist, because until very recently, whole genome sequencing was never a mainstay. And exome sequencing typically understands only the exonic variants, or around 2% of the genome. With whole genome sequencing, we are increasingly understanding the non-coding variants, and we are also increasingly understanding the functionality of these non-coding variants. And of course, there's a long way to go, but the way is quite very well set, and there are methodologies to do this today. In India, do they legally allow to check genomic mutations of apparently asymptomatic child in affected families till the child legally becomes an adult? Yes, in India, you can check a fetus or a newborn as long as the parents do consent to the testing. Could you please share a bit more about predicting clinical diseases through genomics? Okay, I'll give you one good example. One of the most prevalent genetic conditions which is underdiagnosed is uh, cardiac channelopathies and cardiomyopathy. Now, if you have cardiac channelopathies and cardiomyopathy, very often you do not get diagnosed because most often the first clinical symptom of this disorder is a cardiac arrhythmia ending up in sudden cardiac death. Now, most people would have found in news apparently young, healthy individuals who would have been in active life, either marathon runners or in a gym, suddenly collapse and die. And that collapse and die because of sudden cardiac, there's a condition called sudden cardiac death, is largely because of the fact that they develop an arrhythmia or an abnormal rhythm of the heart, and the heart stops, ending up in arrest, and they die. Now, people with cardiac channelopathies and cardiomyopathy should not take up such strenuous exercises because it is very well known predisposing factor to developing arrhythmias. Now, since you don't diagnose them and you don't diagnose them post-mortem also, because post-mortem of sudden cardiac death is not something that is routinely done, you typically do not diagnose these individuals. But population scale prevalence estimates suggest that approximately 2% of individuals would suffer from these two conditions put together, cardiac channelopathies and cardiomyopathy. Now, a genetic test can identify people at risk and then modify their lifestyles, their treatment, and including putting in a pacemaker, and that can provide them a very normal life going forward. So I think I I explained how we could go about with that. Yeah, Dr. Muralidhar has a question. Is WGS valid for a lifetime? What mutations happen after that? What if mutations happen after the test? Yeah, very valid question. Your germline genome is constant from the day you're born to the day you're dead. Cell divisions could accumulate additional genetic mutations, and these are somatic mutations which are not inherited and cannot be transmitted to your offspring. Typically, cancer is the best example where you develop a somatic mutation during cell division, and these somatic mutations could be localized to very small number of tissues. So they do develop during the lifetime of the individual, and they need to be handled separately. What we're talking about today is inherited genetic mutations or germline genetic mutations, which are constant from the day you're born to the day you're dead. What is the cost of a whole genome sequencing for a patient in India? I will not put it here, but probably I can connect back later. All participants in this course also has an opportunity to get their whole genome sequencing done. After this course and after the exams, we'll probably connect with them. Wouldn't it be advisable to do a cardiomyopathy genetic test in a postmortem of all cases with sudden cardiac arrest with no known cardiac issues? Absolutely agree and absolutely love that idea, but provided um, who's going to do the test? Because most of the patients with sudden cardiac death are never ever going to even meet a clinician, because the first instance they're going to die, and most families are not aware about the prevalence of this genetic condition. And of course, if the genetic testing is done, it is very, very more important, being an autosomal dominant condition, to advise additional family members of the risk and avoidance of some physical or lifestyle measures that can prevent sudden cardiac death. What is the additional yield of gene deletions in genome-based carrier screening? Approximately around 10 to 15%. So in some cases, up to around 20% if you follow the literature. If you have WS negative but high clinical suspicion of primary immunodeficiency, would it be the next step? Would WGS or clinical exome or higher depths of WS? Now, depending upon the clinical scenario, if you suspect a somatic mosaic variant, then you need to have higher depth. But in most cases, we also have done quite some work on primary immunodeficiency disorder. Most cases are diagnosed not on a somatic test, but on a whole genome sequencing. just because there are genetic variants and types of variants which are typically not covered in whole exome sequencing, and typical additional diagnostic yield, as I mentioned before, is approximately 15 to 20%. Yeah, if there are many more questions, I could, of course, address them very specifically. We could go on with the next session.
That's just great, sir. Thanks a lot for enlightening us. This was really a wonderful kickstart to our session, sir. And a lot of questions that are actually clinically relevant and actionable from the audience is very happening, and it is very wonderful to see these questions coming ahead. Thanks a lot for asking the questions. Keep them coming. Post the sessions as well. We are delighted to have you all here. So let me introduce the next upcoming speaker for the upcoming session on genetic variants, types, and evidence by Ms. Ramya Venkatesha. So this is Ms. Ramya Venkatesha is a skilled and detail-oriented bioinformatician. She's a part of our team. She specializes in germline whole genome sequencing, clinical reporting. She holds a bachelor's degree in biotechnology from Bengaluru University and a master's in bioinformatics from Manipal Academy of Higher Education and Research. Prior to joining Karkinos, she did research work at the prestigious IISc, Indian Institute of Sciences, Bengaluru, where she further honed her analytical skills. At Karkinos, Ramya plays a key role in the germline reporting team, contributing to high quality clinical interpretation and variant curation workflows. Known for her creativity, precision, and a sharp eye to detail, she excels at identifying clinically relevant variants in accordance with ACMG AMP guidelines. Her contributions are instrumental in ensuring accurate clinical actionable reports that impact patient care. Ramya's passion for genomics, combined with her technical rigor, makes her a valuable asset to the clinical genomics ecosystem. Wonderful. Please welcome to the stage, Ramya, and please share your screen. Let's get started.
Kind introduction. Okay. I just hope my screen is visible. So let me begin. A very good morning to everyone. I'm Ramya Venkatesha, and I will be speaking a few words on genetic variants, types, and evidences. Now, what is a variant? Let's go back to the basics, shall we? To put it in simple terms, changes in the DNA sequence with respect to the reference genome. So fascinatingly, in our case, 99% of our genome is the same. Only that 1% makes us different from who we are, especially the minute changes such as eye color, nose shape, height, and other such characteristics. In this part of 1%, there are also certain variations that can lead to genetic disorders. In this case, you can compare it with the reference genome. We have substitution, insertion, and deletion has taken place. Now, what these terminologies are, I will explain in the further slides. So variants can be broadly classified into 5 types. SNVs, that are single nucleotide variations. Small indels, also called as either frame shift or non-frame shift or in-frame mutations. Splice variants, that are splice acceptors or splice donors. Structural variants or CNVs, copy number variations, and non-coding variations. So these are some of the variant types we can encounter in a WGS report. So SNVs or single nucleotide variants, as the name suggests, it is a change in position of a single nucleotide that causes a change in the protein function. So think of it in this way. You are playing a piano and a change in the single key can affect the song. So single SNVs can mostly be of three types: synonymous, missense, and nonsense. So synonymous is a type of change where the change in the nucleotide does not cause the amino acid to change. So in this case, you can see our normal sequence is CAG, CCC, and ACT, which the corresponding protein change is glutamine, proline, and threonine. What happens in a synonymous case is CCC is getting replaced or is getting mutated to CCA, and both these codons code for proline. So the amino acid change remains untouched. Hence, they do not have any effect on the protein. In missense, the change in the nucleotide affects the amino acid change. So here CCC is getting converted to CAC, and CAC codes for histidine, which will in turn affect the entire protein structure and function. These variants can also lead to the formation of what we call an abnormal protein. In nonsense variants, a change in the nucleotide creates a stop codon, leading to the truncation of the amino acid chain. So in this example, CAG is getting mutated to TAG. So TAG, if we all are aware, it is a stop codon. Hence, no protein product is formed since the stop codon is present in the beginning part of the sequence. So this leads to a premature protein product formation. So hence leads to a non-functional protein. Coming to our next classification of variants which we may encounter are small indels. So small indels, unlike the single nucleotide variations, one or many nucleotides may be deleted or inserted. So this can happen in two different scenarios. Frame shift, this type of indel causes a shift in the reading frame of the gene. So reading frame is like reading a sentence, but we read it with letters in groups of three. In this example, our normal sequence is again CAG, CCC, and ACT. A T is getting inserted after G in this particular case, so which causes the frame to be read differently. So now we will have to read the frame as CAG, TCC, and CAC. And if we are all aware, so we have codons are read in groups of three. So even our protein chain will get impacted. So in this case, glutamine, proline, threonine gets converted to glutamine, serine, and histidine. And again, this can lead to a non-functional protein. A non-frame shift are indels that do not alter the reading frame of this gene. So in this case, CCC is being deleted and TTT is being added in. And so although it removes the amino acid proline, the reading frame is not being changed. So depending on the scenario, this can lead to a relatively functional protein. Our next class of variants are splice variants, and these can be characterized into splice donor variants and splice acceptor variants. So splice variants, also known as alternative forms, are different mRNA transcripts are produced from a single gene through the process of alternative splicing. The process includes a differential inclusion or exclusion of the exons. The coding regions of the DNA during the splicing of pre-mRNA into mature mRNA. So to make things little easier, imagine the DNA is a huge story with a lot of irrelevant things. So what happens here is the spliceosome identifies these important parts called exons and cuts out the unnecessary introns. The spliceosomes then joins the exons together, creating a shorter, more polished version of the RNA called the mRNA. However, there is a twist. Spliceosome has some flexibility in choosing which splice sites it can use. It can sometimes skip an exon, removing it along with the introns. This leads to different combinations of exons being joined together in the final mRNA. So we have what we call the 5' and the 3' splice sites. These are specific locations on the pre-mRNA molecule where the splicing occurs. As a splice donor or splice acceptor variant, so it refers to a change in the DNA sequence which alters the splice donor or the acceptor site. So depending on where the splicing is happening, it is called the splice acceptor or the splice donor site. So splice donor variants, they are typically found on the 5' end of the intron, that is the beginning of the intron. And I have just taken a relative example. It is typically GU in the eukaryotes. It signals a start of the intron and where to make the first cut. Splice acceptor is found in the 3' end of the intron, that is found at the end of the intron. Typically AG in eukaryotes, it signals the end of the intron where to make the second cut. So splice variants can have the following consequences. It might change the reading frame or it may lead to exon skipping, which again may cause some changes in the report. Coming to the structural variants or copy number variants. So these are types of variants where solid chunks of DNA, that is a large number of base pairs, are either inserted, deleted, or duplicated or translocated. The minimum number of base pairs for it to be classified as structural variants are greater than 50 base pairs. They are of 4 types: deletion, insertion, duplication, and translocation. The minimum, the blue lines here represent the several nucleotides of a sequence. I wanted to represent a huge base pair of nucleotides, so to better visualize the concept. So let us understand that these are chunks of sequences. Now let's look at deletion first. The black box that is highlighted, so the chunk of nucleotides you can see are getting deleted. Insertion, the chunk of DNA is getting inserted in between a sequence. Duplication, the segment of DNA is getting duplicated multiple times within the sequence. And lastly, translocation. This mainly occurs between 2 different chromosomes. So I have labeled them as red and blue respectively. So the black box here represents the segments of DNA that is being exchanged between the 2 chromosomes. Hence the term translocation. This sort of variants are generally how fusions are created. So the last class of variants are the non-coding variants. These are intronic. These can be of 2 types: intronic variants and intergenic variants. Now, what are non-coding variants? So non-coding regions of DNA are the other parts of DNA that do not provide instructions for making proteins. This actually surprisingly includes a vast amount of our genome. So non-coding variants are changes in the DNA sequence that occur in the non-coding regions. They are classified based on the location of the variant. So intronic variants are generally found or located within the gene. So here I have represented it with a purple color. You can see that it is located between exon 1 and exon 2 of the red colored gene. That is gene 2. Now the question may arise, why are intergenic variants and intronic variants different? So for that we need to understand that intronic variants are a part of the gene, whereas intergenic variants are the mutations found between a gene. Now that we have learned about the different types of variants, ever wondered that the variants you see in a report vary a lot, right? It is very different in terms of notation. This is because we use a standard convention and we name them using something called as the HGVS nomenclature. Now, what is HGVS nomenclature? Let us take a few steps back before the HGVS was introduced. Before HGVS, there was a patchwork of different ways to describe mutations, making it difficult to compare results between studies or clinical labs. So different labs used to represent variants in different ways. For example, you can see lab 1 is having a BRCA mutation with codon 185A to G. Lab B says it is a BRCA1 mutation again, but the position is a 555. It's at position 555, and substitution is A to G. Lab C is saying BRCA1, but the nucleotide change at 185 A to G. These may all look like they mean the same things, but with the increasing amount of discovery of the variants and clinical data on them, the need to standardize a single format globally became a priority. So a program or an association society called as HUGO, Human Genome of Variation Society, comprising of a group of scientists, established the rules and regulations for annotating a variant. Now let us go through some rules for HGVS nomenclature. So the first thing you should notice is it always starts, our variant is always starts with a reference sequence. They can be of multiple times, but the most commonly used are the NCBI IDs. This is followed by the colon or a separator. Then based on the sequence type, we either have a G dot for the genomic DNA, C dot for the coding DNA, or P dot for protein. The NCBI IDs also vary accordingly. NC for G dot, NM for C dot, and NP for P dot. Now, according to the different variations, the symbols can also vary. So for silent mutations, we have an equal to symbol. For deletion, we have a DEL. For a duplication, we have a DUP. For a frame shift, we have an FS. For intronic, we have a plus or minus according to where the position of our mutations are. So again, this may all look very complicated, hence why we have kept different specific scenarios to better understand how this works. So this is how you might encounter variants in a WGS report. So starting with a BRCA, so that is the gene name, and it is a well-known tumor suppressor gene associated with increased risk of breast and ovarian cancer, followed by a reference sequence ID and version number. So here the NM007294.4, this is the reference sequence accession number. NM indicates the mRNA reference sequence followed by a unique identifier. .4 is a version number, so this indicates that this is the fourth revision of the reference sequence. So missense variant, this is clearly a missense variant because a single nucleotide change is happening. Then we can look at the chromosome number and locus. So 17 here indicates the variant is located on chromosome 17, and the number following that 4304567 specifies the exact genomic position or the locus of the variant on chromosome 17. So this information especially is very important in terms of when we are looking for hotspots in the region. So this is our C dot. So at position 5266 in the genes code, the letter C has been replaced by the letter T. So cysteine has been replaced by thymine. And if we have to look at the protein change, what is the consequent protein change? At position 1756, the protein block glutamine is changed to TER. So a stop codon has been inserted, leading to a formation of a premature protein. Coming to our next example, again, it's a very similar gene we are using, BRCA2 in this case. It is a frame shift variant, which means the DNA sequence is getting altered. Again, we have a chromosome number with locus. So here it is 13 chromosome and followed by the locus. The C dot here, 5946, is the position in the code, and del T means that the T, that is thymine, is being deleted. The resulting protein consequence is a frame shift of the amino acid serine at position 1982, and it's been changed to arginine. So a frame shift of about 22 nucleotides. Indicates a stop codon occurs 22 amino acids after the frame shift. So this means that at position 1982, serine is being replaced by arginine. And because the sequence is being altered, the stop codon is happening 22 amino acids later. Few more examples. So we have an MLH gene. This is involved in DNA mismatch repair. And we also have, this is a splice site variant, so it indicates that the variant affects a splice site, which is crucial for gene splicing. We also have a Dellins variant. This is a type of variant where GC in the reference sequence is being removed and is being replaced by TT. So these are some of the examples we may encounter in a WGS report, and as complicated as it sounds, it's a very essential concept. So now that we have learned about all the different types of variants, and let us try and understand an interesting case study on how the different types of variants are useful, specifically in WGS. So, Code Red, a variant unmasked, is a story of a 7-year-old patient. Born out of a first degree consanguineous marriage, he lost an older sibling during neonatal period, and the provisional diagnosis after health checkups was given as Jervell Lange Nielsen syndrome, that is associated with prolonged QT syndrome, syncope, and hearing loss. Now, a WES test yielded a negative result. So next, a WGS, that is a whole genome sequencing, was performed. Surprisingly, the WGS, due to its versatile sequencing that covers everything from coding regions to non-coding to UTRs, identified a novel mutation, approximately 9.2 kilobases of deletion in the gene KCNQ1, in whose clinical phenotypes matched that of the patient. So the young boy with such scary cardiac symptoms, thanks to WGS, was able to get the right diagnosis. To summarize, the notations vary across genomic landscapes. So if the variant ID is found in genome, it's denoted as G dot. In the coding DNA, it's called C dot. And if on the protein, P dot is used. But this would be my takeaway message to you. The variations can happen anywhere in the genome, coding, non-coding, or UTR. The key is to look at the whole story and not just a few chapters. And with this, I would conclude my session. Thank you. If there are any questions, I would be gladly able to take them.
Okay, a few questions are rolling in, as I can see. Uh, so one question is from Gyanesh P. Are SVs and CNVs reliably picked up on WES?
Okay, can you please repeat the question?
Are SVs and CNVs reliably picked up on WES?
So to answer that question, Gyanish, I would say SVs and CNVs are not as reliably picked up from WES because their most focus is not on the large deletions insertions that happen, and also you need to have that amount of coverage, I would say. So a WGS would be better.
Okay. And also a follow-on question on top of it from him is, are intronic variants important?
So actually, I would say intronic variants can either have a splicing consequence or not. So it would completely depend on the type of location it is present in. So it is definitely contextual, but I'm unable to think of any sort of example right now. But I can tell you, yes, intronic variants can be important depending on the location at which it is placed.
Okay. This is another question from Mohammed El Bagori. How to make accession number of sequence on GeneBank? No, this is not too related into this, but yeah.
Yes. So this is something I might have to have a very
Sure.
detailed discussion about. How to? Can you please?
Sure. How to make accession number of sequence on GeneBank?
So this is something which I have not yet really looked into, but I would say probably Gene Bank would definitely have a option to submit the sequence, and I think according to me, if I'm not mistaken, that accession number is clearly automatically generated.
Yes. Wonderful. So the next question is, can we use WGS to diagnose HB structural variants in hemoglobinopathies?
Yes, I would agree. I'm sure we can definitely diagnose
Okay.
hematological disorders, and it can detect any large SNVs.
Okay. And Dr. Emily is telling it's a very wonderful presentation. Thank you, ma'am. Uh, and the next question is from Shrikant Raj. The question is the variant found in the boy, the case you discussed, is
Yes,
it pathogenic or VUS?
it was definitely a pathogenic variant. It was definitely a pathogenic variant, and it matched the clinical phenotype of the patient exactly. So
Okay,
actually, it was quite a relief that the boy was able to get the right diagnosis.
that's just wonderful. And another question is from Kusumita C. Can you share a bit more about Intel non-frame shift variants, please?
Uh, okay. So indels and non-frame shift variants, right?
Yeah.
Okay, so let me start with non-frame shift variants. So non-frame shift variants does not alter the reading frame of the gene. So what happens in this case is you either have an insertion of a multiple of three or a deletion of a multiple of three. So this is the only way the
Mhm,
reading frame does not shift, right? And in terms of
mhm,
in terms of functionality, again it definitely varies on what amino acid is being replaced or at what position. So it's very tough to
mhm.
say, but generally in-frame deletions are not as I would say harmless as compared to a frame shift which alters the entire protein sequence.
Great. I'll just put up two more questions and we'll move on to the next session speaker. The next question is from Dr. Naresh Tayade. The question is, can newer WES may not, I guess there's a problem in the sentence formation here. But the question in simpler terms is, can the newer versions of the WES that come up, would they be able to pick up structural variants in WGS?
If your question is like, can a newer WAS pick up structural variants? I would say the answer is no,
No.
because WAS by its nature is a capture-based technique, which means it is able to sequence and span only the captured regions of the genome. So we cannot by confidence say it was a deletion by the WES or an insertion. You do need a WGS to accurately identify structural variants.
Okay. The last question is from Elizabeth Anthony. The question is, are intragenic variants more likely to be pathogenic compared to intergenic ones?
Are you saying are intragenic variants most likely to be pathogenic
Be
than
pathogenic?
intergenic variants?
Yes.
I would say, according to me, I think intragenic variants are most likely to be pathogenic, but because there are not much context to this, because again lot of things will depend on the location of where the gene is located, what type of gene, but my answer would definitely be intragenic variants.
Okay. Wonderful. Wonderful. That's really great to hear from you, and thanks a lot for enlightening us with your cheerful spirit as well as great information as usual, Ramya. Again, so one of the best things about Ms. Ramya here is her ability to break down complex things into simpler ways. Thanks a lot for the wonderful session on genetic variants, types, and evidence. Moving on
Thank
to the next session. Yes, thank you, Ramya. Moving on to the next session. The session is on what is in a WGS report. The session is by Dr. Bani Jolly from our Karkinos team. So, a quick intro about Dr. Bani Jolly. Dr. Bani Jolly is a senior scientist in genome informatics at Karkinos Healthcare, specializing in translational genomics and genome informatics. Her work integrates informatics and genomics to advance our understanding of infectious diseases and inherited disorders, with a focus on germline variant interpretation and whole genome sequencing. She played a pivotal role in shaping India's SARS-CoV-2 genomic surveillance, working closely with the World Health Organization to strengthen outbreak response capacities across Southeast Asia. Dr. Bani also led the development of the widely adopted GenePy Box toolkit, empowering global researchers in outbreak analytics. Beyond infectious diseases genomics, she has contributed to landmark NGS projects including Indigen, India's first 1,000 genome initiative, and Guardian, India's largest clinical genomic rare disease network. A PhD in computational biology from CSIR IGAB, she has over 30 peer reviewed publications, believes in communicating simplified science to all, and regularly contributes to the communication pieces to the Hindu. So, this is wonderful to have Dr. Bani here. The stage is all yours.
Thank you so much, Harish, for that very, very kind introduction. Good morning, everyone. Very pleased to have you here with us today. So I'll just start presenting my slides. Okay, so good morning, everyone. So in this session, we are going to dive deeper into what a whole genome sequencing or a WGS report really contains and how we can interpret its contents in a clinical context. We have seen from the previous sessions how WGS is important, how it is growing, and what are the different kinds of variations that can be explored, that can be detected through a whole genome sequencing test. But given that the most confusion lies that since WGS is a very broad coverage test and it actually detects a lot of variations in the genome, a lot of different types of variations in the genome, it does lead to a little clinical confusion about how to really go about it. So in this session, we'll specifically explore the different categories of findings and how they might matter and how they might be included in a WGS test report, which is in context a clinical WGS test report. So let's begin. So as we discussed in the previous sessions also very extensively, whole genome sequencing is no longer a last resort test. It is rapidly becoming the first line of investigation in many scenarios, from rare disease diagnosis to cancer genomics and newborn screening and even other applications, including population studies. Its ability to detect not just single nucleotide changes, but also copy number variations, structural variations, things like mitochondrial variations and pharmacogenomic markers makes it a very versatile test and actually a very unparalleled tool in genomic medicine. We have also seen that the quality of being a single genetic test in one makes WGS so powerful compared to other genomic tests. It provides uniform coverage across coding and non-coding regions, enabling us to go beyond exomes. It identifies non-coding variations which could affect regulatory regions, splicing regions, or even structural integrity of our DNA. And it also allows you to look for structural rearrangements and mitochondrial variations in the same dataset that we have generated through one single test that can be done on the same sample. All of which would otherwise require multiple tests and in extension multiple turnaround times that the patient will have to wait for. So this multi-dimensional advantage is why WGS is now being adopted across different use cases, starting from oncology to rare disease diagnosis, pediatrics, and even screening of healthy individuals. So what exactly does a whole genome sequencing report include, or what are the types of things that a whole genome sequencing report can include? At a high level, it spans across several different categories. First, we can have clinically relevant germline variations, which are basically pathogenic or likely pathogenic variations that can explain the phenotype or the symptoms or disease or the disorder that a patient or a person is presenting with. These clinically relevant findings can also be relevant in case somebody has a past family history of a particular disorder in their family. Then we have carrier status variants, which may not affect the individual but could be very relevant for family planning. Next is pharmacogenomic markers, which can guide drug dosing and safety for all patients and healthy individuals. We can have structural variations, including deletions, duplications, rearrangements that can be reported as a part of the clinically significant findings. Additionally, we can have secondary findings as per ACMG recommendations. And lastly, we can have mitochondrial variations, which are especially important in cases of suspected mitochondrial diseases, which in essence span multiple systems in your body. And all of this is embedded into one single comprehensive report, and we'll talk about how each one of these gets evaluated individually. So starting with phenotype-driven or clinically relevant germline variations that can be detected through whole genome sequencing. So these are the cornerstone of most diagnostic WGS efforts. In this, we are looking for variants in genes that match the patient's phenotype and explain the disorder, especially that those variations that are classified as pathogenic or likely pathogenic using standard guidelines for reporting a variation as clinically significant, such as those mentioned by ACMG. But remember, interpretation is not just about a variant, it is about what fits with the clinical symptoms, inheritance pattern of the disorder, and the zygosity of the variant that is detected in the patient. So often this section will answer the primary clinical question for a diagnostic WGS test and will thus form the basis for future medical management for the patient. So to look at this particular component of the report in a deeper detail, so the phenotype-driven variant findings are one of the most important sections of a WGS report because these are the findings that directly explain the clinical presentation of the patient. So these are basically variants in genes that match the phenotype or the symptoms of the patient. And here we are typically looking for pathogenic or likely pathogenic variations that are consistent with the inheritance model and clinical features. Sometimes we may also report high confidence variants of uncertain significance in this section, especially if the gene is well matched to the phenotype and there is some supporting evidence. In terms of format, these findings or these variations can appear under headings that can go by names like primary findings, diagnostic findings, significant genomic findings, and so on. The key fields to pay attention to should include the gene and the transcript of the variation in which the variation lies, basically as explained by Ms. Ramya in her previous slide, that it is very important to denote the gene and the transcript in a very standardized notation. And thus the report should also contain the consequence the variant has on the gene function, meaning that is it an intronic variation, is it a splice site variation, is it an exonic variation, is it something that spans the intergenic locations or the UTRs, or is it something that lies in the non-coding region. Next, we should have the actual change that happens at the at the genome, cDNA, and protein level that can be denoted through the G dot, C dot, and P dot HGVS nomenclatures that were explained to you in the previous session. Then, very importantly, the report should also have zygosity and coverage for the variation that has been detected. along with the associated disorder of with the gene, along with the inheritance pattern that the disorder follows. And finally, the ACMG classification for the variant along with the evidence codes. I don't want you to worry about what is ACMG classification, what is evidence codes, and what is clinical interpretation of a variant right now, because these will be very extensively discussed in the next sessions as to why these should be done and why are they very important to do so. So in this this particular section of the report, of a WGS report, is something that most clinicians will go straight to. So clarity, accuracy, and alignment with the phenotype or the clinical presentation of the patient is something that matters a lot here. So let's, for example, look at a case to bring this concept to life. So, for example, we have an 8-year-old girl presenting with neurodevelopmental delay, global developmental delay, dysmorphic features, and encephalopathy that a doctor has screened in his clinic. So, a WGS or a whole genome sequencing test was ordered for this particular patient, and it revealed a missense variant in the CACN1AC gene. Specifically, if in terms of HGVS nomenclature we speak, it was a C.959C2G substitution, or at the genomic level, it is a substitution at chromosome 12 at position 2493232, which leads to a amino acid change of THR322 ARG. The zygosity of the variant that was detected in the patient is heterozygous, with the coverage being stated as 32x. So now why does this matter? Because CACN1 gene is linked to Timothy syndrome, an autosomal dominant condition that aligns very well with her phenotype. The variant, according to ACMG attributes, is classified as likely pathogenic, indicating that functional evidence, the variant being rare in healthy populations, and there is a prior known association of this variation to this particular disorder. So this is an example of how a phenotype-driven finding can directly explain a patient's condition and support diagnosis and management. And let's break this example down into a bit more detail, because it brings together multiple different concepts that are key to interpreting a whole genome sequencing approach. So first you see coverage 32x. What does this actually mean? So this means that the genomic position where the variant was found was read at least 32 times. So when we sequence a DNA or sample, or we sequence a genome, the machine does not read the sample provided in just once. To increase the accuracy that the base that is being read by the sequencer is actually correct, it is read multiple times by the sequencer. So for a whole genome sequencing test, a read depth of 32x is a strong number, because 30x is considered as a clinical gold standard universally. And this 32x coverage gives us high confidence that the variant call is actually accurate. Low coverage at a variant position might make us question whether the variant is real or it's just an artifact or something erroneously detected by the sequencer. Next is the zygosity, which is for this patient heterozygous. This tells us that only one of the two copies of the gene that the patient holds carries the variant. And this is important when we need to link this information to the inheritance pattern of the disorder. In this case, Timothy syndrome follows an autosomal dominant inheritance pattern, which means that one faulty copy of DNA or one faulty copy of that particular gene is enough to cause the disease, which aligns with the heterozygous finding. Now look at the table given below. This data comes from a database called Omium, or Online Mendelian Inheritance in Man, which systematically curates which genes are related to which Mendelian genetic disorders. So Omium links this gene to multiple conditions, including Timothy syndrome, long QT syndrome, and so on. So these are disorders that are associated with neurological, cardiac, and developmental symptoms. And then thus here is the clincher here. The patient's clinical features, things like global developmental delay, encephalopathy, dysmorphism, all line very well with the expected phenotypes for these particular disorders that have been captured by Omium linked to this particular gene. So this clinical match combined with the zygosity, the inheritance pattern of the disorder, and the coverage for this variant position supports why this variant is also actionable for this particular patient. So in summary here, coverage tells us how trustworthy the data is. Zygosity will tell you how many copies of the genes that the patient has carry the variant. The inheritance model thus helps us map the zygosity to whether the patient has the disease, will get the disease or not. And the phenotype match helps us decide whether this variant truly explains the condition that the patient was tested for. So beyond single nucleotide variations, a very important concept here is that whole genome sequencing also gives us access to structural variations, and especially large deletions and duplications known as copy number variations or CNVs. So when a pathogenic gain or loss involving one or more genes are detected in a patient, they are especially important in conditions like neurodevelopmental disorders, different syndromes, and some forms of cancer also. So in reports, they will also be placed under primary or significant findings if they are clearly linked to the phenotype of the disorder of the patient that has come in for testing. The key reporting fields here are something that are a little different from what we saw for normal or for smaller variants that are detected by a WGS test. So these fields can include whether the structural variation detected is a deletion or a duplication in a gene. So whether that gene is being deleted or a copy of that gene is being deleted, or whether multiple copies of a particular gene is present in the patient's DNA. It can mention the genomic coordinates of that particular deletion or duplication along with the cytoband location, meaning that on which particular segment of your chromosome does that deletion or duplication lie. Then we can have information such as what are the affected genes that span that particular structural variation, along with the zygosity for the patient. So is that gene deleted in just one copy of their DNA or both copies of their DNA in case of a deletion? Or if in case it's a duplication, then how many copies of that particular gene is present in the patient will also be mentioned in the report. Along with this, just like the previous cases, the associated disorder and inheritance model will be given in the report. And then, of course, the clinical classification for the structural variation. So WGS can detect even small exonic CNVs that traditional tests might miss, making it a very critical advantage when it comes to clinical diagnostics. So just for an example, we have a case here now that shows how WGS can detect pathogenic structural variations that would probably be missed out on routine sequencing, such for example, using whole exome sequencing. So in this case, we have a 1-month-old male with microcephaly, brachycephaly, thin upper lip vermilion, and global developmental delay. The report has identified a heterozygous deletion on chromosome 22, spanning about 3.7 kilobases and is located at cytoband 22q11.2, meaning that this is the segment in chromosome 22 where the deletion has been detected in. Specifically, this region includes the TBX1 gene, which is very well known to be associated with a disorder called DiGeorge syndrome, which is an autosomal dominant disorder. So based on the phenotype, gene involvement, and known pathogenicity of this deletion, it has been classified as pathogenic. And if we look at this particular result in a very detailed way, we can see the exact genomic coordinates that span this particular deletion in this patient covers approximately 3.7 kilobases in the chromosome 22 region 22q11.2. The deletion overlaps TBX1 gene, which you can see in this figure here. And this has been confirmed via, and this region and this particular segment of chromosome in which the deletion lies has been confirmed here via the genome browser snapshot that is shown here. So clinically speaking, this deletion aligns very well with DiGeorge syndrome as listed in Omium, with an autosomal dominant inheritance reported for this particular disorder and a direct association with the gene TBX1. So, this combination of genomic mapping, phenotype correlation, and literature backed gene disease relationship allows this deletion to be confidently classified as pathogenic for the case of this patient. So, this kind of layered evidence from sequence to phenotype to databases is what gives WGS its diagnostic power. Now, coming to incidental and actionable genetic findings or secondary findings, we will shift our focus to something that is probably not being asked for in most cases in a general whole genome sequencing test. So, incidental findings are the results that are not related to the reason for being tested but are still important because they offer actionable medical insights. So, for example, finding a BRCA1 mutation in someone undergoing whole genome sequencing for a neurological condition can be an example of a secondary finding or an incidental finding. So, the ACMG or the American College of Medical Genetics and Genomics has curated a list of genes that are currently 84 genes in number in the version 3.3 list that cover approximately 52 unique disorders across 4 phenotype categories. This can include hereditary cancers, cardiomyopathies, arrhythmias, metabolic conditions, and other important health conditions that can be genetically determined. So, the principle here is that if a variant is detected in one of those genes and is classified as pathogenic or likely pathogenic, it must be reported. Because in these disorders, early intervention could save lives even if the individual is currently healthy. So, this is one of the biggest strengths that a whole genome sequencing test can offer, which is the ability to find unexpected things and still make it clinically meaningful. So, let's break down what qualifies as a secondary finding in a whole genome sequencing report. So, when we talk about actionable secondary findings, these are variants in a curated list of genes defined by ACMG. Version 3.3 is the current list that they have brought out just very recently, a week ago probably. And these genes have established clinical guidelines for intervention and follow-up. Crucially, these findings are unrelated to the original clinical reason for ordering sequencing, and they usually thus appear in a section titled Secondary Findings. And since these are just normal variations, they are formatted just like the variants that are given as primary findings. So, in case of incidental findings, the goal here is to give patients and families clinically meaningful, actionable health information, even if it is not immediately relevant to the current diagnosis. So, here is a real world example of a secondary finding that might be clinically important. So, we have a healthy 35-year-old woman who underwent whole genome sequencing for an unrelated reason. Probably she just wanted to check if everything is right with her, and she had an interest to get an whole genome sequencing test done for herself. And interestingly, the test identified a frameshift deletion in the BRCA1 gene, specifically a C.2766 del A variation, leading to a premature stop codon at the 923rd amino acid of BRCA1. It is a heterozygous change in this patient with a good coverage of 35x. And this variant has been classified as pathogenic, supported by ACMG attributes, indicating that it is rare in common populations. It causes loss of function for the BRCA1 gene, and it has also been previously reported as pathogenic by very reputable databases. So, BRCA1 is a gene that is associated with hereditary breast and ovarian cancer syndrome, something that probably most of us know. It is an autosomal dominant condition, and even though this finding wasn't related to the original clinical concern that the patient might have had, it is still medically significant because it will enable early screening, surveillance, and even preventive strategies for this particular woman. And this is why secondary findings should be reported, because sometimes the most important result that comes out of a whole genome sequencing test might be the one that you were not even looking for. And we will actually discuss more in detail about incidental findings and how to go about them, why they are important. and how these should be communicated to the patient in our session that has been dedicated to this topic. So coming to the next thing that can be included in a WGS report, these are other incidental germline findings. So these are findings that don't fall under in the genes that are listed under the ACMG secondary findings list, but they still might have some clinical relevance for the patient. So these are typically variants that are classified as likely pathogenic or pathogenic and might act as risk modifiers or be associated with future disease risk for that particular patient. So these appear in a separate section in the report that might be labeled as other germline findings, for example. The structure for reporting these variations is similar. The gene, transcript, consequence, zygosity, coverage, and ACMG classification can be provided in the report. And these findings may not demand immediate action, but they are worth monitoring or discussing in, for example, a genetic counseling session. So for example, here is a healthy 28-year-old male with a missense variant that was detected in a gene called NOTCH3, a gene that is known to be associated with a condition called CADASIL, which is an autosomal dominant disorder that affects neurological and cardiac functions in your body. The variant that has been detected, which is C.1690G2A, has a strong evidence of pathogenicity with attributes that have been assigned according to ACMG guidelines. And even though the patient is asymptomatic right now, this could inform future neurological surveillance or family screening for this particular patient, because for a condition like CADASIL, sudden cardiac death is something that is a high risk for such patients. So such findings walk a very fine line when it comes to reporting, because they are not immediately actionable, but they will definitely impact long-term care for these particular patients. Coming to the next thing that you may find in a whole genome sequencing report, which is clinically actionable pharmacogenomic variations. So pharmacogenomics is a powerful application of whole genome sequencing, and it helps predict how a person will respond to medications, thus guiding drug choice, dosage, and risk management for all patients. So whole genome sequencing test can thus not only detect or diagnose a particular patient, in fact, they can also give a treatment plan for these particular patients. The categories that might be included in a pharmacogenomic variation report can include adverse drug reactions, which can identify individuals that are at a high risk for some very commonly used drugs. It can inform dosage and efficacy adjustments for a particular patient undergoing the test, like needing a lower dose or predicting no response to particular drugs based on a patient's genotype. And it can also give the metabolizer type for a particular patient. For example, if they are able to metabolize a drug very rapidly compared to other people in the population, or whether they are not able to metabolize that drug at all compared to other people, which are then basically termed as ultra rapid metabolizers or poor metabolizers. For example, a DPYD gene poor metabolizer is flagged before giving the chemotherapy drug 5-fluorouracil to a particular patient, which will enable the clinician to proactively modify the treatment plan for this patient. Otherwise, the patient, since they are a poor metabolizer for this particular drug, might have had a very severe and very severe risk of developing toxic side effects to that particular drug. So this is basically real-time precision medicine in action based solely on pharmacogenomic variations. And how to report a pharmacogenomic variation is beyond the scope of this session, and we will discuss it more in detail in the session that has been particularly dedicated to PGx variants. Coming to the next thing, which is reporting carrier status for Mendelian genetic disorders through a whole genome sequencing report. So carrier screening now identifies individuals who carry one copy of a pathogenic variant in a recessive gene. So when both partners are actually carriers, there is a 25% chance that the child will be affected. And this isn't a rare event. Over 500 million people globally have single gene disorders, and over 10 lakh babies are born each year with a single gene disorder that is genetic in nature. So, whole genome sequencing allows us to screen for multiple conditions in one go, unlike panels that will only screen for particular conditions, and can thus allow, for example, screening of parents before planning a pregnancy. So, carrier status basically applies to autosomal recessive or X linked recessive or X linked dominant conditions, because a carrier is someone who harbors a pathogenic or a disease causing variation in their genome but does not show symptoms, because only one copy of their gene is mutated while they carry a healthy copy of that gene also. And for autosomal recessive conditions, you need to have both copies of your genes to be defective. So, in essence, if you have one copy of your gene that is affected and the disorder is autosomal recessive in nature, then you will not get the disorder. But if both parents are carriers for this particular one variation, or if it is an X-linked condition in, for example, a male, then there might be reproductive implications to this particular finding. This information is vital for very, very vital for informed reproductive decision making and family counseling for these patients, then, for example. So, the American College of Medical Genetics and Genomics actually provides a tiered guideline for reporting carrier screened variants, in which they include that tier 1 variant should be based on risk based screening, for example, conditions like cystic fibrosis and spinal muscular atrophy. Tier 2 conditions are conditions that have a carrier frequency greater than 1 in 100 in a particular population. And tier 3 something that they recommend, which includes testing for 97 autosomal recessive conditions and 16 X-linked conditions. And then tier 4 are a screening for ultra rare conditions, which is not recommended generally for routine use by ACMG guidelines. ACMG also states that screening is best offered pre-conception, especially in consanguineous populations where the risk is higher. The reason being that preconception screening is a better approach than prenatal screening, because it reduces the mental stress and tension that the patient might go through while getting tested for that particular disorder. So here is an example of a 30-year-old female. She carries a missense variation in the CFTR gene that is classified as pathogenic. And notice that the disease is autosomal recessive, while the variant is in a heterozygous state for this particular woman who came in for a whole genome sequencing test. Meaning that she does not have the disease herself, but she is now a carrier. While she is asymptomatic, this finding is significant in the context of family planning, especially if her partner is also a carrier of a pathogenic variation in the same gene, or in fact carries the same pathogenic variation in their gene. Now, this increases the chances that the child will inherit this particular variation from both parents. In fact, the chances of this happening are 25%. So, carrier screening findings like these are very commonly detected in all patients and all people who undergo a whole genome sequencing test, and are thus clinically important because these can help in the context of family planning. And lastly, a whole genome sequencing test, and more importantly, actually, can include mitochondrial DNA variations. So let's first take a moment to understand what is the mitochondrial DNA or mtDNA and how its variants may be interpreted in a whole genome sequencing test. So all of us from school remember only one thing probably, or unlike, if you are not like me, then you probably remember a lot more things about mitochondria. But we do remember that mitochondria is the powerhouse of the cell, meaning that all of the cell's function that is occurring is thanks to the energy that is being generated by the mitochondria. And thus, if something goes wrong in the energy generating mechanism of our body, it will affect a lot of different systems that are very high energy in demand. So, for example, neurological operations of the body, heart functioning, muscular functioning, things that require a lot of energy to function are things that will then get very severely affected if our mitochondria does not work properly. And thus, mitochondrial disorders are seen as multisystemic things in a particular patient. And unlike the DNA in our chromosomes or our nuclear DNA as it's called, mitochondrial DNA is a small circular genome found inside the mitochondria. So, mitochondria is not just an energy producing organelle in our body. Each cell that contains thousands of mitochondria actually contains multiple copies of its own mitochondrial genome also. So mitochondria is not just an energy producing machine in our body anymore. It is also something that has its own independent mitochondrial DNA. And mitochondrial DNA is inherited exclusively from the mother, apart from one very special documented case in literature, which is an exception, which makes its inheritance pattern quite distinct. So disorders caused by mitochondrial DNA variants, as I told before, affect organs with very high energy demands like brain, muscles, heart, and eyes. So now whole genome sequencing allows us to sequence the mitochondrial genome along with the nuclear genome in a single test, which is something that is missed by traditional whole exome sequencing tests and panel-based approaches. But what is unique here in terms of a mitochondrial DNA variation is now the concept of heteroplasmy and homoplasmy as opposed to heterozygous and homozygous conditions for a germline variation. So homoplasmy means that all copies of the mitochondrial DNA carry the same variation or the same mutation. So either all are normal or all are mutated. And heteroplasmy means that there is a mixture of different mitochondrial DNA types in your cells. Some copies carry the variation while others don't. And you can see it from this diagram here that, for example, a wild type percentage, if 100%, means that all the mitochondrial cells, all the copies of mitochondria in your cell do not carry any variation. And as this percentage decreases and the percentage of mutated copies of mitochondria in your cell increases, a disease phenotype threshold might be reached. And now this matters because symptoms often appear only when the proportion of mutated mitochondrial DNA or heteroplasmy level for that particular variation crosses a particular threshold, which is typically around 60 to 80%. Below that, the patient may remain asymptomatic or have very mild symptoms. So understanding this threshold is very, very important and a very key concept to interpreting mitochondrial disorders because it affects not only disease severity, but it also tells whether a variant will manifest clinically or not. So then, for example, while reporting mitochondrial DNA variants, we include the gene and the variant. We report whether it is homoplasmic and heteroplasmic, the heteroplasmy level of that particular variation as a percentage, and then of course the classification and the associated disorder. So mitochondrial DNA variation findings are particularly very rare, but very critical, especially in cases that involve metabolic, neuromuscular, or multisystemic symptoms. So here is a clinical example of a mitochondrial finding. We have a 3-month-old male patient presenting with very severe symptoms such as sensory neural hearing loss, optic atrophy, lactic acidosis, seizures, and dystonia. A whole genome sequencing test was ordered because the clinician suspected a mitochondrial disorder in this patient since the patient had a lot of different multisystemic symptoms. So whole genome sequencing revealed a variation in a gene called MTTV, which is a mitochondrial tRNA gene, specifically M.1644G2A change. The variant is homoplasmic, meaning all copies of the mitochondrial DNA carry the variation, with 100% heteroplasmy level detected at a coverage of 388x. This matches a condition or a disorder known as Leigh syndrome, which is a severe mitochondrial disorder. The variant is classified as likely pathogenic, and this diagnosis would have been very difficult to achieve without a whole genome sequencing test because empty DNA is not captured in a standard exome sequencing. And this finding enables not just diagnosis, but also appropriate genetic counseling from the family, keeping in mind very sensitively that mitochondrial disorders follow maternal inheritance. So, before we wrap up this very extensive session, I'll say it is important to understand that a whole genome sequencing report might also include something more than just variations, such as things like quality control metrics that will tell you as a clinician or as someone who's interpreting the report that how reliable the data is. So, if we break these concepts down, the mean coverage is the average number of sequencing reads that are covering each base in the genome. So if your report says a coverage of 30x means that each base was at an average sequence about 30 times by the sequencer. So this gives us a bit more confidence in the accuracy of the variant calls, especially when detecting heterozygous or low frequency variants. Next is coverage uniformity. This tells you that how evenly the sequencing was done across the genome. And a good report might say something like 95% of the genome is covered at a sequencing depth of 10x or more in the case of whole genome sequencing, which means that at least 95% of the genome is read at least 10 times on average, which is usually the minimum for the reliable detection of certain of most variations. And poor uniformity could mean that some important genes weren't covered properly, and that will limit interpretation. Then we have the percentage of mapped reads. This shows that how many reads that were generated by the sequencer successfully aligned to the human reference genome. Ideally, we want this number to be high, meaning that because low mapping rates could actually mean that there is a contamination in the sample, the DNA was degraded, for example, or even technical problems that may have occurred during sequencing. So even before we start interpreting variations, we can use these metrics to decide, is this data good enough to trust? And if it isn't, we might pause and troubleshoot this particular thing first. There are also several special applications that can be additionally there in a whole genome sequencing report, and these might be some interesting findings, and also including clinically relevant findings like repeat expansions that are implicated in certain neurodevelopmental disorders like Huntington's, HLA typing, which is useful in transplantation and immunogenetics, rare blood group genotypes, which is important for transfusion planning, ancestry inference, which can support variant classification, and runs of homozygosity, which may indicate consanguinity or recessive disease risks for a particular patient. So, in summary, while interpreting a whole genome sequencing approach report for a patient, always start with a phenotype driven finding because these are the usual diagnostic anchors. Look for secondary findings as they might offer preventive value even outside the range of presenting complaints for the particular patient. Use pharmacogenomics to tailor drug treatments for a particular patient coming in for a test. Report carrier status for reproductive planning. And more importantly, do not ignore things like mitochondrial or structural variations, especially in syndromic and multisystemic cases. And always check QC metrics before you start interpreting. In summary, I'll say whole genome sequencing is not just a comprehensive test, it is a future ready test. So, with this, I'll stop and I'll probably take questions now. So, please feel free to put your questions in the chat box.
Thanks a lot for your wonderful session, Dr. Bhatt. That was really insightful. Yes, a lot of people have asked that whether recordings would be available. And yes, of course, the recordings are available and the entire session is live on YouTube. So if in case you want to take a look into that for future reference. And should we drop into the questions, Dr. Bani?
Right, sure. So I'll go through the questions a bit in the chat box.
Mm-hmm.
So Arun has asked what are the ideal types of sample and what precautions while collecting and transport to the lab should be taken. So for germline testing, the ideal sample can be anything that can include your genomic DNA. So for example, a blood samples that we can then use to isolate WBCs and in essence the your genomic DNA from can make a good sample for a whole genome sequencing test. In terms of patients for hematological diseases, for example, we can also collect bone marrow for these particular patient types. CACNA variant, is it a depth of 32x or coverage as mentioned in this slide? So depth and coverage are kind of slightly interchangeable terms. For this particular variation that was shown in this example, the depth of sequencing for that particular position or the coverage at that particular position was mentioned as 32x. Manita has asked, kindly share the estimated cost for WGS per patient. So as Dr. Vinod also mentioned in his session that I will probably not discuss it here, but we will keep in touch with you and you can come back to us for a detailed cost breakup for this particular test. And also a reminder that we do offer the participants of this course a whole genome sequencing at a discounted price. What is the established cutoff for depth of a whole genome sequencing test and a whole exome sequencing test? So for whole genome sequencing, clinically accepted gold standard is a coverage of 30x, beyond which it has been shown by a lot of different labs across the world that a lot of different variation types can be detected very confidently at a minimum depth of 30x. For whole exome sequencing, the coverage depth for this particular test has to be a lot higher compared to a whole genome sequencing test, because whole exome sequencing by its nature it's a capture based test, and when we do a capture based test, the accuracy has to be increased by increasing the coverage. So for whole exome sequencing, for a germline test, a depth of 100x is what has been deemed as clinically acceptable by multiple labs across the world. What is the minimum depth that can be reported? If a low depth is observed but associated with a phenotype, can it still be considered valid? So since whole genome sequencing, a clinical grade 1 can have an average depth of as low as 30x. So within this range, if we find a variation in a report, it can be clinically associated and very confidently reported. If, for example, you are seeing a variation that has a very low depth of, for example, 5x, it might make sense to either resequence the sample to confirm whether that variation is present or not, or as confirmation, you can go in for other testing options also. In case there is a novel inheritance pattern for a disease, such as an autosomal dominant inheritance for an autosomal recessive disorder in literature, which is still not included in Omnium, should the whole genome sequencing report include the novel inheritance pattern, though it is still not in Omnium? I will say yes, it might be included in the whole genome sequencing report, along with the disclaimer that probably mentions the new evidence that has come in that the particular disorder might manifest in patients in, for example, in context of incomplete symptoms in an autosomal pattern, a dominant pattern also. So for example, SLC4A1 is a gene that follows both inheritance patterns, autosomal dominant and autosomal recessive. In the dominant cases, the phenotype may not be as strong as in the cases that are found to be homozygous for that particular variation, but it still makes sense to report these variations because they also might be clinically correlating to the patient. So it always makes sense to first of all go back and check whether the patient does have the clinical phenotypes for that particular disease, and then report back with a disclaimer with the updated evidence for that particular disorder. Can you please enlighten us about highly significant variant of uncertain significance? Yes, so these are variations that are right now as classified by ACMG AMP guidelines as variants of uncertain significance, but these are lying in genes that very highly correlate with the disorder that your patient is presenting with. I will not touch upon this in a lot of detail because in the next session, Ms. Ramya will be also discussing ACMG AMP guidelines in a very vast detail and how to deal with the VUS for these clinical cases. Was a whole exome sequencing sufficient in this case as it was clinically quite representative of DiGeorge syndrome? So testing it through, the symptoms that are presented in the patient might not exactly always overlap with a particular disorder. So, for example, like I explained, in mitochondrial disorders and in conditions like DiGeorge syndrome, the clinical presentations can span multiple systems. So these conditions generally confuse doctors a lot as to where the treatment should start or where the clinical management should start, since there is not a pinpoint evidence towards one particular disorder. So in this case, whole genome sequencing should have been done because it will help you directly correlate now that this is the genotype that is leading to this particular phenotype. And in cases like cardiac channelopathies, for example, where the treatment is informed by the gene in which the variation is lying, this then becomes very important to do so. How to approach two heterozygous variants in an autosomal recessive disorder. How to confirm compound heterozygosity if sequencing is not available for patients. So inherently, NGS will not give you information about the strand in which that particular variation lies. But if we find two pathogenic or likely pathogenic variations in a patient that are associated with a autosomal recessive condition and the variations are found as heterozygous and the patient does display the phenotype for that particular disorder, these can be then reported in a whole genome sequencing report with a disclaimer. MLPA can pick up DiGeorge syndrome very well at a much cost effective way. Yes, I agree, but if the disease was not DiGeorge syndrome, then MLPA would have not detected it. So why whole genome sequencing helped here is that if in case the disease was something else, this would have been picked up by the single test that we had ordered for the patient. And the patient would not have to run around, the patient's family would not have had to run around to do different tests for just getting the diagnosis for their child. And this is often the case for rare genetic disorders where the patient family spent at least 5 to 7 years into getting a diagnosis for their condition. So if there is one test available that can help reduce the diagnostic odyssey, then we should probably always opt for that instead of using a piecemeal approach in which we screen for different conditions using different types of tests. In what setting Sanger validation is required for a finding in whole genome sequencing? So traditionally, Sanger sequencing was thought to be as the most accurate type of sequencing approach because it gave an accuracy of 99%. And now that NGS has advanced even further, we now know that there are different quality metrics that can be used to describe an NGS or in extension a whole genome sequencing test. This includes a quality metric called a Q score or a FRED score, which tells you how reliable your NGS or a whole genome sequencing test data is. And a Q score of 30, for example, means that the test has been 99.9% accurate in detecting or in reading the sample that you had provided for sequencing. So if we now compare it to Sanger sequencing, which was also 99.9% accurate, a Q30 whole genome sequencing test is also 99.9% accurate. And Q30 is the clinically accepted gold standard for reporting a whole genome sequencing test, meaning that every variation detected is at least detected at a quality score of 30 or beyond. So a Q40 score will mean that the data is 99.99% accurate, Q50 means 99.999% accurate, and so on. So we have already reached the threshold for accuracy of testing by a Q30 score itself, and which is why whole genome sequencing is now directly comparable to the accuracy of a whole genome of a traditional Sanger sequencing, along with the additional benefit being that turnaround time is lower and the effort requires is much lower compared to Sanger. Is it right to report a likely pathogenic carrier variant in any non-carrier testing requests? For reporting carrier variants, always follow the guidelines in terms of in clinical interpretation of that variation, which comes to ACMG and AMP guidelines, and also make sure that the inheritance and the of the associated disorder is something that matches the zygosity of the disease. So I think I'll stop here because there are a lot of different questions that have come in, which is very encouraging to note. And we will be happy to follow up with these questions and we'll give you the responses back soon.
That's great. Okay. Let's look into further questions and more can be shared to us through lab@karkinos.in, which is our official email ID for all diagnostics and Knowmics related activities. Please share your questions there. You could also reply back to your event invitation emails that has been sent to you. Please feel free to follow us on our Knowmics YouTube channel for such live events and updates going ahead. Thanks a lot for being in the audience. We are just going to take a quick short break of around 20 minutes and we will be back with our next session. So grab some water, grab some food, and come back to us. We'll continue hosting. The next session would be with Ms. Ramya Venkatesha after the break, where she will be explaining about guidelines for evaluation of pathogenicity. Thank you all.
Welcome to Karkinos Healthcare's network of Advanced Center for Cancer Diagnostics and Research Labs. As a technology-led, purpose-driven oncology platform, we operate 6 state-of-the-art labs across more than 120,000 square feet. Our histopathology division offers a complete range of services. From routine tissue analysis to advanced immunohistochemistry for biomarker detection, fluorescence in situ hybridization for genetic abnormalities, and special stains for enhanced tissue insights. Every sample receives the most detailed and clinically relevant evaluation. Efficiency is at the core of our operations. Through advanced automation and technology, we ensure industry-best turnaround times, cost efficiency, and seamless logistics, delivering temperature-controlled sample collection and rapid reporting right to your doorstep. As part of our molecular diagnostics, we offer over 70 unique molecular tests, which are tailor-made to provide clinicians with critical therapeutic, prognostic, diagnostic, and actionable insights. our test menu is continually evolving to reflect the latest research and clinical guidelines, ensuring access to state-of-the-art diagnostics. We employ cutting-edge technologies, including the thermo sequencing platform, featuring the Ion Chef for automated sample prep and the Ion Gene Studio S5 sequencer for diverse oncodiagnostic applications. The Genexus system offers a fully automated NGS workflow, from sample to report. At the heart of our innovation is sequencing at scale. 2 NovaSeq 6000 systems with 3 Illumina NovaSeq X Plus. We also house India's first PacBio Revio ultra-long read NGS sequencing platform. With India's largest private sequencing capacity, we offer rapid, targeted, custom, and comprehensive genomic sequencing for clinical and research needs at affordable costs, while also delivering results twice as fast as the industry standard. Our team of leading Indian scientists, clinicians, and technologists, combined with our commitment to scientific rigor, truly distinguishes Karkinos. Join us in making personalized medicine a reality for every Indian. At Karkinos, we believe every life deserves the best chance. And we are here to make that vision a reality for all, today and for generations to come. Welcome to Karkinos Healthcare's network of advanced Center for Cancer Diagnostics and Research Labs. As a technology-led, purpose-driven oncology platform, we operate 6 state-of-the-art labs across more than 120,000 square feet. Our histopathology division offers a complete range of services. from routine tissue analysis to advanced immunohistochemistry for biomarker detection, fluorescence in situ hybridization for genetic abnormalities, and special stains for enhanced tissue insights. Every sample receives the most detailed and clinically relevant evaluation. Efficiency is at the core of our operations. Through advanced automation and technology, we ensure industry-best turnaround times, cost efficiency, and seamless logistics, delivering temperature-controlled sample collection and rapid reporting right to your doorstep. As part of our molecular diagnostics, we offer over 70 unique molecular tests which are tailor-made to provide clinicians with critical therapeutic, prognostic, diagnostic, and actionable insights. Our test menu is continually evolving to reflect the latest research and clinical guidelines, ensuring access to state-of-the-art diagnostics. We employ cutting-edge technologies, including the thermo sequencing platform, featuring the Ion Chef for automated sample prep and the Ion Gene Studio S5 sequencer for diverse oncodiagnostic applications. The Genexus system offers a fully automated NGS workflow, from sample to report. At the heart of our innovation is sequencing at scale. 2 NovaSeq 6000 systems with 3 Illumina NovaSeq X Plus. We also house India's first PacBio Revio ultra-long read NGS sequencing platform. With India's largest private sequencing capacity, we offer rapid, targeted, custom, and comprehensive genomic sequencing for clinical and research needs at affordable costs, while also delivering results twice as fast as the industry standard. Our team of leading Indian scientists, clinicians, and technologists, combined with our commitment to scientific rigor, truly distinguishes Karkinos. Join us in making personalized medicine a reality for every Indian. At Karkinos, we believe every life deserves the best chance. And we are here to make that vision a reality for all, today and for generations to come. Welcome to Karkinos Healthcare's network of Advanced Center for Cancer Diagnostics and Research Labs. As a technology-led, purpose-driven oncology platform, we operate 6 state-of-the-art labs across more than 120,000 square feet. our histopathology division offers a complete range of services. From routine tissue analysis to advanced immunohistochemistry for biomarker detection, fluorescence in situ hybridization for genetic abnormalities, and special stains for enhanced tissue insights. Every sample receives the most detailed and clinically relevant evaluation. Efficiency is at the core of our operations. Through advanced automation and technology, we ensure industry-best turnaround times, cost efficiency, and seamless logistics, delivering temperature-controlled sample collection and rapid reporting right to your doorstep. As part of our molecular diagnostics, we offer over 70 unique molecular tests which are tailor-made to provide clinicians with critical therapeutic, prognostic, diagnostic, and actionable insights. Our test menu is continually evolving to reflect the latest research and clinical guidelines, ensuring access to state-of-the-art diagnostics. We employ cutting-edge technologies, including the thermo sequencing platform, featuring the Ion Chef for automated sample prep and the Ion Gene Studio S5 sequencer for diverse oncodiagnostic applications. The Genexus system offers a fully automated NGS workflow, from sample to report. At the heart of our innovation is sequencing at scale. 2 NovaSeq 6000 systems with 3 Illumina NovaSeq X Plus. We also house India's first PacBio Revio ultra-long read NGS sequencing platform. With India's largest private sequencing capacity, we offer rapid, targeted, custom, and comprehensive genomic sequencing for clinical and research needs at affordable costs, while also delivering results twice as fast as the industry standard. Our team of leading Indian scientists, clinicians, and technologists, combined with our commitment to scientific rigor, truly distinguishes Karkinos. Join us in making personalized medicine a reality for every Indian. At Karkinos, we believe every life deserves the best chance. And we are here to make that vision a reality for all, today and for generations to come. Welcome to Karkinos Healthcare's network of advanced Center for Cancer Diagnostics and Research Labs. As a technology-led, purpose-driven oncology platform, we operate 6 state-of-the-art labs across more than 120,000 square feet. Our histopathology division offers a complete range of services. From routine tissue analysis to advanced immunohistochemistry for biomarker detection, fluorescence in situ hybridization for genetic abnormalities, and special stains for enhanced tissue insights. Every sample receives the most detailed and clinically relevant evaluation. Efficiency is at the core of our operations. Through advanced automation and technology, we ensure industry-best turnaround times, cost efficiency, and seamless logistics, delivering temperature-controlled sample collection and rapid reporting right to your doorstep. As part of our molecular diagnostics, we offer over 70 unique molecular tests which are tailor-made to provide clinicians with critical therapeutic, prognostic, diagnostic, and actionable insights. Our test menu is continually evolving to reflect the latest research and clinical guidelines, ensuring access to state-of-the-art diagnostics. We employ cutting-edge technologies, including the thermo sequencing platform, featuring the Ion Chef for automated sample prep and the Ion Gene Studio S5 sequencer for diverse oncodiagnostic applications. The Genexus system offers a fully automated NGS workflow, from sample to report. At the heart of our innovation is sequencing at scale. 2 NovaSeq 6000 systems with 3 Illumina NovaSeq X Plus. We also house India's first PacBio Revio ultra-long read NGS sequencing platform. With India's largest private sequencing capacity, we offer rapid, targeted, custom, and comprehensive genomic sequencing for clinical and research needs at affordable costs, while also delivering results twice as fast as the industry standard. Our team of leading Indian scientists, clinicians, and technologists, combined with our commitment to scientific rigor, truly distinguishes Karkinos. Join us in making personalized medicine a reality for every Indian. At Karkinos, we believe every life deserves the best chance. And we are here to make that vision a reality for all, today and for generations to come. Welcome to Karkinos Healthcare's network of advanced Center for Cancer Diagnostics and Research Labs. As a technology-led, purpose-driven oncology platform, we operate 6 state-of-the-art labs across more than 120,000 square feet. Our histopathology division offers a complete range of services. From routine tissue analysis to advanced immunohistochemistry for biomarker detection, fluorescence in situ hybridization for genetic abnormalities, and special stains for enhanced tissue insights. Every sample receives the most detailed and clinically relevant evaluation. Efficiency is at the core of our operations. Through advanced automation and technology, we ensure industry-best turnaround times, cost efficiency, and seamless logistics, delivering temperature-controlled sample collection and rapid reporting right to your doorstep. As part of our molecular diagnostics, we offer over 70 unique molecular tests which are tailor-made to provide clinicians with critical therapeutic, prognostic, diagnostic, and actionable insights. Our test menu is continually evolving to reflect the latest research and clinical guidelines, ensuring access to state-of-the-art diagnostics. We employ cutting-edge technologies, including the thermo sequencing platform, featuring the Ion Chef for automated sample prep and the Ion Gene Studio S5 sequencer for diverse oncodiagnostic applications. The Genexus system offers a fully automated NGS workflow, from sample to report. At the heart of our innovation is sequencing at scale. 2 NovaSeq 6000 systems with 3 Illumina NovaSeq X Plus. We also house India's first PacBio Revio ultra-long read NGS sequencing platform. With India's largest private sequencing capacity, we offer rapid, targeted, custom, and comprehensive genomic sequencing for clinical and research needs at affordable costs, while also delivering results twice as fast as the industry standard. Our team of leading Indian scientists, clinicians, and technologists, combined with our commitment to scientific rigor, truly distinguishes Karkinos. Join us in making personalized medicine a reality for every Indian.
Hello all. Hi. Welcome back. Welcome back to the session. We will be starting back shortly. Next session will be by Ms. Ramya Venkatesha. The session is on guidelines for evaluation of pathogenicity. Let's get started here without further ado. In the meanwhile, I'm looking more participants joining in here into the session. Let's wait for a couple more minutes for more participants to join back into the session, and we will start ahead. Okay. Okay. Miss Ramya, I'm unmuting you and I'm setting up your screen. So let's get started.
Yes, thank you, Harish. Okay, so I hope my screen is visible. Let me begin. So now that we have understood how a WGS report works and the variants in it, let us understand how we can evaluate these evidences. So we have to remember, sometimes mutations can be due to a chain reaction. For example, cake becomes coke, coke becomes joke, joke can become poke, poke becomes pike, pike becomes pie, pie becomes pick, pick becomes pick again, but insertion of K in this case, and pick can become sick. So do you see how that chain reaction went? From cake, it went all the way to sick. Now sick can have two different interpretations. A, it can lead to a disorder, and B, if we have Gen Zs like me, sick is often used to imply coolness. So this is where the importance of context comes in. And from this came from a community of scientific members who developed something called as the ACMG guidelines. Now let's go back a little to understand the history of ACMG. Imagine a time when doctors struggled to understand why some families had higher risk of genetic diseases. It was the early 1990s and the genetic test was still in its infancy. Terms like mutation and polymorphism was often confused. A mutation was defined as permanent change in the nucleotide sequence, whereas polymorphism defined as a variant with a frequency above 1%. So, scientists and clinicians were making groundbreaking discoveries, but there was no standardized way to interpret these genetic variants. Without guidelines, different labs would often classify the same genetic change in completely different ways, leaving doctors and patients alike confused. So, recognizing this problem, a group of visionary geneticists and clinicians came together in 2013 to form the American College of Medical Genetics and Genomics, that is ACMG. So, they suggested a 5-tier classification like pathogenic, likely pathogenic, VOS, or what we call variants of uncertain significance, likely benign, and benign. So, here, uh, key to note is pathogenic and likely pathogenic are what we can also call disease causing, and benign and likely benign are what we can call non disease causing or harmless. So, they were made of 28 standard attributes. Now, let's go into these attributes in detail. So now let's start with the two big categories on the slide. You can see that I have shown pathogenic attributes in a pink color and a benign attributes in green color. So these represent the direction we are going while interpreting a variant. Are we leaning towards it being disease causing, that is pathogenic, or harmless, that is benign? So now evidence generally comes in levels. So now ACMG doesn't treat all evidence equally. They've grouped them on basis of how strong the evidence is, from very strong to supporting. So now let's go again to the pink part, that is pathogenic side. On the top right, you'll see that we'll have very strong evidence like PBS1. This is for things that are known for non-loss of function mutations in critical genes. Next, we have strong, that lies from PS1 to PS4. Then we have moderate, that is PM1 to PM6, and supporting, that is PP1 to PP5. So these are the pathogenic attributes of ACMG guidelines. So generally, if a variant has several pieces of moderate or strong evidences or supporting evidences in combination, it may be classified as a likely pathogenic or pathogenic. Think of these like different weights. You combine them like building blocks to arrive at a final classification. Coming to the benign side. On the other hand, so if a variant has strong evidence that it's harmless, like it appears in, say, like 5% of the general population, we use the benign criteria. BA1 is the strongest. It stands alone. Then we have a BS1 to BS4, which is strong. So BA1 is the very strong criteria here. BS1 to BS4 is the strong criteria. BP1 and BP7 to BP7 are supporting criteria. So the more confidence that we have that the variant isn't causing the disease, it is likely that the variant is benign. So in total, there are 28, and these 28 attributes are spread across 8 domains. Like we have population data, functional data, computational predictions, and segregation data. So we mix and match these attributes depending on what kind of evidence we have for a particular variant. So there are 8 evidence domains used in the ACMG guidelines. So basically, these are the different types of data we rely on while deciding whether a genetic variant is pathogenic or benign. Each colored box represents a domain of evidence, and the small labels which you can see below those evidences, like in red and green, like PP3, there is BS2, and there's a PP1, there's a BS4, all these are the specific ACMG attributes linked to this type of evidence. So we have computational data, population data, variant type data, databases, functional data, segregation data, case versus control data, and allelic data. So let's start with the first criteria, that is computational data. So coming to computational data, we can obtain this data from 3 softwares, each of whom give us a score whether the variant is deleterious in nature or not. So we can start with SIFT. They have a score range of 0 to 1, 0 being detrimental and 1 being tolerated. So the cutoff range to be considered for deleterious is about 0 to 0.5. Polyphen is slightly different. Here 0 here means benign or no effect on the protein. 1 is deleterious, and the cutoff for deleterious is 0.5 to 1. CAD is another software which we can consider, where it does not have a fixed score range but can be represented from, say, a 0 to 40, and a CAD score above 15 is generally considered deleterious. Now, generally, a consensus of 2 out of 3 is taken. So if two tools claim that the variant is deleterious in nature, we consider it to be deleterious. So the sources to gather these evidences are SIFT, CAD, and Polyphen. So coming to population data, we generally look at what we call the MAF of the population. That is the number of times a minor allele appears divided by the total number of alleles in the population. So if a variant's prevalence is less than a certain number in the total population, the variant is considered to be rare and might be pathogenic in nature. So the sources to collect this population data are generally sources like Nomad, which is a very famous population database, or Varzome can also be used. Now, based on the variant type, different types of data are collected to mainly identify the impact or protein function. As discussed earlier, the different variant types can be, say, missense, nonsense, synonymous, frame shift, in-frame, splicing variants, intronic. So these different variant types have different attributes assigned to them. Sources to find these types of information is either you can study the HGVS nomenclature, And also we also have a tool called VEP. ClinVar is also a great tool to access this sort of data. Coming to the databases, so the following evidences are collected. So now if you want to check for the presence of a functional domain in the variant, we have something called as the UCSC genome browser. That would be a great way to search for the variant. To check for presence of repeat regions in the variant, we can use again UCSC is a visualization tool where you can see the different domains. You can use several tracks like PFAM track. There is a Uniprot track to check for protein domains. To check for repeat regions, repeat masker is of course a very great tool to use. And if you want to verify if a reputable source is classifying the variant as a pathogenic or likely pathogenic variant, ClinVar would be your best go-to tool. So functional data. These are mostly literature evidences where keywords such as say gene name with P dot of variant can be used, functional effect of protein, damaging effect of gene, whether it's deleterious in function, all of these. So if there is any paper with using certain relevant keywords, we can check if there have been any in vitro studies done to make sure if the variant is deleterious in nature or not. So again, web search using keywords is one great idea. ClinVar has literature section under it, under the references. So that would be one more key area to search for data under if the variant is already reported in ClinVar. Then we also have PubMed, which is a very good literature tool that can be used. Moving to segregation data. Now, what is segregation data? So it is used to determine whether a genetic variant is used to determine whether a genetic variant is associated with the disease by tracking how it's inherited within the families. So now let's look at the first scenario. So this is something called as a pedigree chart. So the black filled symbols represent affected individuals. In that also we have squares that represent males and circles to represent females. So if you see here the genetic notation, T27R and wild type. So you have these are the genetic notations that show the variant carried by each person. So WT here stands for wild type, that means no mutation has occurred, and T27R is a mutation that causes a change in threonine to arginine. Here the variant, that is T27R, is present only in the affected individuals and not in the unaffected individuals. So this supports the idea that the variant is likely pathogenic because it's consistently linked with the disease. So the variant segregates with the disorder and it's not there in the unaffected individuals. But if we go to the right pedigree chart, the variant does not segregate with the disorder. So if you again see here, affected and unaffected individuals both carry the variant. So carrying is represented by a half circle in this case, or a half square. So this is how we represent a carrier. So some affected individuals do not have the variant, and some unaffected individuals do have it. So this can again suggest that the variant is benign or incidental in nature. So sources to collect these evidences are mostly by looking up some case studies with multiple familial generations for this specific variant. Please keep a note that to apply, say, this particular information, we need to have the same specific variant to be mentioned in the family studies. Or you can also look at if there is a family history of the patient whose genetic testing is done, like triad studies. That is also one way to search for this evidence. Now coming to case versus control studies. Now researchers generally compare two groups. The case group, that is people with the disease, and the control group, that is healthy individuals. They check how often variants appear in each group and calculate an odds ratio, which helps determine having the mutation increases disease risk or not. So if the odds ratio is significantly high, so this criteria I'm talking about is PS4, that is case studies. It's a very strong pathogenic criteria. So if the odds ratio, which is significantly high, that is above 0.5, above 5, for example, the variant is considered a strong risk factor for the disease. So now let's take a very simple example to understand this. Suppose this is a study of breast cancer with the BRCA mutation. In this case, 40 people have the mutation and the disease. 60 people have the mutation but don't have the disease. 20 people do not have the mutation and have the disease. And 80 people don't have the mutation or the disease. So it's a permutation and combination of everybody who has the variant or doesn't have the disease. So odds of disease in people with mutation is, in this case, 40 by 60. So we have about 0.67. Odds of disease in people without mutation is 20 by 80. So it would round up to 0.25. So when you have to calculate odds ratio, it's odds in case group versus odds in control group. So 0.67 divided by 0.25. So we have 2.67%. In this case, so one of the sources to collect this data is either GWAS studies, that is genome-wide association studies. So they generally have a lot of case control studies with odds ratio and confidence interval mentioned. So for PS4 criteria, you can look for GWAS studies or also do a simple literature search. There will be a lot of case studies available if the variant is reported to be pathogenic. So these were all evidences for pathogenic and benign variants. So here comes the question. What about variants of uncertain significance or VUS? Are they really as uncertain as we call them? Any answers? Okay. So yes, the VUS or variants of uncertain significance is called so due to lack of evidence. The best way to explain this is when a variant does not fall into pathogenic or benign category. So I have represented it in form of a weighing scale. So you can see that on one scale, so VUS is when the scale is balanced with 4 evidences on each side. Suppose you have 4 evidences towards pathogenic and 4 evidences towards benign. So this can be one of the scenarios. Of course, there are variants where you have almost very less evidences to even categorize them as benign or this. But for this is an ideal case scenario. So as soon as another evidence is added to the pathogenic criteria, the VOS gets reclassified as pathogenic. Thus, it is a very volatile group of variants in terms of classification, and it cannot be considered junk data. It can also be the other way around. If the evidence was added to the benign part of the scale, then the variant would be classified under benign. These types of variants baffle scientists even to this day. So here are a few pointers on how to deal with them. We can try to gather more evidence, look back at all the other ACMG attributes to see if something has been missed, if any family studies have been done, and we can try and evaluate those evidences. We also have some patient-specific attributes to consider, like say a PP4. So some of these are generally missed by looking at ACMG guidelines. So this would be one more way. Sometimes enough research may not have been done on the variant. In this case, we have to reassess them over time as new evidences are published every day. If it seems to match the patient's phenotype but is in VUS, then genetic counseling is advised. In this case, it is not a diagnosis. It is not proof of being a carrier. It does not confirm or rule out the condition. Remember, reassurance is the key. And lastly, collaboration is key. So discuss with other clinicians, fellow bioinformaticians, geneticists. Getting a different perspective from everyone may be a key breakthrough. Now that we have discussed about why ACMG is considered the gold standard for clinical evaluation of the variants, let us understand its importance through a very simple case study. So this is a 5-year-old patient. The patient has exhibited symptoms of global developmental delay, that is GDD, neuropathy affecting motor skills, and she also got an ECG which showed signs of small PFO, that is patent foramen ovale with left to right shunt, and a mild tricuspid regurgitation. So these were all the symptoms, and we also found a variant in CACNA1C, which was not reported in ClinVar. Right? So here comes our question. What would be the right approach? A, do we label it as a VUS? B, do we not report it at all since it is not found in a reputable database? Or C, do not rely on one evidence alone and search for other evidences? Can I get any answers in the chat for this? Okay, so I can see a few answers popping up already, and I would say the correct answer is do not rely on one evidence alone and search for other evidences. Not all databases are updated as regularly. So let us start by putting together evidences for a little patient now. So it would almost feel like a jigsaw puzzle where we collect all the pieces and we get the right diagnosis. So we have a blank screen right now. I will present evidence 1 to you. So this is evidence 1. We have a PM2 attribute. Since the variant is not found in NOMAD, a population database, it implies that the variant is very rare. We have evidence 2, where we have PM1, and PM1 is used when the variant is located in a known functional domain of the protein. So that's a big red flag because any change in that particular domain can affect how the protein works. Evidence 3 comes in form of tools like SIFT, Polyphen, or CAT. So it is our computational evidence in this case that predict whether a variant is likely damaging. So while these tools aren't the most strong evidence out there, if multiple of them agree, as I mentioned in my earlier slides, if we take a 2 out of 3 consensus, so if multiple of them agree that the variant is deleterious, we can count that as a supporting evidence. And lastly, we have our fourth evidence in form of a functional study. So this is a very strong evidence because we found a study where in vitro studies conducted on the variant said that the variant has a damaging effect. So hence PS3 was assigned. So we have 4 pieces now of evidence now: a PM2, a PM1, a PP3, and a PS3. Now, at first glance, these puzzle pieces will look like a very random pattern. But here is where the magic happens. That's right, it's a smiley. So hence, the variant was reclassified from VOS to pathogenic with a combination of one strong attribute, 2 moderate attributes, and 1 supporting attribute, leading to the correct diagnosis. So we have a PM1, a PM2, a PP3, and a PS3. So a pathogenic variant, CACNA1C variant, was found matching the exact phenotypes of the patient. Thereby, the patient reached the correct diagnosis through ACMG guidelines. So with this, I conclude my session. I hope I was able to convey how these guidelines can completely change our perspective of looking at a variant, thereby improving the accuracy for our patient diagnosis. Hence why ACMG guidelines is very, very crucial for patient reporting. So if there are any questions, I would love to take it up. Thank you.
Hey, thanks a lot for the wonderful session, Ramya. This was really great. Please feel free to look into the questions on the comment and please feel free to ask them to us and answer them. Yeah.
Okay, so we have a question asking how can we evaluate or predict whether a UTR variant has any evidence of pathogenicity? So UTR variants can be evaluated for pathogenicity by assessing the impact on the gene regulation rather than protein coding. So key factors can include evolutionary conservation, disruption of regulatory elements. Computational tools also are a great way to look for the functional impact. And additionally, if it's rare in the population database or any other existing literature or clinical database entry, or if ClinVar supports pathogenicity, so all of these can be considered. Any tool recommendations for this kind of analysis? So for if you want majority of the information, VEP is a great tool. It gives you your PM2, that is population data. It gives you computational data. It can also give you, say, if your variant is a splice variant and you want to look at splice AI scores to see if it has any consequence, that would also be a good way. Okay, so there is one more question, very interesting question, by the way. If a variant is pathogenic or likely pathogenic in ClinVar, but benign or VUS, according to ACMG, what should we follow? So I would suggest that, again, ClinVar has a lot of references, and also if you notice under the people who would have submitted the entries, sometimes it is just automatically generated. So I would not completely rely on ClinVar in this case. I would mostly try and look for all the other evidences if possible, club them together, and then reach a conclusion. Okay, so when we say functional studies, how in-depth should we go? Can you please elaborate? Thank you, Priyanka Setia, for that question. So when we say functional studies, because it is a very, again, strong parameter, it should be very stringently assigned. First thing would be that we should look at, say, in vitro studies, and we should also make sure that the variant they are evaluating is exactly the same variant. That includes the C dot, or that is the coding transcript, as well as the P dot should, or either one of them, whatever is mentioned, should be the exact same as the variant which you are looking for. So this, and it should also say that it functionally damages, it has a functional, it's deleterious in nature. Some of these keywords should be mentioned in the paper. How many evidence domains will we consider for VUS for pathogenic? So actually, Sindhu Sahu, again, I would say that ACMG actually has a set of full set of evidences and combinations which you can try for pathogenic and benign. But I would say at least you need to have two moderate evidences, one supporting evidences, or one strong evidence, or two moderate evidences and two supporting evidences. So there are a combination of things you can try, but these are some of the ways you can reclassify your VUSs. Coming, any other questions? If a variant, Najma Khan has another question. If a variant is likely pathogenic or pathogenic, but after applying MAF, it is filtered out, the one we should prioritize or leave this? So actually, ma'am, if BA1 is applied, it is already a very strong benign criteria. It wouldn't mean that you leave out the variant, because sometimes you might have another strong pathogenic evidence also you might find. Say like PS4, maybe you might find some case studies done on the variant. In this particular case, I would say to weigh out all the evidences again, and maybe end up getting a VUS. It completely depends on the types of evidences you find. So why, there's a question by Elizabeth Antony, why benign and likely benign variants are not reported? Ma'am, so likely benign and benign variants, as the definition states, it is harmless. So we try to report only things that are very relevant to the patient. VOSs, I can tell you why VOSs are reported so it would make a great comparison. VOSs are reported because it does not mean that it may affect the patient or it may not affect the patient. So we are keeping it just in case in future some extra evidence is added and it might actually be beneficial for the patient. However, if it is already said likely benign or benign, it would make no difference to the patient. So this is all in terms of patient relevance. So if we get a novel mutation like missense, can we publish a paper based on this, or we have to go for another wet lab experiment? Ma'am, novel mutations, I think we have to try and evaluate whatever necessary, but yes, I think wet lab experiments are required because it is really considered a very strong set of evidence. If there are any more questions. Okay, maybe I can take one or two more questions. So any guidelines for reporting structural variants? An excellent question. Again, I would say as of SV variant reporting guidelines, say currently there are none for AMP, ACMG for small variants, but again, guidelines may evolve, and the more we sequence, the more important it becomes for consortiums to come up with this. So the more WGS we do, the better answers we might get. I think I would end my session with this in the interest of time. Thank you very much.
Sure. Thanks a lot. Thanks a lot, Mr. Ramya. The session was really wonderful. We were able to explore a lot of further things in terms of pathogenicity evaluation through your session here. And the questions are really, interesting. Like the case studies that we usually discuss in internal forums and Karkinos, similar questions are popping up in external forums. It's great that we could share this knowledge throughout to everyone. Thanks a lot for sharing your knowledge, Ms. Ramya. Thank you. As for the next session, we will be going with Pharmacogenomics, Guidelines, Interpretation, and Action by Dr. Barney Jolly. Pharmacogenomics was one of the keystone sessions in Knowmics with a very large crowd when we initially started. It's a very important topic and very most clinically relevant part from a lot of angles. Let's get Dr. Barney Jolly to guide us further in terms of pharmacogenomics here. Ms. Barney, the stage is yours.
Thank you so much, Harish. And actually, before I begin my session here, I would also like to introduce our host today, who is Mr. Harish Ramanathan. Mr. Harish is the Manager of Technology at Karkinos Healthcare in the Navi Mumbai lab, where he bridges healthcare and technology to support innovative cancer care solutions. with a very strong foundation in biomedical engineering, initially from Anna University, and an MBA in health IT management from IIHMR University, Harish actually brings a unique blend of technical and strategic expertise to our company here. And his diverse professional journey includes leadership roles in the US healthcare market research and digital health initiatives, including stints at Spry Movement App, Truly Human Consulting, and the Data Congress, where he served as played a very key role. And he has also gained experience in operations research at Narayana Health, furthering grounding his understanding of the Indian healthcare system. He is very well known for his entrepreneurial ideas and systems thinking, and is very passionate about leveraging health informatics and digital tools, especially genomics and whole genome solutions, to drive impact in a very patient-centric way. So thank you, Harish, for being our host today, and I'll now continue with my session.
Thanks a lot. Thanks a lot, Dr. Bani, for the kind intro. Great. Thank you.
So I'll start presenting my screen. Right, so good afternoon, everyone, and now welcome to session 5, where we shift gears into one of the most probably clinically actionable components of a whole genomic testing report, which can benefit not only just patients but healthy individuals alike. So in this session, we'll explore the role of pharmacogenomics, which is basically how a person's genetic makeup can influence their response to medications. So we will walk through key guidelines to understand how to interpret pharmacogenomic findings in a whole genome sequencing report, and most importantly, how to translate those into clinical action. So this story about pharmacogenomics actually goes way back than we can probably think about. We all have heard about Pythagoras, and we all know how much he loved triangles. Pythagoras is believed to have warned his followers not to eat fava beans. In fact, he was very infamously known to have a very strong aversion to fava beans, which is coincidentally how I also feel about maths and Pythagoras theorems. And why is that? Because Pythagoras used to think that fava beans can suck the life out of a person's soul. Some of them unknowingly, now we know, would develop a hemolytic crisis if some people would develop a hemolytic crisis if they consume fava beans because of a condition we now understand as G6PD deficiency. So if in a person there is this particular condition present, which is known as G6PD deficiency, the compounds that are present in fava beans can be very toxic to them, causing a hemolytic symptom. So this is one of the earliest records of a pharmacogenetic condition that was also very well documented in Pythagoras' life too. The lesson here is timeless. There is no such thing as an average patient. Genetic diversity that is there amongst us humans and amongst us as a country especially makes drug responses incredibly variable, and thus the one-size-fits-all approach of traditional medicine can lead to harm in a certain section of patients who just simply are not average. So to illustrate this is a very heart-breaking case that shows us how the stakes of not doing pharmacogenomic screening properly will lead to. So Dr. Anil Kapoor was a practicing physician who was diagnosed with stage 4 colon cancer. He was started on a 5-fluorouracil therapy, which is a very standard chemotherapy agent in case of colon cancer. But within weeks, he died not from the cancer that he had, but from toxicity from the treatment that was supposed to make him feel better. A DPYD gene variant that increases the risk for 5-fluorouracil toxicity went undetected during his limited pre-screening test that he underwent. And this case reminds us that the adverse outcomes from standard, very commonly used drugs for certain conditions are not rare. They are preventable if we look into the genome first. So this jar shown in the picture here holds approximately 7,000 M&Ms, which are basically candies that in India we call as gems. So each one represents a person in the US who died over a 10-year period from preventable toxicity to fluoropyrimidine drugs like 5-fluorouracil or capecitabine due to an untested DPYD deficiency. The advocacy group behind this display uses this visual to ask a very simple question, that how many lives could be actually saved if PGx testing were standard before even prescribing these drugs. The number isn't hypothetical for USA's case, it is right there in the jar. And when you think about such things happen in India, most of them go undocumented, and thus we don't even know how many M&Ms we would have to put in a jar that is labeled for India. So when we prescribe medications without knowing the patient's genomic background, we are actually gambling. So this slide shows just how ineffective that gamble often is. Up to 75% of patients on cancer drugs and approximately 70% patients on Alzheimer's related medications don't even respond to these drugs as expected. And these are not rare exceptions, these are the majority. So 75%, if you think about it, is a very staggering number of people who are not getting the prescribed benefit from the therapy that they are assigned. And thus, these are not rare exceptions, they are actually the majority of cases that are occurring in these particular clinical scenarios. And this is why precision medicine and pharmacogenomics in particular is our way out of this gambling inefficiency. It is not about replacing the drugs, it is about knowing who will be benefited from which drug and who won't be benefited. So here is the difference between classical and personalized medicine. Traditional care treats all patients with the same diagnosis and symptoms in probably the exact same way, but the outcomes can be wildly different. One patient might improve based on a particular therapy, another might not have any response at all, while a third category of patient, which is not in the range of the average patient that has been clinically described in previous textbooks, a severe adverse effect can also be seen. But with personalized medicine, we consider the patient's unique genetic sequence, and because a few changes in their DNA, like even a single base substitution, can inform which drug and how much dose and what risk are we dealing with while prescribing a particular therapy. And this is then the core of pharmacogenomics, tailoring care to the individual based on their genetic makeup. Let us now talk about a very real world example of a commonly used cancer chemotherapy drug, which is fluoropyrimidine drugs. These are some of the most commonly used chemotherapies prescribed for cancers of the colon, breast, lung, and many more solid tumors. Drugs like 5-fluorouracil or 5-FU, capecitabine, and tegafur are all different forms of these fluoropyrimidine drugs, 5-fluorouracil being the intravenous kind, and tegafur and capecitabine being the oral ingestible kinds. All of these work by interfering with DNA synthesis. But despite their widespread use, they can be life-threatening in patients with certain metabolic deficiencies. And this is where pharmacogenomics becomes not just useful but life-saving in the case of these particular patients. So to explain pharmacokinetics and pharmacodynamics, let's see a simplified view of how fluoropyrimidine drugs are metabolized in the body. So capecitabine and tegafur, for example, which are the oral forms of this particular drug, are converted into 5-FU, the active form of this drug inside the liver. Now, the majority of the drug, which is over 80%, needs to be broken down and cleared by different enzymes like the DPD enzyme that is encoded by the DPYD gene. And thus, because this particular gene plays a very important role in the metabolism pathway and the breakdown pathway of this particular drug, if a patient has a mutation in this particular gene, the breakdown process may fail because the gene now does not function properly. This will result in toxic metabolites accumulating in the body, leading to severe or even fatal toxicity in these patients. And this is why understanding this pathway and in essence the role of DPYD gene and testing for genetic variations in this gene becomes very important before starting chemotherapy with 5-fluorouracil. So the DPYD gene, which is located on chromosome 1, encodes an enzyme called DPD. This enzyme plays a very critical role in inactivating fluoropyrimidine drugs. So individuals with DPYD variations have reduced or absent DPD activity, putting them at a higher risk for drug toxicity. These patients need drastically reduced doses or even alternate treatments altogether based on how much deficient are they in that DPD enzyme. And the best part of all of this is that this risk can be very well identified with a single genetic test, ideally before a single dose of 5-fluorouracil is even administered to this particular patient. So thus we now have international consensus on the importance of DPYD testing before fluoropyrimidine therapy. And these have been around for a very long time now. The US FDA, for example, has already issued warnings on drug labels for 5-fluorouracil, and the ESMO guidelines recommend germline testing to reduce the risk of severe toxicity using DPYD germline testing. And very recently, the NCCN guideline for solid tumors in gastrointestinal cancers have also recommended that testing for germline variations in DPYD should be done. And these are not just Western issues. It's not a problem of USA, for example. It is just that they might have a lot more numbers to show for it, but we don't have that kind of evidence yet in India. But the Indigen study, which is a study that sequenced approximately 1,000 healthy individuals from India, we found in that data that 3% of the Indian population carries variations that increase the risk of 5-fluorouracil toxicity. And up to 10% of the population might actually need a reduced dose for this particular drug. Now, the percentage may seem small, 3%, but if you put it in context of India's population of several crores, this number is actually in millions. So, like millions of people in India would benefit from a reduced dosage or an alternative therapy compared to 5-fluorouracil. These numbers reflect preventable risk and actionable insight that we can then offer to these millions of people. Pharmacogenomics isn't just a Western luxury as previously thought about. It is an Indian necessity actually. A 2022 study showed that every individual in India on average carries at least 8 clinically actionable pharmacogenomics variants. And this includes warfarin sensitivity, altered antipsychotic metabolism, and even anesthesia risks. For example, in a community called the Arya Vaishya, individuals show a higher incidence of adverse drug reactions due to pseudocholinesterase deficiency. Pharmacogenomics can thus save lives and improve drug-related safety, especially in our population, which is genetically very diverse, even within the country. So this landmark study that was done by the Marshfield Clinic in USA analyzed whole genome sequencing data from 300 deceased individuals that had a very rich EHR data available. And they found that 93% of these carried at least one clinically actionable pharmacogenomic variant, and 1 in 3 of these patients had a CYP2D6 loss of function variation. When extrapolated to the general population, in summary, 80% of the people who were admitted to this particular clinic and deceased would have benefited from pharmacogenomic testing by the age of 50 itself. These are not rare findings. This should be actually the rule and not the exception. PGx or pharmacogenomics is not a niche area anymore. It is a population scale medicine approach that should be applied. And now interpretation of pharmacogenomics data must be rooted in evidence-based guidelines. And this slide highlights exactly that what are the key global bodies that are influencing these guidelines. So Consortiums like CPIC, DPWG, and PharmGKB, which grade gene-drug relationships based on the strength of evidence, are something that are supporting reporting of pharmacogenomic variants in WGS reports. These organizations thus ensure that what we report is clinically meaningful, reproducible, backed by evidence, and it is not just data that we give off to the patient. So for each variant drug pair, they guide us on whether we can recommend dosage changes, avoidance, and or increased monitoring for that particular patient. So for example, PharmGKB, a database that that curates all these guidelines given by different consortium into one single place and categorizes these gene drug associations into different levels. Level 1A being the gold standard for reporting. So these are variation or actually gene and drug associations with a particular variation that are supported by clinical guidelines or FDA drug labels. Level 1B being the kind of gene drug pairs that still carry strong evidence, though without formal prescribing instructions. So levels 2 to 4 represent decreasing confidence or applicability of this evidence in clinical care. And in a clinical setting, PharmGKB recommends that we prioritize level 1 variants while reporting, ensuring that recommendations we make are actionable and supported by solid science that these consortiums have gathered evidence for. And here's the sheer scope of pharmacogenomic relevance in our everyday clinical practices. At least 98 drugs across different therapeutic areas already have a level 1A or 1B PGx annotation in PharmGKB. And you will probably see names that are very familiar to things like oncology. So, for example, 5-fluorouracil, capecitabine, tamoxifen, which is very regularly given in breast cancer, irinotecan, and many others. So, pharmacogenomics is not just restricted to one specialty, it cuts across medicine, giving every clinician a region or a reason to engage with genomics for their patients. So among PharmGKB's 29 level 1A and 1B pharmacogenes, 7 are directly oncology associated, including genes like CYP2D6, DPYD, UGT1A1, and EGFR. And these are the genes that affect how patients respond to 11 key drugs that are commonly used in cancer or are commonly prescribed to cancer patients. For these, we already have 35 known variants or haplotypes that can significantly alter response, toxicity, or dosage if needed. So these aren't just futuristic findings that we may need to report to our patients sometime later. These are something that are useful now and should thus be reported if detected. So when we interpret a pharmacogenomic variant, we are not just identifying a mutation in this case, we are also predicting the phenotype that the patient will display. So does the variant increase the risk of adverse drug reactions? Should we reduce the dose? Will the drug even work on the patient? All these questions can be answered by the framework that is shown here about how a single variant can influence everything from gene activity to drug metabolism to clinical outcomes in a patient. And our jobs as clinical interpreters of whole genome sequencing data that also might include a pharmacogenomic report is to translate these insights into clear, actionable recommendations for a patient. And this visual shows how single nucleotide variations in very important pharmacogenes influence cancer therapy. DPYD alone is linked to 15 variant drug interactions that are mentioned in level 1A and 1B guidelines of PharmGKB, many of which increase toxicity risk with drugs such as 5-fluorouracil and capecitabine. EGFR and NUDT15 also play roles in predicting efficacy and dosing of these particular drugs. And when these variants are level 1, we know that the association is strong. It is backed by a lot of evidence as curated by a lot of different consortia and are thus enough to impact treatment decisions. And this is, for example, where genomics meet, germline genomics, in fact, meets oncology. Beyond SNVs, haplotype level variations also have a very profound impact on how a patient might metabolize a particular drug. So, for example, a very important pharmacogene CYP2D6 influences tamoxifen and ondansetron response. G6PD impacts safety of a particular drug called rasburicase, and so on. And thus, TPMT and NUDT15 guide dosing information for other cancer-related drugs also. So each connection here represents a chance to prevent harm or improve efficacy, especially for cancer patients, where every dose actually counts towards their chances of survival. So, to put all of this in context, let's now start by understanding a few examples of how a pharmacogenomics report might look like and how we can interpret each of the sections that are mentioned in a probable pharmacogenomic report. So here is a snapshot of what a real pharmacogenomics report might look like. It lists some very important information about a particular pharmacogenomic variation, such as key drug-gene interactions that might have something of clinical relevance. For example, a variant in DPYD gene influencing fluorouracil metabolism is mentioned. In CYP2C9 affecting warfarin, poor metabolizer status has been mentioned in this report. For NUDT15 gene linked to mercaptopurine dosage modification, this is also mentioned in the report. There is an EGFR variant mentioned in the report that influences the usage of the drug gefitinib. And finally, there is an entry for UGT1A1 gene, which impacts irinotecan with genotype-guided dosing. So, for each of these, we can see that these are supported by a level 1A evidence, meaning that there is a strong guideline for action for each of these variations. Now, if we go step by step into interpreting each one of them, so let's first zoom into the DPYD5 or the 5-fluorouracil associated variant. You can see in the genotype that the patient for that particular variation has a genotype of TT. So thus the patient is homozygous for the risk allele or the alternate type allele, which leads to a no enzyme function for this particular gene called DPYD. And now this is a significant finding because 5-fluorouracil or 5-FU is very commonly used in chemotherapy, which is what we've seen in our previous slides. And a non-functional or a no-function phenotype that is shown in this particular report means that the drug can accumulate, causing life-threatening toxicity for this particular patient. So for this patient, dose reduction or alternative therapy becomes very critical. And this variant is classified as level 1A by PharmGKB, meaning that there is a strong guidance for action in this case. So here's another example from the same report for a gene called CYP2C9 that affects warfarin metabolism. So this patient in particular carries two types of haplotype variants for this particular gene called CYP2C9, which is STAR3 and STAR2. The combined effect of having this STAR2 and STAR3 haplotypes is that it makes the patient a poor metabolizer of warfarin. This means that the patient processes warfarin much slower, increasing the risk of bleeding complications unless the dose is adjusted. And again, this is also based on a level 1A evidence coming from PharmGKB, thus guiding towards actionable clinical guidelines. And of note, we see that in the current guidelines, STAR2 is a decreased function allele and STAR3 on its own is a no function allele. So this particular genotype should prompt immediate dose consideration or alternate anticoagulation strategies for these particular patients. And now this particular example highlights drug efficacy and not toxicity like the previous two examples. So here we are looking at a variation in the EGFR gene and how it may impact a drug called gefitinib, which is used in the treatment of non-small cell lung cancer. The patient has a TT genotype at this particular variant position, and if you may notice that this is now the reference, this is now not the reference genotype, it is now sorry, I misspoke. This is now the reference genotype as compared to the alternate genotype that we saw in the case of the TPYD variant, meaning that the patient is homozygous for the reference type now. In essence, meaning that this variation is not present in the patient at all. And this particular genotype, which basically means that the patient lacks the activating mutation required for optimal response to gefitinib, means that there is a decreased drug efficacy in this particular patient, meaning that gefitinib may not work well in this particular individual. So thus whole genome sequencing can help identify patients who are more likely to benefit from a particular therapy, also reducing wasted time and toxicity for a particular drug. So again, we see that the level of evidence here is also level 1, guiding this decision. And now this is a case of dosage adjustment based on a gene called UGT1A1 and its genotype. The drug in focus in this example is irinotecan, which is a chemotherapy agent too. The patient has the genotype *1*1 for this particular gene, and thus is a normal metabolizer. And being a normal metabolizer, this patient is likely to tolerate a higher dose of this particular drug without the increased risk of toxicity. Had the patient carried a decreased metabolism or a no function allele in this particular gene, for example *28 or *6, then they would have been a slow metabolizer or a poor metabolizer, which would mean that we would need to adjust the dose due to risk of neutropenia in these particular patients. But since our patient here, for example, is *1*1 and the guideline says that this particular genotype is a normal metabolizer, they are able to tolerate a higher dose without the increased risk of toxicity. So this particular case now highlights how fine-tuning can be possible using these pharmacogenomics insights that can ensure both safety and efficacy of a patient of using a drug in a patient. And now the power of whole genome sequencing that we have discussed in our previous slides also is that it captures all types of genetic variations from large chromosomal abnormalities to single nucleotide variations in a single test and on a single platform. So this means that pharmacogenomics information for anyone coming in for a WGS test can also be provided using the same test. And thus, we not only can give a diagnosis for a patient along with the carrier status and all for a particular patient who is coming in for a whole genome sequencing test, we can now also give them information that can help inform treatment strategies in a better and more genome-informed way. So we can thus truly personalize medicine for them by helping them throughout their clinical journey, starting from diagnosis and supporting throughout through therapy planning. So whole genome sequencing gives us a complete view of pharmacogenomics compared to whole exome sequencing because it captures what standard tests like whole exome sequencing have often missed, especially variations in intronic and regulatory variations, mitochondrial drug responsive variations, structural variations in very important pharmacogenes like CYP2D6 and G6PD, along with high resolution HLA genotyping. So all of these applications that I have mentioned here are something that have been included in the level 1A and 1B markers in PharmGKB. And in total, whole exome sequencing misses key variants in 12 of the 123 PharmGKB level 1A, 1B markers, many of which are very critical for guiding dosing for drugs like warfarin and 5-fluorouracil. But with whole genome sequencing, as we have mentioned multiple times, we can truly achieve a comprehensive pharmacogenomics analysis for these particular 111A and 1B markers, along with supporting the discovery for newer markers in these very important pharmacogenes. So put a number to all of this. Here is a real world example of what is actually missed in whole exome sequencing. So if you look at these variants in DPYD, VKORC1 and MTRNR1 genes, they are all linked to very key and commonly used drugs in certain clinical areas like fluorouracil, warfarin, capecitabine, different antivirals and antibiotics. Mitochondrial variations, which are something that are not captured in a standard whole exome sequencing test, is something that is very well detected in a whole genome sequencing test in the same using the same sample and the same approach. So many of the variations that we see here, intronic, intergenic, lying outside coding exons, and mitochondrial DNA variants are basically invisible to a standard whole exome sequencing test. And some of them, as you can see by their allele frequencies given in the indigenous population, these are quite prevalent in India. So if these patients were to be sent for a whole exome sequencing test, these variants are something that we would have missed for them. But yet they have a very clear clinical relevance, and they should have been acted upon if detected, which only WGS right now will enable us to do. So to summarize pharmacogenomics reporting, PGx in essence allows us to connect genome to drug response, thus personalizing therapy for all of us. Whole genome sequencing ensures that we do not miss any clinically actionable pharmacogenes and the variants that are lying in these pharmacogenes. Guidelines for from different consortiums such as CPIC, PharmGKB, and others can standardize interpretation of these particular variations. Reports must clearly link genotype to recommended actions for these particular drug and variant pairs, like dosage changes or alternative drug to be recommended, and so on. But most importantly, pharmacogenomics must always be interpreted in a clinical context. It is not a standalone report, but it is a part of the precision care that we now want to lend to our patients. So with this, I will stop and I will be happy to take up any questions. So please feel free to drop them in the chat box. Thank you. So I see there's still a lot of questions for the previous sessions, and we will try our best to answer all of these to either the chat or by getting back to you post this session is completed. And I request all of you, if you have any pharmacogenomics related questions, to please post it in the chat box. Okay, so we have one: is pharmacogenomics being practiced in India at present by our leading oncophysicians? Sadly, it's not even that this is the case in India, this is the case everywhere, that despite having such strong recommendations from even the US FDA, for example, this kind of testing is not being routinely done, which is a very sad thing if you think about it, because these are the kind of toxicity related risks that we are putting our patients in by avoiding doing a very simple test. So right now we have started increasing awareness for pharmacogenomics amongst different clinicians, but I would say we still need to do a lot more so that every patient that is undergoing a therapy that does have a pharmacogenomic recommendation to it should get tested. Can anti-epileptic drugs have an adverse effect based on dosages? Yes, so even some very widely used anti-epileptic drugs do have a pharm-GKB level annotation for them in the level 1A and 1B itself, and thus making it very clinically actionable. So, based on the different evidence that is available for these particular drugs, we can then go in for a dosage reduction or an alternative therapy prescription for these particular drugs, all depending on what is the current evidence for these particular drug and gene pairs. And yeah, just like the ACMG AMP guidelines revolving around pharmacogenomics reporting is also something that evolves very quickly as new evidence starts coming up. So even if there are some genes and drugs that are currently in a lower level of evidence, as more evidence starts coming up and more people start doing these studies and start building the evidence around it in different populations, these might get promoted to a clinically actionable level, which is level 1A and 1B. So where can we check the guidelines for pharmacogenomics reports? So these can be checked either on the CPIC website, wherein the latest guidelines for very standard drugs is given. And if you want a consolidated information about pharmacogenomics guidelines coming from different institutions, consortiums, and organizations, then PharmGKB is a great database that actually curates and collates all of this information in one place. But the key here is to stay updated, because guidelines are continuously evolving, and thus what is relevant right now what is not relevant right now may become relevant sometime in the future as new evidence comes up. Okay, so Kautilya asks a very important and a very interesting question, whether WGS reports pharmacogenomics variations normally, or any pharmacogenomic WGS test needs to be separately requested. So by its nature, a whole genome sequencing test can report pharmacogenomics variations along with the indication for which the request was probably requested. So it might be a good practice to report in a whole genome sequencing report everything that might be of benefit to the patient, which includes things that are clinically relevant, immediately clinically relevant, and in the context of people who are healthy currently and just come in for a screening. These things can include a report on pharmacogenomic variants, which can then help guide dosage guidelines for very commonly used drugs in these particular persons also, in fact. So yes, so in essence, pharmacogenomic variants can be included in a standard whole genome sequencing test. Okay, so I think there is a lot of interest about if pharmacogenomics variations can be included in a standard WGS test. So I can confirm here that the WGS test reports that we offer as a part of Karkinos Healthcare is something that does have pharmacogenomic information, even if you are referring for the test as a patient for a particular condition, or you are coming in for screening as a healthy individual also. So yes, pharmacogenomics is something that we on routine report in our whole genome sequencing tests. Okay, Dr. Preeta has asked any studies on statins and cardiogenic drugs. So for statins, I can confirm that pharmacogenomics is something that is very, very relevant, because Firstly, familial hypercholesteremia is something that we have previously found to be very prevalent through genomic studies in India. And a lot of people are carrier for these particular conditions, and statins is something that is very regularly given to these particular patients. And yes, there are guidelines included in PharmGKB level 1A and 1B that influences the dosage of statins. So considering that India is a population in which familial hypercholesteremia is prevalent, and meaning that there will be a high percentage of population that will be put on these particular drugs called statins, it makes a lot of sense to include pharmacogenomic markers for these drugs while screening for familial hypercholesteremia also. So in one single test, you can not only give them a not only state the risk of developing hypercholesteremia, you can also give them a treatment strategy regarding the usage and the dosage of statins that might be more suitable to their genotype. Okay, so somebody has asked an interesting question about how posology or dosage adjustment as a part of pharmacogenomics can be done. So, for every drug that has a pharmacogenomic guideline by CPIC, for some cases they also give a very standard guideline on how to uh, influence the dosing for that particular drug. So all this information for a particular gene and drug pair is available on their website, and these guidelines are something that are updated very regularly. So for example, if some lab sees that a particular DPYD variation is not currently included in a guideline, but they have screened their population of patients who had an adverse drug reaction to 5-fluorouracil, and they deem that it is functionally now proved that the variation may lead to a severe or a toxic effect in these particular patients, it might then be added to a newer set of guidelines. And thus the dosage recommendations can also and then also actually follow. And for things like DPYD and CYP2D6, PharmGKB is also something that captures all these dosage guidelines that have been recommended by CPIC for these particular drugs and genes. Okay, so somebody has asked, pharmacogenomics links genome to drug response, but how do we account for environmental or lifestyle factors that may override genetic predictions in real world settings? So, very important question. And the answer here being that everything that is being reported as a clinically actionable pharmacogenomic finding is something that indicates that you are at an increased risk of developing that particular phenotype compared to other people. So, for example, if I have a variation in the DPYD gene that is included in level 1A guidelines by PharmGKB, I am at an increased risk of developing toxicity to 5-fluorouracil compared to other people who do not have that variant. So it is not a direct correlation causation link that is being given here, that if I have the variation, it is 100% sure that the particular phenotype will be given or will be shown by me if that therapy is administered. We are only stating, or the guidelines in fact are only stating, that the people who harbor the mutation are at an increased risk or at a significantly higher risk compared to people who do not have that variation. So there is a question about how to best test CYP2D6. So for not just CYP2D6, for other very important pharmacogenes like CYP2B6 also, for example, we now know that these haplotype variations, some of them are made up of variations that lie in non-coding regions of these particular genes, or even in intronic regions of these particular genes. including some deletions that span the intronic regions also. So in essence, for these genes, whole exome sequencing or a capture-based test might not be a good approach to do so, because it might not be able to capture the clinically relevant haplotypes in these very important pharmacogenes. And thus, whole genome sequencing is something that can be very easily used to solve this problem, because it not only captures this data and calls variants very accurately. Most tools that are using whole genome sequencing data have now evolved to very sophisticatedly and accurately and sensitively detect and call haplotypes for CYP2D6 and CYP2B6, for example. So, for example, the sequencing and so the secondary analysis platform that comes along with the Illumina sequencing platforms, which is the onboard Dragon, it has a specialized caller for genes like CYP2D6 and CYP2B6, meaning that they have very well tried, tested, and validated calling of haplotypes for these two very important pharmacogenes using the short read WGS data that is generated on their sequencers. Sindhu Sahu has asked, is precautionary pharmacogenomics testing done? And I will say yes, it is done, and it is something that is probably very important to do, because we might be healthy now, but we might require these particular drugs that were shown in this session at some point of time in our life. So, for example, as I mentioned the example of familial hypercholesteremia, once the symptoms start manifesting, you might also need to be prescribed statins. But before prescription, if we already had a precautionary or a preemptive pharmacogenomic test done for that particular drug, we would have had the information that would have guided not just dosage recommendation, but also if that drug should be given to me or not. So yes, preemptive pharmacogenomics testing is important. It should be done, and it is something that we do in essence follow reporting here at Karkinos too. Can AI or machine learning be integrated with pharmacogenomics and WGS data to predict adverse drug reactions that have not yet been documented? So I will answer this in probably two ways. That yes, right now currently the guidelines that are that include the guidelines that currently include a set of genes or variations for particular drugs, and the recommendations that are given are limited in the sense that they are based on the evidence that is being gathered till now. But since, for example, for India, we really do not have that data yet, that if there are any rare variations in our population that can also, for example, influence DPYD gene function and thus lead to 5-fluorouracil toxicity, this is still now understudied, which is why it makes it a very important point to screen more people in the population to understand what are the rare variants that we have that are not shared amongst other populations, especially in the Western nations, which we might miss because current guidelines do not, for example, take into account limited data coming from other countries. So to solve this problem of understanding variations that are very specific to our population, machine learning can now probably be augmented over the current testing and analysis efforts that we are doing. So it is not something that is straightforwardly applicable to WGS data, but it is something that can support detection of these particular rare variants that our country might have, or novel variations that may be detected in any population, which can help support building this evidence for future guidelines. I will probably take one last question. Can PGx reports be automatically generated using specific pipelines with WGS analysis? So, in terms of its doability, yes, it can be done. Whether there are tools that are openly available right now, I don't think so. There are any tools that can support a direct PGx analysis using WGS data. But this is something that is a very simple bioinformatics and in general an informatics problem to solve. So it can be done, it can be implemented, and PGx reporting is something that is actually automated here in our reports at Karkinos. Okay, I'll take the last question here. Is there a list of drugs for which pharmacogenomics is recommended? Yes, there are approximately 98 drugs that are included in PharmGKB level 1A and 1B guidelines, which is something that is directly clinically actionable. So these 98 drugs, the list can be found on PharmGKB, or it is also available in the slides that we have here. These are the ones that have a very direct clinical implication in terms of the commonly used drugs that we see. So yes, this list is available and people can go through it.
Okay. Thanks a lot for that wonderful session, Dr. Bani. It was really helpful for all of us. Again, pharmacogenomics is one of the key topics that I continue to explore on a personal basis. With a lot of learnings that I'm getting from a lot of sources. That is something that I feel like that has the most relevance to clinical actionability based on genomics for a lot of patient care globally. It's wonderful that you shared details on that. Now a quick break from the learning. Let's go for our lunch breaks and be back at around 1:45 maybe. I guess that a 45-minute lunch break would work best for everyone. So please stay tuned to us. We will be back shortly after the lunch break. And we will continue to the next session on incidental and actionable genetic findings by Dr. Bani Jolly. So looking forward to continue learning along with all of you and sharing our knowledge and answering your questions. Thank you.
Welcome to Karkinos Healthcare's network of Advanced Center for Cancer Diagnostics and Research Labs. As a technology-led, purpose-driven oncology platform, we operate 6 state-of-the-art labs across more than 120,000 square feet. Our histopathology division offers a complete range of services. From routine tissue analysis to advanced immunohistochemistry for biomarker detection, fluorescence. Welcome to Karkinos Healthcare's network of Advanced Center for Cancer Diagnostics and Research Labs. As a technology-led, purpose-driven oncology platform, we operate 6 state-of-the-art labs across more than 120,000 square feet. Our histopathology division offers a complete range of services. From routine tissue analysis to advanced immunohistochemistry for biomarker detection, fluorescence in situ hybridization for genetic abnormalities, and special stains for enhanced tissue insights. Every sample receives the most detailed and clinically relevant evaluation. Efficiency is at the core of our operations. Through advanced automation and technology, we ensure industry-best turnaround times, cost efficiency, and seamless logistics, delivering temperature-controlled sample collection and rapid reporting right to your doorstep. As part of our molecular diagnostics, we offer over 70 unique molecular tests which are tailor-made to provide clinicians with critical therapeutic, prognostic, diagnostic, and actionable insights. Our test menu is continually evolving to reflect the latest research and clinical guidelines, ensuring access to state-of-the-art diagnostics. We employ cutting-edge technologies, including the thermo sequencing platform, featuring the Ion Chef for automated sample prep and the Ion Gene Studio S5 sequencer for diverse oncodiagnostic applications. The Genexus system offers a fully automated NGS workflow, from sample to report. At the heart of our innovation is sequencing at scale. 2 NovaSeq 6000 systems with 3 Illumina NovaSeq X Plus. We also house India's first PacBio Revio ultra-long read NGS sequencing platform. With India's largest private sequencing capacity, we offer rapid, targeted, custom, and comprehensive genomic sequencing for clinical and research needs at affordable costs, while also delivering results twice as fast as the industry standard. Our team of leading Indian scientists, clinicians, and technologists, combined with our commitment to scientific rigor, truly distinguishes Karkinos. Join us in making personalized medicine a reality for every Indian. At Karkinos, we believe every life deserves the best chance. And we are here to make that vision a reality for all, today and for generations to come. Welcome to Karkinos Healthcare's network of Advanced Center for Cancer Diagnostics and Research Labs. As a technology-led, purpose-driven oncology platform, we operate 6 state-of-the-art labs across more than 120,000 square feet. Our histopathology division offers a complete range of services. From routine tissue analysis to advanced immunohistochemistry for biomarker detection, fluorescence in situ hybridization for genetic abnormalities, and special stains for enhanced tissue insights. Every sample receives the most detailed and clinically relevant evaluation. Efficiency is at the core of our operations. Through advanced automation and technology, we ensure industry-best turnaround times, cost efficiency, and seamless logistics, delivering temperature-controlled sample collection and rapid reporting right to your doorstep. As part of our molecular diagnostics, we offer over 70 unique molecular tests which are tailor-made to provide clinicians with critical therapeutic, prognostic, diagnostic, and actionable insights. Our test menu is continually evolving to reflect the latest research and clinical guidelines, ensuring access to state-of-the-art diagnostics. We employ cutting-edge technologies, including the thermo sequencing platform, featuring the Ion Chef for automated sample prep and the Ion Gene Studio S5 sequencer for diverse oncodiagnostic applications. The Genexus system offers a fully automated NGS workflow, from sample to report. At the heart of our innovation is sequencing at scale. 2 NovaSeq 6000 systems with 3 Illumina NovaSeq X Plus. We also house India's first PacBio Revio ultra-long read NGS sequencing platform. With India's largest private sequencing capacity, we offer rapid, targeted, custom, and comprehensive genomic sequencing for clinical and research needs at affordable costs, while also delivering results twice as fast as the industry standard. Our team of leading Indian scientists, clinicians, and technologists, combined with our commitment to scientific rigor, truly distinguishes Karkinos. Join us in making personalized medicine a reality for every Indian. At Karkinos, we believe every life deserves the best chance. And we are here to make that vision a reality for all, today and for generations to come. Welcome to Karkinos Healthcare's network of advanced Center for Cancer Diagnostics and Research Labs. As a technology-led, purpose-driven oncology platform, we operate 6 state-of-the-art labs across more than 120,000 square feet. Our histopathology division offers a complete range of services. From routine tissue analysis to advanced immunohistochemistry for biomarker detection, fluorescence in situ hybridization for genetic abnormalities, and special stains for enhanced tissue insights. Every sample receives the most detailed and clinically relevant evaluation. Efficiency is at the core of our operations. Through advanced automation and technology, we ensure industry-best turnaround times, cost efficiency, and seamless logistics, delivering temperature-controlled sample collection and rapid reporting right to your doorstep. As part of our molecular diagnostics, we offer over 70 unique molecular tests which are tailor-made to provide clinicians with critical therapeutic, prognostic, diagnostic, and actionable insights. Our test menu is continually evolving to reflect the latest research and clinical guidelines, ensuring access to state-of-the-art diagnostics. We employ cutting-edge technologies, including the thermo sequencing platform, featuring the Ion Chef for automated sample prep and the Ion Gene Studio S5 sequencer for diverse oncodiagnostic applications. The Genexus system offers a fully automated NGS workflow, from sample to report. At the heart of our innovation is sequencing at scale. 2 NovaSeq 6000 systems with 3 Illumina NovaSeq X Plus. We also house India's first PacBio Revio ultra-long read NGS sequencing platform. With India's largest private sequencing capacity, we offer rapid, targeted, custom, and comprehensive genomic sequencing for clinical and research needs at affordable costs, while also delivering results twice as fast as the industry standard. Our team of leading Indian scientists, clinicians, and technologists, combined with our commitment to scientific rigor, truly distinguishes Karkinos. Join us in making personalized medicine a reality for every Indian. At Karkinos, we believe every life deserves the best chance. And we are here to make that vision a reality for all, today and for generations to come. Welcome to Karkinos Healthcare's network of Advanced Center for Cancer Diagnostics and Research Labs. As a technology-led, purpose-driven oncology platform, we operate 6 state-of-the-art labs across more than 120,000 square feet. Our histopathology division offers a complete range of services. From routine tissue analysis to advanced immunohistochemistry for biomarker detection, fluorescence in situ hybridization for genetic abnormalities, and special stains for enhanced tissue insights. Every sample receives the most detailed and clinically relevant evaluation. Efficiency is at the core of our operations. Through advanced automation and technology, we ensure industry-best turnaround times, cost efficiency, and seamless logistics, delivering temperature-controlled sample collection and rapid reporting right to your doorstep. As part of our molecular diagnostics, we offer over 70 unique molecular tests which are tailor-made to provide clinicians with critical therapeutic, prognostic, diagnostic, and actionable insights. Our test menu is continually evolving to reflect the latest research and clinical guidelines, ensuring access to state-of-the-art diagnostics. We employ cutting-edge technologies, including the thermo sequencing platform, featuring the Ion Chef for automated sample prep and the Ion Gene Studio S5 sequencer for diverse oncodiagnostic applications. The Genexus system offers a fully automated NGS workflow, from sample to report. At the heart of our innovation is sequencing at scale. 2 NovaSeq 6000 systems with 3 Illumina NovaSeq X Plus. We also house India's first PacBio Revio ultra-long read NGS sequencing platform. With India's largest private sequencing capacity, we offer rapid, targeted, custom, and comprehensive genomic sequencing for clinical and research needs at affordable costs, while also delivering results twice as fast as the industry standard. Our team of leading Indian scientists, clinicians, and technologists, combined with our commitment to scientific rigor, truly distinguishes Karkinos. Join us in making personalized medicine a reality for every Indian. At Karkinos, we believe every life deserves the best chance. And we are here to make that vision a reality for all, today and for generations to come. Welcome to Karkinos Healthcare's network of advanced Center for Cancer Diagnostics and Research Labs. As a technology-led, purpose-driven oncology platform, we operate 6 state-of-the-art labs across more than 120,000 square feet. Our histopathology division offers a complete range of services. From routine tissue analysis to advanced immunohistochemistry for biomarker detection, fluorescence in situ hybridization for genetic abnormalities, and special stains for enhanced tissue insights. Every sample receives the most detailed and clinically relevant evaluation. Efficiency is at the core of our operations. Through advanced automation and technology, we ensure industry-best turnaround times, cost efficiency, and seamless logistics, delivering temperature-controlled sample collection and rapid reporting right to your doorstep. As part of our molecular diagnostics, we offer over 70 unique molecular tests which are tailor-made to provide clinicians with critical therapeutic, prognostic, diagnostic, and actionable insights. Our test menu is continually evolving to reflect the latest research and clinical guidelines, ensuring access to state-of-the-art diagnostics. We employ cutting-edge technologies, including the thermo sequencing platform, featuring the Ion Chef for automated sample prep and the Ion Gene Studio S5 sequencer for diverse oncodiagnostic applications. The Genexus system offers a fully automated NGS workflow, from sample to report. At the heart of our innovation is sequencing at scale. 2 NovaSeq 6000 systems with 3 Illumina NovaSeq X Plus. We also house India's first PacBio Revio ultra-long read NGS sequencing platform. With India's largest private sequencing capacity, we offer rapid, targeted, custom, and comprehensive genomic sequencing for clinical and research needs at affordable costs, while also delivering results twice as fast as the industry standard. Our team of leading Indian scientists, clinicians, and technologists, combined with our commitment to scientific rigor, truly distinguishes Karkinos. Join us in making personalized medicine a reality for every Indian. At Karkinos, we believe every life deserves the best chance. And we are here to make that vision a reality for all, today and for generations to come. Welcome to Karkinos Healthcare's network of Advanced Center for Cancer Diagnostics and Research Labs. As a technology-led, purpose-driven oncology platform, we operate 6 state-of-the-art labs across more than 120,000 square feet. Our histopathology division offers a complete range of services. From routine tissue analysis to advanced immunohistochemistry for biomarker detection, fluorescence in situ hybridization for genetic abnormalities, and special stains for enhanced tissue insights. Every sample receives the most detailed and clinically relevant evaluation. Efficiency is at the core of our operations. Through advanced automation and technology, we ensure industry-best turnaround times, cost efficiency, and seamless logistics, delivering temperature-controlled sample collection and rapid reporting right to your doorstep. As part of our molecular diagnostics, we offer over 70 unique molecular tests which are tailor-made to provide clinicians with critical therapeutic, prognostic, diagnostic, and actionable insights. Our test menu is continually evolving to reflect the latest research and clinical guidelines, ensuring access to state-of-the-art diagnostics. We employ cutting-edge technologies, including the thermo sequencing platform, featuring the Ion Chef for automated sample prep and the Ion Gene Studio S5 sequencer for diverse oncodiagnostic applications. The Genexus system offers a fully automated NGS workflow, from sample to report. At the heart of our innovation is sequencing at scale. 2 NovaSeq 6000 systems with 3 Illumina NovaSeq X Plus. We also house India's first PacBio Revio ultra-long read NGS sequencing platform. With India's largest private sequencing capacity, we offer rapid, targeted, custom, and comprehensive genomic sequencing for clinical and research needs at affordable costs, while also delivering results twice as fast as the industry standard. Our team of leading Indian scientists, clinicians, and technologists, combined with our commitment to scientific rigor, truly distinguishes Karkinos. Join us in making personalized medicine a reality for every Indian. At Karkinos, we believe every life deserves the best chance. And we are here to make that vision a reality for all, today and for generations to come. Welcome to Karkinos Healthcare's network of advanced Center for Cancer Diagnostics and Research Labs. As a technology-led, purpose-driven oncology platform, we operate 6 state-of-the-art labs across more than 120,000 square feet. Our histopathology division offers a complete range of services. From routine tissue analysis to advanced immunohistochemistry for biomarker detection, fluorescence in situ hybridization for genetic abnormalities, and special stains for enhanced tissue insights. Every sample receives the most detailed and clinically relevant evaluation. Efficiency is at the core of our operations. Through advanced automation and technology, we ensure industry-best turnaround times, cost efficiency, and seamless logistics, delivering temperature-controlled sample collection and rapid reporting right to your doorstep. As part of our molecular diagnostics, we offer over 70 unique molecular tests which are tailor-made to provide clinicians with critical therapeutic, prognostic, diagnostic, and actionable insights. Our test menu is continually evolving to reflect the latest research and clinical guidelines, ensuring access to state-of-the-art diagnostics. We employ cutting-edge technologies, including the thermo sequencing platform, featuring the Ion Chef for automated sample prep and the Ion Gene Studio S5 sequencer for diverse oncodiagnostic applications. The Genexus system offers a fully automated NGS workflow, from sample to report. At the heart of our innovation is sequencing at scale. 2 NovaSeq 6000 systems with 3 Illumina NovaSeq X Plus. We also house India's first PacBio Revio ultra-long read NGS sequencing platform. With India's largest private sequencing capacity, we offer rapid, targeted, custom, and comprehensive genomic sequencing for clinical and research needs at affordable costs, while also delivering results twice as fast as the industry standard. Our team of leading Indian scientists, clinicians, and technologists, combined with our commitment to scientific rigor, truly distinguishes Karkinos. Join us in making personalized medicine a reality for every Indian. At Karkinos, we believe every life deserves the best chance. And we are here to make that vision a reality for all, today and for generations to come. Welcome to Karkinos Healthcare's network of Advanced Center for Cancer Diagnostics and Research Labs. As a technology-led, purpose-driven oncology platform, we operate 6 state-of-the-art labs across more than 120,000 square feet. Our histopathology division offers a complete range of services. From routine tissue analysis to advanced immunohistochemistry for biomarker detection, fluorescence in situ hybridization for genetic abnormalities, and special stains for enhanced tissue insights. Every sample receives the most detailed and clinically relevant evaluation. Efficiency is at the core of our operations. Through advanced automation and technology, we ensure industry-best turnaround times, cost efficiency, and seamless logistics, delivering temperature-controlled sample collection and rapid reporting right to your doorstep. As part of our molecular diagnostics, we offer over 70 unique molecular tests which are tailor-made to provide clinicians with critical therapeutic, prognostic, diagnostic, and actionable insights. Our test menu is continually evolving to reflect the latest research and clinical guidelines, ensuring access to state-of-the-art diagnostics. We employ cutting-edge technologies, including the thermo sequencing platform, featuring the Ion Chef for automated sample prep and the Ion Gene Studio S5 sequencer for diverse oncodiagnostic applications. The Genexus system offers a fully automated NGS workflow, from sample to report. At the heart of our innovation is sequencing at scale. 2 NovaSeq 6000 systems with 3 Illumina NovaSeq X Plus. We also house India's first PacBio Revio ultra-long read NGS sequencing platform. With India's largest private sequencing capacity, we offer rapid, targeted, custom, and comprehensive genomic sequencing for clinical and research needs at affordable costs, while also delivering results twice as fast as the industry standard. Our team of leading Indian scientists, clinicians, and technologists, combined with our commitment to scientific rigor, truly distinguishes Karkinos. Join us in making personalized medicine a reality for every Indian. At Karkinos, we believe every life deserves the best chance. And we are here to make that vision a reality for all, today and for generations to come.
Hello all. I guess there was a bit of a network difficulty. Sorry about that. Hello all, welcome back. Hope you had a great lunch. We had here as well. Up next, we'll be going ahead with a session by Dr. Bani Jolly from our team again, followed by our next session with Dr. Jyoti Bajpai Dikshit as well. Dr. Bani, the stage is all yours. Please go ahead. Thank you.
Thank you so much, Harish, and welcome back, everyone. I hope you had a nice break and are now energized and refreshed to continue learning about whole genome sequencing analysis. So, in this session, session 6, we will now be talking about specifically incidental findings in whole genome sequencing data. So basically, as we expand the scope of WGS, we begin to see that it is more than just a diagnostic test result. We uncover unexpected medically relevant insights also through this single test. So, here in this session, we'll explore a very key aspect of genomic reporting, which is what happens when a whole genome sequencing test uncovers something unexpected, yet something that is still clinically important. So, we'll talk about what these findings are, how they are categorized, how to handle them, and what are the ethical and clinical implications about reporting them. So, this is where basically genomics and whole genome sequencing goes beyond diagnosis into long term health foresight. So, let me start this with a story, a hypothetical one, of a 34-year-old man who is perfectly healthy and goes in for a routine hernia operation. It is a common low risk procedure, but after surgery, something is off. The patient is not waking up, hours pass, and he thus needs ICU care, ventilation, and monitoring. So, for a simple low risk procedure, what is something that went wrong here? It wasn't the surgery, it was the anesthetic drug that was given, a drug that his body could not metabolize properly. And the reason here being that a genetic variant that no one suspected he had was causing this kind of side effect to this particular anesthesia drug. So this wasn't bad luck, it was hidden biology. Something a whole genome sequencing test, for example, could have revealed even before anyone would have picked up a scalpel to treat this particular patient. When we looked deeper, we found something very fascinating. This patient belonged to a community called the Arya Vyasa community, or the Chettiar community as they are very popularly known, which is a very close knit trading community with shared ancestry and unique health patterns. In fact, they claim through their folklore that they are the original descendants of the Aryan race, and hence the name of the community. Turns out this community has a significantly high incidence of a condition known as pseudocholinesterase deficiency, which is an inherited trait that makes common anesthesia drugs like scoline potentially life threatening to these people. And this is something that is prevalent not just here. Similar findings have been seen in other populations also, including the Multani community in Pakistan, among some Middle Eastern tribes, and even in parts of Europe like Germany. So, what this tells us is that genetic risks can actually cluster in populations, and sometimes unlocking the key to understanding these key risk factors lies both in our DNA and our clinical history and our ancestral history. What is powerful is what happens after. So, if a WGS test was done for this particular patient, it would not just explain the reaction, it would have changed the course of care. The care team that was involved in treating that particular patient could have avoided that drug altogether in future procedures. Furthermore, the patient's sibling for example, could also have been tested, and if found to have the same risk, could have been informed of the associated risks as well. So therefore, not just the patient, but their family could also be saved from this potential disaster. And that is the power of whole genome sequencing now. It gives us a chance to intervene before the harm actually occurs, not just for the patient, but also their family. And this is a perfect example how incidental findings can turn into something that is surprisingly actionable and even into life saving insights, for example. So, traditionally, as we have seen in the first session, also, we've practiced a phenotype first medicine kind of an approach where we wait for symptoms and then investigate. Genomics has flipped that approach. With whole genome sequencing, we can now adopt a genotype first approach where we start by sequencing the genome, identifying risk variants proactively, and then acting early, even in apparently healthy individuals. So we are shifting from reaction to anticipation, from diagnosis to prevention. As a very common saying goes, that prevention is better than cure. So we can now go from what went wrong to what could go wrong and how do we stop it. And this is where whole genome sequencing and genomic screening comes into play, because this is something that it allows. Before we dive deeper into what are incidental and secondary findings, let's clarify these very two important terms here and how they are a little different from each other. Incidental findings are something that are unexpected. You weren't looking for them, but WGS, for example, uncovered them anyway. Secondary findings are a special subset. So you intentionally look for them even though they are unrelated to the primary reason for testing. So the ACMG, or the American College of Medical Genetics and Genomics, for example, has created a list of genes where we must actively look for variants and report them as found. And here is a simple rule here that all secondary findings are incidental, but not all incidental findings may be a secondary finding. The key difference here is the intent and the actionability now of that finding. The ACMG's list, currently at version 3.3, which came out just last week in 2025, includes 84 genes across 52 different genetic conditions. These include genes like BRCA1, APC, RYR1, and TP53, which are commonly associated with things such as cancer risk, cardiac disorders, metabolic conditions that are inherited in nature, and many more other conditions. So what makes this list powerful is that every gene on it is backed by strong evidence and every associated condition is actionable, meaning that we can do something about this finding now. So even if WGS is ordered for something like developmental delay, we can still examine these 84 genes because the consequences of missing something important in these genes can be enormous. And the ACMG does not take this lightly, neither did they make this list very lightly. They created it because early intervention changes outcomes in the disorders included in this list. So, for example, if someone has a TP53 variant, they can begin cancer surveillance early. If someone has an RYR1 variant, they can avoid dangerous anesthetic drugs. If a child has a variation in GAA, they can get enzyme replacement therapy before their symptoms actually worsen. So, this list is the result of consensus coming in from medical, genetic, and ethical experts, all agreeing that variants in these list of genes must not be missed, no matter why the test was ordered, because these are conditions that can lead to a very crippling disease in patients in some cases, and in some cases it also increases the risk of sudden cardiac death, for example. And these are thus findings that should be reported to the patient so that they can manage the risks better. So there are four major criteria for a gene to be on the ACMG SF, or the secondary findings list. The first one being that there should be an established gene disease relationship, meaning that we need strong scientific evidence linking the gene to a condition. The disorder should be of high penetrance, meaning that most people having a pathogenic variation in the gene implicated in the disorder will go on to develop the condition. Then there is effective intervention that is already existing. So there must be something that you can do about it. So it is an incidental and actionable finding, meaning that screening can thus guide treatment and prevention if detected. And then the last one being that it should be of a significant health impact. So the disorder must be serious, often life threatening if not caught early. So, incidental actionable findings is not just about what to report and what not to report, it is about how to change lives by starting and screening for these disorders early. So, let's talk about the logistics of how these things may be reported. So, each gene, for example, has associated inheritance patterns. It may be autosomal dominant, recessive, or X linked. So for each gene, the ACMG specifies which variant should be reported. So these are typically those that have been classified by ACMG AMP guidelines as either pathogenic or likely pathogenic. So for example, if a person is heterozygous for a BRCA2 pathogenic variation, even if, for example, WGS was ordered for a cardiac condition, we can still report that BRCA2 pathogenic finding because it is of medical importance. The reason being, hereditary breast and ovarian cancer, like many other inherited cancers, is an autosomal dominant disorder. And harboring a BRCA2 mutation, a pathogenic BRCA2 mutation, in fact, increases your lifetime risk of developing breast cancer. Zygosity, inheritance, clinical classification, and associated conditions all clearly laid out for clinicians and patients so that they can be well informed about these incidental findings that have been detected in their genomes. So this example probably brings all of this information together. For example, we have a case in which a 37-year-old male had a whole genome sequencing done for chronic anemia, which is a very unrelated indication to what was detected in his genome. The report actually uncovered a pathogenic BRCA2 variant. So he was referred for cancer risk assessment. His mother and sister were both tested and found to be positive for this particular variation. His mother then went on to get enhanced surveillance done, and early breast cancer was detected and treated in this case. And while the sister is also positive, it now is a good starting point for her to keep a track of when she should be screened to manage her health risk and the risk of her developing breast cancer in the future. Now that these two things have really increased her risk of developing breast cancer a lot more than it was previously thought of, because not only does she have has a positive finding in the BRCA2 gene, meaning that she has a pathogenic BRCA2 variant, but also she has now a family history of cancer because it was detected in her mother. So this is a textbook definition of how a secondary finding can save lives. And this is why whole genome sequencing is more than just a diagnostic tool, it is a preventive health strategy. So one single WGS that was done at an appropriate time, I'll say, helped save not just one but eventually 3 lives here. And now here is another probably powerful example. A teenager undergoing whole genome sequencing for developmental delay was found to have a pathogenic variation in a gene called SCN5A. Now, although this wasn't related to the reason why the test was ordered, but what we uncovered might have life-saving potential, because the child, thus because of this pathogenic variation, has a genetic predisposition to developing long QT syndrome, a condition that can lead to sudden cardiac death if a particular trigger is achieved for that particular patient. So this trigger can be something as simple as running after a school bus or just playing with your friends. Any kind of physical trigger could have caused a sudden cardiac arrest in this particular. But thankfully, due to cardiology evaluation that could be done before any of the symptoms appeared, because we now know that this patient has this variation, lifestyle changes and modifications started, and thus, more importantly, we also enable detecting another sibling in the same family with the same risk. So WGS in this case did not just explain something, it prevented a tragedy in this family by ensuring that interventions began before a very severe thing like sudden cardiac death happened in any one of these affected individuals. The ACMG thus recommends that if we find pathogenic or likely pathogenic variations in this predefined list of 84 genes called the secondary findings list, we must report them. And this applies to all kind of germline sequencing that we do, not just whole genome sequencing, in fact, regardless of patient age, but the exception here being fetal samples. But there's an important caveat to this also, that patients should have the choice to opt out of receiving information about incidental findings, which is why pre-test counseling is so critical while prescribing a whole genome sequencing test. So genetic counseling is actually essential on both sides of testing. Before testing, we need genetic counseling to make it clear to the patients and their family that we may find health risks that are beyond the reason for which they were being tested. And that is why the opt-in and opt-out decision for secondary findings comes in, which is something that has been recommended by ACMG. After testing, counselors help families make sense of what is found, understand the next steps, and thus manage emotions. This human bridge between the lab and life is what makes genomic medicine truly effective, because just communicating results in a simple, standard way to patients is expected to increase a lot of anxious thoughts in them. So, why do we emphasize pre-test counseling so much? It is because there is an expectation setting that needs to be done before a person is prescribed a broad test like whole genome sequencing, to prevent things like shock and ensuring people know exactly what they are signing up for. So patients and the people who are getting enrolled for getting or are interested in getting sequenced must be informed of what to expect out of a report, what is something that is not possible to be generated in that particular report, what signs to look out for, what kind of information might be useful for them, and basically what should they expect out of a whole genome sequencing report. So when basically a patient owns the decision to learn about their incidental findings, they are more prepared and empowered to act upon them. So it is said that in genomics, the best surprise is the one that you are ready for. So genetic counseling and pre-test genetic counseling, in fact, is something that prepares people before their test report is released for dealing with these surprises. But of course, there are also challenges that come in after the whole genome sequencing results come out. So imagine the psychological impact of learning that you carry a very serious health risk. The anxiety and the guilt and the questions like, do I need to tell my family? How do I tell my kids? Should other people related to me get screened? Should my partner get screened? All of these questions start flooding in your brain. Then there is the logistics of this. Coordinating a cascade testing for a for different family members and managing lifelong surveillance in people who are still asymptomatic is something that gets added to it. All of these are not trivial burdens. These are something that people are generally concerned about, clinicians are generally concerned about, and which is why people do not prefer to report these findings to people sometimes also. But even though there are certain burdens to doing this kind of reporting, these are also manageable things that can be helped using the right support system. And this is exactly where post-test counseling steps in. So post-test counseling addresses emotional fallout and helps families plan the next steps after a positive incidental finding, for example. And if in case the action isn't immediately clear, specialists may be looped in to make a clear path to support these families. Classified also. So we need to educate patients about how how to interpret a variant of uncertain significance that might be a part of a whole genome sequencing report. We need to make them understand that right now you have a variant that we do not have enough evidence for to understand that whether it will cause a disease or not. But they need to be reassured that guidelines keep changing, new data keeps coming, more people keep getting sequenced, and thus their families or their own personal insights may gain much more clarity sometime in the future. So we also educate parents about reanalysis thus and staying in touch with their labs regarding if there is any update on their variant of uncertain significance, for example. So post-test support basically ensures that the story for these particular patients doesn't end with just a report, it continues with informed decisions. So, to wrap up, incidental and secondary findings have the power to change lives and even potentially save lives. They enable early intervention, empower families with crucial health information, and reduce the burden of preventable diseases. They also bring us closer to a model of truly preventive and proactive medicine, wherein we already know what to look out for in the future. And sometimes the most important answer is not the one you went asking for. So, this is where the power of reporting incidental and secondary finding lies. So, I will now stop here and happy to take any questions. Thank you. Okay, so we have some questions pouring in. If it is necessary to inform parents that they might get secondary or incidental findings in genetic test, and that to be conveyed to them only if they want to know. So yes, so this is where pre-test counseling plays a very important part. The genetic counselor must inform the patient's family that incidental findings are something that do get detected on a genetic test like whole genome sequencing. And going by ACMG guidelines, the patient or the patient's family, if in case they are the legal guardians for that patient getting tested, they should be made available an option to opt out of receiving incidental findings. So ACMG reiterates that giving the patient the autonomy or the option to get out of knowing that information should always be considered and given. How often do you have to check with the lab to understand the status of a variant of uncertain significance? So this is a very interesting question, and I'll say that the answer is not very simple for this because there is no set timeline as to when a variant of uncertain significance might gather more evidence and become either likely pathogenic or likely benign or something else. But the point here being that the more we sequence and the more we know that what types of variations are found in healthy individuals and what are the ones that are not and that are found in very rare conditions in our country specifically and the different populations we have, the more this information becomes clearer to us. So there is no set timeline as to when a VUS might get classified. Some databases are updated more often than other databases. For example, computational predictions tools are something that were created at some point of time and may not get regular updates on the growing evidence of things. But disease association databases like ClinVar are something that get at least a monthly update every year. So based on the database that is getting updated, based on the amount of sequencing that is happening anywhere in the world, all of these evidences might eventually change sometime in the future, although the timeline for this is not yet set. And yes, there is a need to develop a kind of a futuristic mechanism to understand how to report back a variant of uncertain significance if in case the classification has changed over the course of time. Okay, there is a question: is this testing associated directly with interventions? So, for the genes and the conditions that are included in the ACMG SF list, these are inherently conditions that do have either a treatment or a manageable thing available, or a management kind of a plan available for them. So, for example, for things such as in oncology, testing for a gene like BRCA1, BRCA2 creates an action plan, because now we know that you are at a significantly higher risk for developing cancer. So, you may be screened regularly using standard procedures. For things like for things like lysosomal storage disorders, for them for a subset of whom enzyme replacement therapy is available, if started early, it can prevent the patient from having more severe symptoms. So these are things for which a curability, not a curability, a treatability indication is actually there, and thus can lead to management of these patients better. In fact, for the treatability and the actionability of perspective of this entire genetic WGS testing is something that I will also discuss in my next session. Okay, so how important is WGS in the context of detection of diseases that cannot be treated? So we might think that if something cannot be treated, it should probably not be reported. But if we consider the emotions and sentiments that patients and parents and families of rare disease patients hold, they are actually practically roaming from pillar to pillar in search of just an answer as to why they have what they have. This is very profound in rare diseases, that because you really don't know what is wrong with you, and in some cases, in some communities that are not very well off and are probably on the lesser level of education side, they also might end up thinking that there is a curse of God that has been placed on them. In fact, there is a very interesting example of a family that was actually detected in India itself, of having in the Nalband community of North India. So this family was suffering from a disease called MLC, and the father of the father, who had 8 children, out of which 5 were already affected, had actually petitioned the president of India in 2014 to euthanize their children, because he could not bear to look at how debilitating and how crippled their children were becoming and were not able to support their health. So imagine the plight of the father, who did not know what was going wrong in their family and still wanted an answer. Else he thought that there was an easier way out of this pain for his particular, or for these affected children of his. So for these families and these patients, even getting an answer as to what is the diagnosis for the condition, what do they actually have, it becomes very important, because it then leads to another stream of things that can be done, which includes counseling to tell the family of the risk of inheritability of this condition, so that if possible, and if they do want to have a healthy child in the future, they can consider other options also. Should incidental findings with low coverage be reported? So as a QC kind of a thing, incidental findings with low coverage should not be reported if they have not been validated additionally. Also, variants of uncertain significance related to incidental findings are also something that are not yet recommended to be reported. If a case is detected as a variant of uncertain significance and subsequently dies of the disease, is this death reported to any place for considering this condition and add to pathogenic status of the disease? Considering that if that particular gene in which the variant was detected is something that is very directly linked to the phenotype that the disease causes, then this is actually an ACMG attribute that can be assigned for that particular variant. But only given that the symptoms that the disease that are associated with the disorder must not overlap with some other disorder or other genes, meaning that the syndrome or the disorder should be very well defined. So, for example, if a patient has cystic fibrosis, which has very classical symptoms linked to just one disease, CFTR, this particular thing can be considered as a strong evidence that a patient has phenotypes which are something that are very characteristic of the disorder. But in cases like cardiac conditions, so cardiac conditions are something that might be multisystemic in its cause and effect, and thus can be linked to not just your germline DNA but also mitochondrial disorders. So, if a patient, for example, dies of sudden cardiac death, it is not something that is linked to one gene. It is something that might be linked to a plethora of genes. So, for this, the evidence is still lacking as to if that VUS is something that caused this particular phenotype in the patient or not. So, in the interest of time, I think I'll stop taking questions now and we will try to answer them on chat itself subsequently while we move on to the next session. Hi, Hari. Sorry, you're muted.
Thanks for letting me know, Prabhani. Thank you. The next session again is on genomic newborn screening and public health applications. One of the key missions of our Karkinos team message is that WGS for all. That is something that you might have seen as a tagline, as a hashtag, and a lot of other things. So while we say WGS for all, for most of us, people who are above the age of 1, we can do, of course, in a retrospective, we can do a whole genome sequencing. But the prospective and the future of the country is the children that are being born now. So newborn screening is a very, very critical and an important topic. Uh, Again, so I invite for this session as well, I invite Dr. Bani to share her views, comments, and inputs.
Yes, thank you, Harish. And let's now segue into the next topic here, which is of very important, which is of high importance and relevance in today's scenario now. So far we have talked about the importance of whole genome sequencing, how it can influence in your healthcare decisions and so on. But when exactly should this testing start? This is a question that most of you have asked on chat also, that what is the right time to get tested? And probably this session is something that can answer our doubts regarding the timeline of testing, which might be applicable today also, but will definitely be applicable for the generations to come. So this session is about a topic that is rapidly redefining the future of preventive health, which is genomic newborn screening. So in this session, we'll now explore how whole genome sequencing, particularly when applied at birth, can go far beyond diagnosing disease, and it can thus anticipate risks, enable early intervention, and in many cases completely alter the life trajectory of an individual just because of one reason, that we started early. Right, sorry. So let's consider an example here. So a baby is born. The baby is healthy looking, full of promise, and the routine newborn screening is done for this particular baby. Everything seems fine and the parents are reassured. But within a few weeks, there are subtle signs, including feeding issues, constant crying, and poor weight gain that the baby is now displaying. Although all of this is then brushed off as acid reflux, but at 3 months, the baby is still not reaching certain developmental milestones. For example, the baby has not started rolling over yet. At 5 months, the parents notice that the baby does not smile much, but everyone still reassures them, saying that, oh, she will catch up. All kids develop very differently. So, but still by 6 months, the concerns actually start to grow. The child is now having staring spells, delayed responses, and thus tests begin. The uncertainty here begins, and slowly then we start losing time here. By 9 months, the seizures have now begun in this baby. It is no longer subtle. The signs are much more urgent now. And the family has now entered a maze of neurologists, metabolic tests, and endless scans. But still, they have not yet gotten a clear answer as to what is happening. At 18 months or probably later, somebody maybe suspects a genetic disorder. At 2 years, maybe they finally get a diagnosis. Or maybe they never do. But by then, the critical windows for brain development for this child have already closed. So, interventions that could have been started earlier actually began late, because the answer that could have come earlier did not probably come at all. And the family has already spent 2 years minimum in confusion, distress, and quiet heartbreak because of all of this. So, which brings me to a rhetoric question, that if you could go back in time to change a moment in a baby or a person's health journey, would you? So, let's, for example, now go back in time to the point where this baby was born and change just one variable here. This is the same baby, same birth conditions, same routine checks. But this time, the parents were offered if they want to get a genomic screening done for their child, and they said yes. And by week 2, the results came in and they showed a mutation in a gene called CDKL5. This gene is linked to a rare neurological condition that causes early onset seizures and severe developmental delays. It is serious, it is a very serious condition. But now the family has a diagnosis before the symptoms have begun. So now, for instance, by the 3rd week of life, this baby is already under the care of a pediatric neurologist who knows exactly what to look out for. By the 4th week, a treatment plan has been put in place before the occurrence of the first seizure even is seen. By the 5th week, targeted medication begins for this particular condition, and the family is connected to support groups and experimental therapy options. At 1 year, the child still survives but lives with this condition, but now there is a care plan for the child. There is a guidance and there is no long delays that the family has to go through. The condition hasn't changed, but the outcome has changed, because the timing of doing all of this changed everything in this case. So, in this case, you have just seen timeline 1. The symptoms started creeping in, the missed windows, and the diagnosis that comes too late or probably never at all. But now imagine if the clock had actually stopped and turned back all the way to day 0. The same baby, the same genes, and the same condition is still there. The same condition is still there, but we have added just one thing in this entire process now. We added genomic newborn screening. And suddenly the story unfolded very differently. And actually, this is not just a fictional thing. It is a real, very real story for a very real family. So this very cute baby shown in this photo here is baby Margot. She was born in August 2023, and her parents said yes to enrolling her in a study called the Guardian Study, which is a research program offering genomic newborn screening for people in the USA. Through that, she was diagnosed with a mutation in the CDKL5 gene linked to this severe neurological disorder. But this time, the diagnosis came before the damage. Her care plan started before her seizures did. And this is exactly what it looks like when we act in time. So baby Margot is now 1.5 years old. She is curious, she is chatty, she is growing well. Because the condition still did not vanish, but her timeline changed. And just because her story did not wait for a diagnosis, it actually began with one. So I'll ask this rhetoric question now, that what if the future did not need fixing because we got it right from the very beginning? So we have seen that tiny changes in one gene, a single letter of our DNA getting altered, might lead to serious diseases. This might be a very rare occurrence, but these aren't just one-off cases. There are thousands of rare disorders, each that are caused by different mutations in different genes. And globally speaking, rare genetic diseases affect nearly 400 million people in total. If all of these people were to live in one country, it would be the third most populous nation in the world, right after India and China. In India alone, 70 million people are estimated to be living with a rare disease. That's more than the entire population of the United Kingdom. So we should ask this question: are rare diseases even actually rare, or are they just understudied, underdiagnosed, and underprioritized? Sadly for the patient, the number of particular condition is prevalent. The average journey to a diagnosis for a rare disease patient involves at least 8 physician visits, 3 misdiagnoses, and over 7 years just to get a name. And behind those numbers, those 70 million people living in India are actually families. So this is not just statistics. Families live through years of confusion, pain, and irreversible decline. And let me take you through the story of one such family, which I also gave as an example in the question I answered in the previous session. So this is a family from the Nalband community of North India. In 2014, the father wrote to President of India asking for permission to end the lives of 6 of his 8 children. They had all been born normal, healthy, and normal development occurred until the age of 3. But by the age of 4, everything began to change in this family for all of these kids. Seizures started, progressive motor impairment started, loss of mobility, and enlarged heads. The doctors were confused because the condition right now did not have a name, no diagnosis, and thus imagine the plight of his father, who was willing to go to such extreme, the extremist level of lengths you can to get his kids out of this misery. Eventually, genomics efforts that were led by Dr. Vinod and Dr. Sridhar approximately 10 years ago from today helped point out the cause of this particular condition in this family. It was, in fact, a homozygous frameshift mutation in a gene called MLC1, now known as the Nalband mutation, which leads to a severe degenerative brain disorder called MLC. So this condition was genetic, it was inherited, and it was completely unknown for this family at that time. So the tragedy here isn't just the disease. MLC might not be something that is treatable. The tragedy here is that the family begged for answers for years, and by the time the science probably caught up, the damage was irreversible. And this is exactly why we must start early or at birth. So today, most newborns undergo some level of traditional screening tests like the heel prick blood test for metabolic conditions, and ECG for determining heart defects, hearing screening, and imaging when required. Standard newborn screening, I'll say, has done wonders till now. It catches dozens of metabolic and hormonal conditions, but it is very limited in its scope. It looks for around just a max of 40 conditions, mostly through biochemical changes. But more importantly, these are able to catch signs of a disease that probably has already started to show. Genomic newborn screening takes us a step further here. By sequencing a baby's DNA and using NGS, we can look for genetic changes even before the symptoms begin. So we can thus identify risk, confirm diagnosis, offer personalized care, and even uncover carrier status for inherited conditions. But then what about all the other serious treatable conditions that show up genetically but aren't a part of routine screening by general biochemical tests? So this is exactly what the Guardian study set out to explore some time ago. They enrolled 4,000 newborns and screened for not just 40 but over 400 conditions using whole genome sequencing. And the results of this study were actually very striking. WGS found 120 babies, which is 3% of this total cohort, with serious actionable conditions. Of these 120, only 10 were picked up by standard screening. That is 12 times more. The positivity here is 12 times more on WGS compared to your routine screening. And remember the baby from the previous slides, Margot, she was also one of these 120 that were detected here. Her diagnosis would have never been caught through routine tests, but thanks to Guardian and the genomic screening that she went through, she got a diagnosis in week 2 of her life itself and treatment by week 5. So let's bring all of this back home to India. Every year, over 1.7 million babies in India are born with birth defects. That is more than the population of even some small cities here. So, in essence, we are adding children with a large amount of children with birth defects to our population. The tragedy here being that many of these conditions are genetic and many are treatable if they have been diagnosed early. By conventional screening methods, this will often basically miss these conditions because they wait for symptoms or probably only focus on a very short list of common disorders. And here is the scale that we are dealing with in India specifically. In most countries, 1 in 1,000, for example, might mean a handful of babies in total. But in India, where the population is large, 1 in 1,000 is actually a million. So even the rarest of rare disorders add up really fast. So genomic newborn screening gives us a chance to find all of these conditions before symptoms begin and intervene when it still matters. And in fact, genomic newborn screening, although it looks like a very futuristic kind of a concept, it is not a distant idea. It is something that is happening, has been happening, and growing all around the world. The first pilots began over a decade ago with projects like BabySeq, NBSeq in the US. Since then, we have seen NC Nexus, Baby Beyond, and now large-scale studies like Guardian and Begin NGS occurring elsewhere in the world. In the UK, Genomics England launched a national level program just last year. And across continents of Europe and Australia, programs like Baby Detect and Baby Screen Plus are already starting to enroll families. And also by 2025, the UK's NHS is expected to formally integrate whole genome sequencing into its national system, especially for newborns. So these global programs are mostly testing for conditions that appear early in life and are often treatable. The majority, or over 80% rather, are inherited metabolic disorders, but these conditions being tested for may also include immune, cardiac, and neurodevelopmental conditions. And the genes being tested differ in different countries. For instance, in the United Kingdom, they're testing for mutations in around 500 genes that are linked to over 200 rare conditions. The key point here being that these are conditions which appear early and can be treated through the NHS system itself. And these conditions thus have better outcomes if caught early in time. So just coming to the treatable the concept of what is treatable and untreatable here, I'd like to start with that just because a disorder is rare does not mean that it is untreatable. To decide what to screen for in such newborn screening programs, we look at 4 very important things. Is the disorder prevalent enough in the population? Is it penetrant, meaning that is the variant likely to cause symptoms in that particular patient if found? And is it actionable? Can we actually do something about it? And the sweet spot of the things that we need to test is where all these 4 things overlap, which is what makes a condition truly screenable. But treatable here does not mean curable. It actually means that we can act early to improve outcomes and manage this particular conditions. So, for example, some disorders like galactosemia are managed with small changes like dietary changes. Wilson's disease may need a liver transplant for better management. Cystic fibrosis can be managed with certain medications that have come up and supportive care. Things like severe combined immunodeficiency and thalassemia can be managed with stem cell transplants. Many metabolic and mitochondrial disorders often improve with supplements, and for a very few handful disorders like spinal muscular atrophy, we now also have gene therapy. So all these therapies or these actionable items that we have here are not something that are hypothetical. These are real proven interventions that we can deploy if we catch the condition in time. So now different countries are screening very different genes and disorders. But then when we think about India, what is prevalent, actionable, and treatable in India? Or do we just follow what other countries do? Or else, how do we get to a list of conditions that might be relevant to our own population? So here we decided to ask the experts across India itself. And that is exactly why we started the Genomic Evidence for Newborn Screening in India, or Genesis initiative. So Genesis is a national level effort in India to generate India-specific genomic evidence for newborn screening so that every baby, no matter where they are born, can have access to a genome-informed diagnosis from the very start. Genomic newborn screening is expanding globally, but India needs its own data to have it implemented in its own country. But we lack population-specific evidence on gene prevalence and disease patterns. And then there is also a need for screening models that will work across the very diverse settings that we have in our country. Early detection can change outcomes, but only if the access to all of this is equitable. And to scale this responsibly, we need strong scientific backing to guide policies around it. And this is exactly what Genesis is designed to deliver. So how we have implemented this framework to go about is we invited experts, which includes pediatricians, neonatologists, geneticists, counselors, and so on, to join us, and they did. We begin with a systematic gene curation tailored specifically for India. Each gene is mapped for clinical actionability and Indian relevance. Experts across these various disciplines give their inputs on the genes list to build consensus of what they deem as prevalent and treatable in India based on their clinical experience. And the final list and evidence base will thus be made public for national reference and equity. The entire framework is actually built to evolve, with new data coming in, new voices being heard, and a real world feedback mechanism for all of it. Now, many of us may have come across this very recent news that the UK's NHS has announced plans for all babies to undergo whole genome sequencing after birth. But why whole genome sequencing specifically? Because we have already seen in our previous sessions that WGS is something that changes the game here, because it covers almost the entire genome, including genes, introns, regulatory regions, everything. So with one test, you can now detect chromosomal abnormalities, large structural changes, repeat expansions, deletions, insertions, point mutations, and so on. And even mitochondrial DNA variations, which panels and standard WGS usually miss. So WGS is not just more comprehensive, it is a single unified test that gives you more answers, and that too faster. That's why it's becoming the global standard now for newborn screening. So WGS is not just a broad approach, it is also a powerful approach. Over the last 2 decades, costs of doing a whole genome sequencing have plummeted, while the number of clinically relevant variants that are being identified has exploded. So what was once a 100 million effort can now be done for a fraction of that cost, making WGS a very realistic clinical tool. And it is thus anticipated that very soon WGS will replace whole exome sequencing and panels in clinical settings. And this is why we must be prepared for that change to happen. And the results actually speak for themselves. An analysis of a very diverse heterogeneous cohort of patients from USA shows that WGS gives an additional 8% diagnostic yield for conditions over WES. And this is exactly why very recently the American Academy of Pediatrics recently recommended WGS as a first tier test for children with global developmental delay or intellectual disability. Now, when a baby is born in India, we often gift them gold bangles, silver anklets, and maybe even a fixed deposit in some cases. But imagine gifting something far more important, which is a lifelong health map for your child. Because your germline DNA, the one that you're born with, does not change with time. So one test done at birth can guide health decisions for a lifetime. So WGS isn't just a newborn test; it is actually a lifelong resource from managing or diagnosing, looking for treatable rare conditions at birth, to supporting carrier screening, reproductive decisions, and pharmacogenomic choices in adulthood. WGS data will continue to deliver value throughout every stage of your life. Now, today, genomics is becoming very indispensable in the case of NICUs because of evolving perspectives about neonatal disorders. The old assumption was that most newborn complications, especially in premature babies, were caused purely by external factors like immaturity or injury. But things did not add up because even when the babies had same diagnosis, their outcomes were very different. So, same treatment also led to a very variable response. So, all of this thus led to a new insight that we now know that a baby's genetic makeup can dramatically influence how they respond to certain exposures and treatment. And this is not a rare occurrence. 14% of NICU babies have an underlying genetic condition, and up to one third of neonatal deaths are linked to these genetic disorders. In fact, we are now realizing that many neonatal conditions that were once attributed purely to external factors actually have a strong genetic basis to it. And now understanding this helps us move from a reactive to a more targeted and preventive care approach for the most vulnerable kinds of patients that we have. Now, most of these conditions, especially genetic and metabolic ones, can mimic very common issues like seizures or encephalopathy. But what's often missed is that they may be the real underlying cause of that particular condition. So these disorders can affect multiple systems, for example, leading to diagnostic delays. And the stakes here are very high because they are linked. These things are linked to both neonatal mortality and long-time morbidity in these patients. Yet, many of these are treatable if caught early enough. Which brings us to the best question, that are current WGS reporting turnaround times fast enough to provide a diagnosis to actually change outcomes? So, when we think about neonatal intensive care, we often imagine a race against time because there are tiny lives in a very fragile balance where every decision matters now. And in this high stakes environment, genomics has emerged as one of the most powerful tools we have. What makes all of this possible is speed. With a rapid whole genome sequencing, for example, we now have the ability to move from sample to report within a short span of even, for example, 48 hours. And all of this starts with a simple heel prick or saliva sample or a blood draw from a newborn, followed by DNA isolation and library preparation. The next generation sequencing generates data, and here's where the heavy lifting happens. The analysis part of it that has become now so sophisticated and so fast is something that has really reduced turnaround times for whole genome sequencing tests. Finally, trained genomics teams who manually review and interpret these variants are then lead to something that can be offered through a clinical report that will land on the neonatologist's desk in time to start making real decisions for that particular patient. So, in essence, all of this is not just about fast sequencing, it is about making these insights that are uncovered by whole genome sequencing available fast to inform action. So, as we look to the future of genomic newborn screening, especially in India, it is important to acknowledge some challenges that still lie ahead. So firstly, for example, we often encounter variants of uncertain significance or VUS, and these are basically changes where evidence is not strong enough to say whether the variant will cause the disease or not. That creates uncertainty both for clinicians and for families. Secondly, ethical dilemmas also exist. What to do when the test finds something untreatable or something that only matters much later in life? Should that be reported in a newborn? And all the questions that surround this kind of a thought. And finally, there is the question of equitable access to all of this. And lastly, to make sure that this reaches beyond just big cities into the community and every healthcare system, we do need a trained workforce that includes pediatricians, counselors, and public health support ready to act on these insights. So we have heard about this term now, variant of uncertain significance, a lot, and why they matter a lot in a whole genome sequencing test is that for a WGS for a broad test like WGS, there will be a lot of variants of uncertain significance that will be detected. In fact, in total, about approximately 20% of any kind of next generation sequencing test that is done, they identify only variants of uncertain significance. The problem here is not the variant, it is the lack of evidence. That maybe that not enough people with that same variant have been studied before. Or maybe we do not yet know what the gene particularly does, or if the variant is rare and specific to a particular population or not. So in such cases, we can't determine whether it is harmful or harmless, and then that becomes a very big challenge in interpretation and communication. So how do we deal with this uncertainty? There is actually a process to it. We go about by gathering more evidence for all of this and check if we actually looked at all of the ACMG AMP attributes or not. We can consider functional and familial studies for that particular case, depending on how equitable access can be provided to that family. It is also important to reassess over time because evidence changes and guidelines also change. And very importantly, provide genetic counseling because this isn't just about science, it is all about also supporting families. Counseling then thus helps people understand the meaning of the result, the limits of our current knowledge, and what next steps can be taken in this case. So the big picture here in conclusion is that genome is lifelong and reading it early unlocks lifelong insights also. So WGS lets us spot rare but actionable diseases at birth, sometimes even before any symptom actually shows up. That opens the door to early intervention, better outcomes, and a paradigm shift from reactive to predictive care. And this is not just about technology, it is about building systems that can act on these insights because the science is ready, now the healthcare system needs to be. And in the end, the real power of genomics is not just in prediction, it is actually in prevention. Provided that we inform the families of the risks of all of it first, and this is where genetic counseling comes into play, and which is something that will be discussed in more detail in the next session by Dr. Jyoti. So in conclusion, we don't need a time machine to go and fix somebody's healthcare journey. We just need to act in time. So thank you. I'll stop here and I'll now take questions. Okay, so in cases where clinical information is unavailable, what is the recommended approach for reporting pathogenic or likely pathogenic variants in autosomal dominant genes? So clinical information is very crucial actually to report back what might be of use to the patient or not. All of this depends on the kind of pathogen or the gene in which the particular variation has been detected in. So for example, if it is detected in a gene that will lead to some kind of disorder later on in life, meaning the patient, depending on their age, might be unaffected till now, it might become a little more important to report, because even though right now it is not actionable, in future it might lead to a disease. But all of this needs to be tied to a clinical context. There should, for patients, be a clinical information available through either a clinician or the treating doctor. And for healthy individuals, there is where genetic counseling and capturing the family history and the clinical history of that particular individual undergoing whole genome sequencing becomes very, very important. Additionally, can post-test genetic counseling aid in interpreting such findings and guiding subsequent clinical decisions? Yes, definitely. Post-test counseling is something that is very important, because it actually leads to kind of a hand-holding scenario for people, so that they can understand what the tests that have, the results of the tests that they have undergone mean. So leaving them alone might cause a lot of anxiety, unsurety, and due to all the lack of direction that and the fears that may arise from it. Post-test counseling is something that will take you through all of this and make sure that you understand now what to do. And Dr. Jyoti will definitely discuss in more details about how these two pre-test and post-test counseling can help in these cases. Do we need to do Sanger sequencing to confirm before reporting a secondary finding? No, definitely not. Next generation sequencing is something that has attained, if not higher levels of accuracy compared to Sanger sequencing now. And thus, we really don't need an orthogonal method to validate whether this secondary finding is whether this secondary finding is there or not. Because in essence, we are then using two sequencing techniques to just confirm the same thing that has already been very confidently reported through this high accuracy test. Yes, but not Sanger sequencing. We might require confirming that particular condition using some other interventions that are clinical in nature and not really sequencing in nature. So, for example, if we detect an incidental or a secondary finding linked to sudden cardiac death in a gene, for example, SCN1, so the patient can undergo now a heart screen to understand whether they are, or an ECG basically, to understand if they really do have long QT syndrome or not, because this is something that can be picked up through a regular ECG also. So this is the kind of confirmatory action that can be taken, but not really undergoing an orthogonal sequencing validation. Findings that relate to neurodevelopment conditions like Parkinson's and schizophrenia, for example, even though they might have are very environmental in nature also. These are complex disorders, multifactorial things come into play about how they are caused and how the disease progression might happen. So these are not something that can have an answer through just one single test in this case. Given the increased diagnostic utility of both WGS and WES, how should clinicians decide which approach to prioritize in undiagnosed cases? Should WGS be considered as a first-line test or only after WES? So as the cost of whole genome sequencing is now coming down, so I will recommend that people in this forum do not be concerned about the cost of whole genome sequencing anymore, because currently WGS costs exactly the same as what a WES costed 5 years ago. And this decline is something that is not even following a logarithmic curve anymore. It is decreasing as the supply and the evidence of its actionability and its importance is increasing. So given that cost is not a factor here, whole genome sequencing is actually something that, as I showed in my slides also, diagnostically much more sensitive compared to whole exome sequencing in detecting reasons for undiagnosed cases like certain rare conditions. So for example, that cohort in USA detected that it had an additional diagnostic utility of 8% over whole exome sequencing. So removing cost as a factor here, it does not make much sense that a patient goes WES and then if it is negative, then go for a WGS when they can directly be referred to a WGS test in the first place. Can our country afford the cost of WGS if it is to be implemented across all district levels and sub-district hospitals? And then what about training of all health staff? Yes, so I'll say that currently we are not ready, but we need to start now to build the basis of what needs to be done in the future, which includes conducting these things at a different level before finally letting it trickle down to the district and sub-district level hospitals. Which is why, for example, for genomic newborn screening, we have started Genesis initiative to drive policy making across India regarding this very important topic. And thus, we also, in extension, want to now start the training of all healthcare staff and medical professionals in general, which is something which is also the objective of this entire course, actually. So all of these things, pun intended, start with baby steps, and if we don't start, it does not lead anywhere. So yes, there is still a long way to walk to our end goal, but we will eventually reach there by starting training and all this evidence building right now. What are the ethical implications of reporting adult onset conditions like Huntington's disease while doing a whole genome sequencing in newborns? Should such findings be disclosed to the family or deferred until the child is older? This is a very important question and exactly why the evidence building for genomic newborn screening has started now, that we need to have set policies regarding what is something that should be reported, not be reported, and what exactly should the family be counseled against before opting in for a genomic screening test for their newborn. So several things come into play here, which is firstly developing a policy around how to go about this, and then obviously enabling genetic counseling of families to understand what is something that should be reportable, because in one child it might not be relevant, but in a case where there is a very strong family history, it might be very, very relevant. How many inborn errors of immunity were picked up in the Guardian study? So in the Guardian study, the most commonly detected condition was actually inborn errors of metabolism. And the second probable highest number of cases that they were found were actually related to immunodeficiencies. In fact, the Guardian study did end up detecting out of these 120 positives that they had detected that were not picked up by regular biochemical screening. One of them was a case of a severe combined immunodeficiency for which stem cell transplant actually helped get that child the early intervention that their condition needed, enabling them to have now a much more normal life as compared to what would have happened if in case that stem cell transplant was not done in that particular newborn. So I think I'll stop here in the interest of time, and I'll pass over the session to our next speaker, Dr. Jyoti. So thank you, everyone. I'll take off now.
Thank you all. Thank you, Dr. Bani. I'm here to glad to introduce our next speaker, Dr. Jyoti Bajpai Dikshit. Dr. Jyoti is a senior genetic counselor at Karkinos Healthcare. She is a level 2 certified genetic counselor from the Board of Genetic Counseling India. She has more than 20 years of experience in the fields of genetics, genomics, bioinformatics, and systems biology. Dr. Jyoti is also an associate member of the Indian Academy of Medical Genetics. Dr. Jyoti has a PhD from IIT Bombay and a certificate in Executive General Management program from IIM Bangalore. And she is an MTech in biotechnology from IIT Lucknow and is a gold medalist in MSc Physics from Lucknow University. She has also done a certificate program in marketing analytics from IIM Rohtak. Her multidisciplinary experience is from many industry named Strand Life Sciences, Core Diagnostics, GenePath, OncoStem, Mahajan, and Medflux, Carer, and Cancel, and Reliance Industries Limited. Dr. Jyoti's goal is to serve humanity by bridging the gap between laboratory research and patient care, ensuring that the knowledge and discoveries made in the lab are effectively applied by clinicians to treat patients and enhance their quality of life. Dr. Jyoti, welcoming you to the screen, and I'm pinning you to the stage.
Sure. Thank you, Harish, for such a nice and kind introduction. And just want to check first if my slides are moving properly. Let me just try it once. Yeah. So are the slides moving properly? Can you see that? Slide 1 to slide 2, slide 2 to slide 3. Is this okay? Harish, can you see my slides?
Yes, Dr. Jyoti, it's visible.
Okay, fine. Thank you. So first of all, namaskar to all my esteemed and dear friends and people attending this session. And I am really honored to have this opportunity to speak on an important topic of effective communication from genomic insights, which is basically the genetic counseling. Which is pre-test as well as post-test. All of us know more or less the definitions of the pre-test counseling and post-test counseling. Let me begin with saying this, that Dr. Mary Claire King, who discovered that breast cancer risk can run in families through the genes like BRCA1, she had said that genetic counselors simplify complex genetics for everyday people by offering empathy and guidance for informed decisions and emotional support in a non-directive fashion. This statement clearly indicates that an effective communication is essential for successful genetic counseling. In the coming slides, we will explore the key components necessary for effective communication in genetic counseling. So the agenda, or you can say that I have organized my talk into 4 main headings as given on the slide. I will discuss each of these in coming slides. So to start with, effective communication. What is it? Effective communication in genetic counseling involves key elements such as understanding the purpose of counseling, utilizing strong communication skills, and managing individuals' perceptions and their emotions to ensure a personalized approach in order to make informed decisions. So, what is the purpose? As I said, the purpose of genetic counseling. The purpose of genetic counseling is to simplify the complex genetic information to help patients and their families and other individuals in making informed decisions. For instance, let us consider a patient concerned about their family history of cancer. The counselor begins by building trust, providing relevant information about germline cancers, their inheritance patterns, and then they also take patients' family history, answer any questions which patients or the families want to know, and then they explain genetic risks in clear and simple terms, very necessary, simple words, so that these patients or the families or the individuals are empowered to make informed decisions about the testing and potential preventive measures. I hope I am clear in my discussion, means the voice and slides, everything is clear. Is that okay?
Yes, all good. Please go on.
Thank you, Dr. Bani. Thank you, Dr. Bani. Now, coming to next slide, what are the key points for an effective communication? The very first thing is counselors must explain genetic concepts in simple words. For example, while discussing cystic fibrosis, a counselor might say, cystic fibrosis is a genetic condition that affects lungs and digestive system. Secondly, the counselors listen to the patient's concerns with empathy. Half the problem is solved just by listening. We have to be active listeners and with empathy. So the thing is, if a parent is concerned about their child, just an example, that if a parent is concerned about their child's cystic fibrosis diagnosis, what we can suggest, what the counselors might say, I understand how difficult this must be for you. Let us talk about how we can manage this together. So we have to become them. We have to become one team. Counselors provide decision support by explaining options and guiding the patients and their families. In the same example of cystic fibrosis, they might discuss the genetic testing, the treatment options, the lifestyle changes to help patients' parents so that they can make an informed and optimal decision. Finally, counselors should encourage, and they encourage the patients and their families to ask questions. It should not be a monologue, it should be a dialogue. And then they provide clear and thoughtful answers. For example, explaining the same case of cystic fibrosis, people would like to know what is the inheritance pattern of cystic fibrosis. And the counselors help the patients help the patients and their families to understand the chances of passing it to the next generation in very simple language with empathy. Now, coming to the next slide, there are multiple types of perceptions, and everyone has their own perceptions. As all of you must have seen the movie, or maybe you will be planning to see the movie Sitare Zameen Par. They say that everyone has their own normal. So now, individuals' perceptions about their normal. This is a key aspect. We have to manage individuals' perceptions in genetic counseling. Health literacy helps in creating awareness and reducing ignorance. For example, while discussing a mutation in Notch3 gene, which is linked to brain stroke, the counselor should simplify the information so that this doesn't become a scary thing for the person being counseled. Next, an individual's cognitive abilities or the thought process also influence acceptance versus denial. Some patients may resist the idea of inheriting the mutation, while others may accept it. Emotional strength plays a role in responses to risk, reducing into hope for some people and anxiety for some other people. Counselors must address the perceived outcomes by balancing the benefits and the risk. For instance, in the same case, genetic testing for NOSH3 mutation may offer preventive options but also reveal potential risks. The counselors should manage these perceptions of individuals to ensure the informed and the right decision making. The next important aspect is personalization. This is very important. Why? Because as we said, each one has their own normal and each one has their own perception and each one has their own thought process. So personalizing genetic counseling is a crucial and helpful aspect of genetic counseling. For example, in the case of male breast cancer, which is often met with denial due to societal stereotypes, it is important to tailor the conversation to the individual's understanding, acknowledging that many men may find it hard to accept the idea of breast cancer due to its association generally with women. We need to address the emotional and psychological needs by acknowledging and validating the patient's feelings of disbelief or fear. By focusing on personal health and family impacts, we should support the decision making, guide them to follow the actionable steps such as genetic testing, treatment options, medical surveillance, and understanding the risk for their family members. Next, another important, or maybe we can say the most important aspect of genetic counseling is ethics. And we had a question on that in the previous session. So ethics is an unavoidable and essential aspect of genetic counseling to guide for decisions to be taken with fairness, respect, and protection. Key ethical principles include beneficence, which means doing good by providing the best care to the individuals. Non-maleficence, which means avoiding harm and ensuring that actions do not cause any problems. These are five principles which are very important. The third one is informed consent in a non-directive manner, which means providing necessary and relevant information so that the individuals can decide whether to undergo a medical test or a genetic test or not. And we should not force the decision, our thinking, our thought process on anyone. Fourth, autonomy, which means respecting person's right to make their own choices without pressure, non-directive manner. Then confidentiality, which is like very important. Why? Because this is someone's private and personal information. And we should keep it private and it should be shared only after permission and with the people to whom they are allowed to permit to share the information. Last but not least, justice. Justice is very important, and this means that we should ensure fair treatment and equal access of care to everyone. It should not be affected by gender, race, financial say status, or anything else for that matter. So these 4 or 5 principles, these are very important for maintaining the ethics of genetic counseling. We will discuss a bit more about ethical considerations. Like, what are the ethical considerations? And these are like, say, they can address several types of concerns. Like we keep asking whether we should disclose it to the people or not. So let us discuss what are these considerations are. The impact of genetic information can affect individuals emotionally as well as socially. The first ethical consideration is about disclosure of information. This focuses on decisions about what genetic data should be shared and with whom. Also, the future use of genetic material raises the issues of consent for research and other purposes. And that is why it is very important to have an informed consent from the person to be tested or from the person who is planning to be tested. Next, as I have been saying, non-directive counseling ensures that individuals can make decisions without external pressure. Genetic discrimination is a concern as genetic data should not be used or misused as in cases of employment and insurance, etc. And that is why we should come up with good guidelines, as Dr. Bani was also mentioning, policies and guidelines, so that people get the good outcomes of the test for the best of them. Finally, eugenics concerns highlight these concerns. They highlight the ethical issues regarding human rights and diversity to ensure that genetic results are not used to promote certain traits or discriminate against certain other traits. So that is very important. So here, whenever we are doing counseling, especially in the case of whole genome sequencing, which is full of incidental findings, actionable findings, carrier status, pharmacogenomics, and information about the predisposition of hereditary cancers and certain other disorders, it is very important to take care of these ethics concerns. So in short, if I say, what is the best practice for ethical considerations? These are the points given here, but if I want to say in a nutshell, I will say that ethical considerations focus on respecting individuals' rights and prioritizing their well-being. The goal is to ensure informed consent, maintain confidentiality, and offer fair, unbiased services. We should be careful about the emotional impact on individuals, protection against genetic discrimination, and ensure that the use of genetic material is therapeutic and not harmful. And of course, we should avoid eugenics. Now, coming to the next important aspect is multicultural issues. Multicultural. As all of us know that Bharat is a country of multicultures, multiple languages. As you can see on this slide, Dr. Winkelmann, what he said on that basis, this is a statement, what it says that the next most important need or the most important need along with scientific and technical discussion is for a genetic counselor is being competent in handling a multicultural population, which becomes vital in a country like us, like Bharat, where diverse cultural beliefs, values, and practices can impact patients for in understanding and also in decision making. It's very important to understand each and every culture. Then the question arises, what are these cultural aspects which have to be taken care? For cultural competence, the key points to be considered are cultural responsiveness, which means adapting counseling approaches to meet patients' unique cultural needs. Second, cultural awareness, which means ensuring an understanding of different beliefs and respecting them, such as importance of family in decision making. Sometimes families get involved in decision making. Cultural sensitivity is the third one, which is helpful to avoid misunderstandings by respecting values. We should value others' values. We should not be judgmental about others' cultural beliefs. Then, cultural skills, which enable counselors to communicate effectively. If we know about different cultures, it helps in talking to different people effectively. Hence, the cultural knowledge of traditions like rituals surrounding illness enriches genetic counseling, fostering trust and leading to better outcomes. So, for just an example, whenever I go for pre-test counseling or post-test counseling, I try to get the information about the patient's or individual's educational background, their, you can say, the way of they live and the kind of, you can say, from the tradition they are coming from. So if I know all that, it helps me making the conversation easy, happy, and useful. The next, what are the advantages of cultural competence and what can be the disadvantages if you are not competent towards cultural sensitivity? So cultural competence in genetic counseling, this offers significant benefits. And I have seen that in real-time cases. By understanding and respecting diverse cultural backgrounds, counselors can provide effective guidance which results in high patient satisfaction. It helps us in reducing healthcare disparities, and it empowers individuals for making right decisions. Additionally, cultural competence fosters cultural collaboration, promoting a supportive environment. So, even with a single patient, you are not just talking to them about the test, but you become a collaborative partner in their patient journey. For instance, in a consanguineous marriage, where the marriages are happening between cousins, cultural competence allows the counselor to acknowledge the cultural significance of this kind of union while discussing the potential genetic risks, such as inherited disorders. By providing clear by providing clear, culturally sensitive information, the counselors build trust and support informed decision making without making any judgment about their patients and individuals or their families. So, in a nutshell, for cultural sensitivity, what I want to say that basically in genetic counseling, it is important to respect people's beliefs and values. This means speaking in a way they understand, speaking in a way the person who is being counseled, he or she understands, by building trust and offering language help. Sometimes language also becomes a barrier, and learning about different cultures. So let us take one example of language barrier. Language becomes a challenge sometimes because not all the genetic counselors are well versed with maybe the official 22 languages of Bharat. So with multiple spoken languages in a country like Bharat, the language can become a barrier in understanding complex concepts, exchanging information, and building rapport, which may lead to miscommunication. So, what to do? How to solve this problem? This problem can be solved, and one of the ways is that an English speaking genetic counselor can counsel a non English speaking patient or the individual by using professional interpreters. The interpreters should be a little bit versed with the genetics or maybe the life science, and also they must be knowing the language of the patient or the individual. That is most important. Good English, good non English language, plus the capability to interpret or translate. By this is one way, and then we should simplify language. So the genetic counselor's duty not to make it complicated. It should be the simplified language. We can incorporate visual aids like pictures, slide decks, and break down the complex concepts into small and clear points so that we can make it easy for the people to understand. In place of saying a long sentence with lots of commas and semicolons, etc., why not to make it a small sentences? In the same way, why not to make a complex concept simple by breaking it into small points? Most importantly, what we should do, we should confirm the understanding of the person who is being counseled by asking them to repeat the key information. So that becomes a good point. So what you can do, what I do generally, suppose if I have such kind of case, I tell my interpreter or the translator to make the notes. I also make the notes, and I tell them to ask the question from the patient or the you can say the individual, that they should repeat, they should tell us the answer once or twice if they have understood or not. And then we match our notes, and then we come up with the conclusion that we have passed the right information. Of course, it is very important to summarize the key points to ensure accurate communication, and documentation is very important in such cases so that there is no confusion. But this is the way we can help multicultural, we can say resolve multicultural issues or multilingual issues. But last but not least, genetic information has a significant psychological impact on people, which can lead to anxiety, fear, or uncertainty about the future. Therefore, it is essential to provide effective coping strategies to help the patients and their families to manage emotional challenges and take appropriate actions about their health. Let us see how. The psychological considerations or the important points which should be considered for psychological impact are: number one, the effect on emotional well being due to anxiety, fear, and sadness. Second, the relationships with family members can also be affected because of sometimes the genetic testing results. Next is social stigma caused by discrimination based on genetic testing results. This can complicate the situation. So, as a result of these factors, decision making can be compromised or can be impacted. Hence, we have to be very, very careful, and we have certain coping strategies for it. Now, what are these coping strategies? Let us see how to handle it. The process of coping strategies for psychological impact involves three important points or three steps. Number one, understanding. This means we should focus on how the patient feels, thinks, reacts, and behaves regarding their issues. It is very important to understand this. Second, exploration. In this stage, the counselor helps the patient to explore and become aware of the issue that they are facing and encourage them for self reflection and give them a deeper understanding of the problem and their psychosocial concerns. Finally, after understanding the issue and exploring what can be done, how it can be done, the counselor and the patient decide when and where to take the action to manage or control the situation effectively. Let me tell you one example. For example, BRCA1 mutation. This increases cancer risk, predisposition for hereditary breast, ovarian, prostate, and pancreatic cancer if it is a germline mutation. But at the same time, it also provides targeted therapy options like PARP inhibitors for cancer patients. The counselor should explain both the need for targeted therapy for cancer patients and as well as proactive management for asymptomatic individuals with BRCA mutations, emphasizing the importance of early detection and preventing measures. So, that is the job of genetic counselors, that we have to make the complex science very, very simple for the everyday people or common people. So, in a nutshell, to end my talk, I will conclude by explaining a quote by Minosh Shafiq with respect to healthcare. Here, what I want to say is that the future of any field, including people's health, will be handled not just through knowledge, but with also empathy and care. And the genetic counselors should, in fact, they have been doing, they should focus on emotional support, ethical decisions, and respect cultural differences of various people. By communicating clearly and addressing psychological needs, counselors can help individuals to make informed decisions, compassionate decisions, which ultimately makes the healthcare more human centered, or you can say human friendly. Thank you for giving me this opportunity and giving me time, and thank you for your time for attentive listening. I hope you found it helpful and valuable. Please let me know if there are any questions. Yes, Dr. Vinod, we can show that. I mean, I can answer the questions even if I can see in the chat box. I can do that.
Also, kindly unmute yourself.
Thank you, Dr. Preeta. That's true, Dr. Meera, what you're saying. So I think there is a question from Surpreet or Dr. Surpreet, I don't know. But the question is, can I switch my career from genome analyst to genetic counselor? And how is the career growth in this field? So my suggestion will be why to switch. Add it to your genome analyst career. A genome analyst can be a very good genetic counselor because they know in and out of genome data analysis. Career growth, yes, of course. We are talking to people. We are bringing you can say the bedside science to bedside, and we get chance to talk to people, and it is helpful in doing better, genetic testing options for them and helping them. So it's always there. I think I could not understand that question, Dr. Preeta. What do you mean by any comments on deliberate? Deliberate genetic counseling means just to force the people to do the testing? No, we don't do that. We guide them, but we don't do any deliberate genetic counseling. Thank you.
Thanks a lot, Jyoti ma'am. That was once again a wonderful talk. It's always great having you with a positive spirit. Thank you, ma'am.
Thank you. Thank you.
You are the most lively person in any room who can guide us. Okay, thank you guys. Hope you found the previous session on genetic counseling and communication of results by Dr. Jyoti interesting. Next up, we have an open house and panel discussion with eminent speakers, including Dr. Vinod sir, Dr. Sridhar sir, Dr. Jyoti ma'am, and Dr. Bani and Ms. Ramya Venkatesha. I'm welcoming all of you to the stage and adding you all in. Stage.
Thank you very much. And this is the last session for today's meeting. And in this session, we hope to discuss some pertinent points we've heard through the entire day, what WGS is, why is it important to do, how is it fast, what are the additional things that WGS can provide. To conclude and take a stock of things and to sort of reiterate on some of the facts that we covered throughout the day, I think we decided to have an open session. So probably the open session will start with me preemptively asking some questions to the panelists. And after that, of course, we could take questions from the audience. You could use the chat box to put in your questions, and we would be more than happy to take. Now, let's start with Dr. Sridhar. Dr. Sridhar has been a pioneer, including part of the first genome programs in India across Southeast Asia, and has seen right from the first genome to what is there in clinical application today. And he's also seen how genomics has become cheaper, faster, more efficient, and he has pioneered the application of genomics in clinical testing. So the first question is to you. What do you think is where the future of genetic testing is leading us? And what really do you think is the tipping point to realizing that?
So, thank you for the invitation to the forum. But can you just repeat the question again? I might have missed part of it.
Yeah, my question was now that genomes are becoming faster. Yes, you're audible, Sridhar. Can you hear me?
I can hear you, but I missed part of the question.
Yeah, the question largely has been that now genomes are becoming faster and cheaper. What do you think is the future of genomics or genomic testing?
Yeah, thank you, Vinod. Thank you, Harish and Bani and Ramya and Dr. Jyoti for inviting me to this forum. Since morning, exciting lectures, very pertinent by the audience. In terms of genomics, I think you only have a very bright future is all I can say if I were to summarize it. Okay, we are at a very tipping point of this whole technology journey. The technology journey began as early as 1990s or 2000s when the NGS came up as a next generation sequencing workflows came up. It was a very cumbersome workflow, but today it has become a very mature technology wherein the diagnostic labs, individual institutions are doing genome sequencing. And the return of results in terms of their interpretation, in terms of clinical actionability is only improving. In some areas, we have seen that it is the positivity or return of results as high as 90%. I can say in dermatology, in cardiology, that number would be somewhere around 65 to 70%. So on an average, we are seeing that about 65 to 70% of the cases that gets referred has very actionable mutation that the patient and the clinician gets back. Of course, in some cases, some diseases that number can be lower, but nevertheless, having said that, genomics come a long way. One test that really fits and can be done in place of multiple cascades that we normally do. Normally, in a usual genetic clinic, you would do a karyotyping, you do an MLPA. Post that, you will offer a very specific test to validate it. Sometimes do a microarray. All of that is done with. Okay, if you could, we are at a point in time where the primary testing can become whole genome sequencing, and that should really be a sum of all the tests together. And the speed at which it can be done, I'm sure today we discussed an extensive speed. So I think we are at a tipping point, and we as a community should be poised to use this tipping point for the benefit of patient care. I'll stop there. Thank you.
Thank you very much, Dr. Sridhar, for that quite elaborate summary of where the genomic testing is leading to. My next question is to Dr. Bani. You have heard Dr. Bani speak about genomics, pharmacogenomics, newborn screening. So essentially, my question is largely to the point, starting from that today genetic testing is done in clinics. Can we now move it further away to the cradle and further even more away to testing apparently normal people who might actually get benefit from genomic testing? Now that there is enough evidence, and all throughout the day we have heard what the evidence base is, what do you think this is leading to? Or what is the best optimum time to sequence an individual?
Thank you, Dr. Vinod, for that very important question. And especially after today's very vigorous session, it is something that should be very well answered in this session itself. So when is the best time to probably offer a whole genome sequencing test? I will say the answer is yesterday. and the second best time is probably today. And it might seem like a very dramatic kind of a line, but it reflects a very fundamental truth here, that the earlier we unlock somebody's genomic insights, the more time we have to act on them. But for adults, then basically, is it something that is too late to be done? Absolutely not. In fact, for many, this might exactly be the right time to get tested. Now that the sequencing technology is available, the evidence is growing, people are getting trained on how to actually interpret all of these results and how to go about it, basically. So, for instance, several large-scale whole genome sequencing studies that were conducted elsewhere in the world, including the very recently commenced UK 100K Genomes project, they have shown that approximately 3 to 5% of healthy individuals actually carry a clinically actionable variation, particularly in very important genes like BRCA1 and TP53. Now, these genes are linked to conditions such as hereditary cancers and familial hypercholesteremia, for example, and identifying them even in adulthood allows for very meaningful interventions that can help these patients now, or these people rather now. Be it enhancing screening, changing lifestyles, or doing any kind of risk reducing treatments for them. Then there is also pharmacogenomics, because generally speaking, adults are the one that benefit or are the most likely the ones to be on chronic medications, I'll say. So for example, antiplatelet drugs, antidepressants, chemotherapies, and so on are something that are generally given to adults, right? And yet adverse drug reactions are still very common because all of them were not screened previously. So, whole genome sequencing, I'll say, offers lifetime access to your pharmacogenomics profile. Does not matter whether you get tested in your childhood, during your newborn period, or when you are an adult. If, for example, you know somebody is already a poor metabolizer for DP or for the DP for 5-fluorouracil, for example, we can avoid giving them these ineffective or very harmful drugs. Another powerful application in all of this will be undiagnosed symptoms, because many adults live for years without an unexplained neurological, cardiac, or even connective tissue problems. And whole genome sequencing has helped resolve a lot of those diagnostic or disease, especially where prior panels or tests have probably failed, or even in cases where people do not even suspect that they have an undiagnosed condition. Because going by personal experience that we have seen in our company here also, that for most people, considering heart attacks in your family is not something that they deem as a family history, but it is something that is very important. It should get recorded, and it does influence how your WGS report will look like, because you might be at an increased risk of having these heart attacks because of a genetic predisposition that you have. And for all of this, let's not forget reproductive planning, because WGS allows couples to assess their combined carrier risk for hundreds of recessive or X-linked conditions, for examples. And for adults who are going about planning a family, this information can be very, very critical and more comprehensive than general routine panels. And finally, the concept of secondary findings, which was discussed in very much detail in this session, is something that is very applicable no matter at which stage of life you are. So to circle back to the question, no, it is not too late for anyone to get screened as long as they are willing to do so. And in our experience that we have seen here at Karkinos Healthcare, it is not just patients who are getting screened with using a whole genome sequencing. A lot of our bulk that comes in for a whole genome sequencing are actually healthy people who want to get a hold of their healthcare journey before it is too late. And these are people who are interested in starting early, knowing their health risks, and thus making informed decisions based on what is uncovered. So sometimes the most valuable insights may not come when you are born, but actually they might come when you are ready to act on them. So, yes.
Thank you. Thank you very much, Dr. Bani. My next question is to Ramya. So all in my life in 20 odd years in genomics, what we used to hear always when a new technology comes is that produces lot more data. We have heard it when Sanger sequencing was a mainstay. Panels used to create lot more data. When panels were in vogue and then exomes came in, no, we should not use exomes because exomes produce a lot of data. And I hear that again and again, even in the WGS era, the WGS produces lot more data. So my question to Ramya is, does actually sequencing a WGS leads to more answers, or it is only going to add more questions? And what is the line that we need to draw between the insight as well as uncertainty?
First of all, thank you, Dr. Vinod, for such a question. I mean, you have definitely put me in a spot, let me tell you that. But to answer your question, according to me, data is something which works only with context. So, more data definitely has potential to bring clarity, but only if we know how to interpret it. So let me give you some examples because we are speaking in context of genomics. So if suppose at WGS, we can tell yes, we do detect thousands of variants per individuals, but with proper tools and knowledge, it is very easy to gather insights from the data. Otherwise, it would lead to getting overwhelmed. Similarly, we have VUSs. I think the thing that still baffles everybody till now is that topic, VUSs. And I believe that, if you follow the right guidelines like ACMG network, ACMG guidelines, and if you follow all the structures and all the different attributes, I truly believe we can gather more insight than like it leading to unnecessary confusion. Also, with the right if you know the right clinical phenotypes, family history, and again, it would definitely be more beneficial to the patient. I think when we talk about data in this case, we should always look at what would benefit the patient more, because ultimately we are working towards the future for this particular patient we are dealing with. And since WGS can detect, variants that WES or panels can't, especially like UTRs, regulatory region, structural variants. Of course, it can also bring. So this can bring clarity to some unresolved cases also. Like I discussed a patient earlier who was struggling with some cardiac symptoms, and we discovered a structural deletion, and that really helped the patient a lot. So I believe. That more data means it can move away from, say, general assumptions. So we get some personalized data backed decisions, whether it's diagnosis or therapeutic decisions, all of it. So in conclusion, I would say yes, more data can definitely create confusion, but with the right tools, training, context, as well as with the right interpretation, it can actually become a very good superpower. So in genomics, data doesn't confuse, but poor interpretation would.
Thank you very much, Ramya, for pointing that out. In fact, data doesn't confuse, poor interpretation is what confuses people. My next question is to Dr. Jyoti. Dr. Jyoti has been, in many ways, pioneering whole genome genetic counseling, right from being a genetic counselor to start with. A whole genome is like talking about a whole book rather than an individual chapter, which used to be the mainstay in genetic counseling. Now, in your opinion, what do you think a genetic counselor, or rather a genomic counselor's role, would be to transform insights when guiding a patient through a whole genome sequencing?
So first of all, thank you very much, Dr. Vinod, for giving me this opportunity to talk on this forum and also for this question. And I feel that Dr. Bani has already told about targeted panels and whole genome sequencing, but who can be better than me to answer this question, not just as a genetic counselor, but also as the personal user of whole exome sequencing as well as whole genome sequencing. So what I feel that there are certain things like whole exome sequencing or targeted panel, they have limited scope. They can tell you they can give you limited results. Of course, the expectations are limited and the targeted results are there. If I talk from the point of view of counseling, it becomes easier. But because of limited scope, we will not be able to give, as you said, the book, the complete we will not be able to read the complete book of the person's say genome. Whereas whole genome sequencing covers almost 100%. Right? Let us consider whole exome sequencing also as a targeted panel, which is like 2% of the genome, whereas whole genome sequencing covers both exonic and intronic and almost 100% genome, which provides a wide range of results, which includes all types of findings, including incidental, carrier status, etc., and also pharmacogenomic findings. This makes WGS complex for counseling as well as, of course, for getting the results, as Ramya said, the data analysis. But the advantage is that if I do it early in life, I will be ready to handle my health care, my lifestyle choices, and that is what I explain to the patients or to the healthy individuals when they come for counseling and they want to know what is the use of whole genome sequencing in comparison to targeted panels. So the point is here, who does not want to know about their whole life and if they can take some action on it? It's like known devil is better than unknown devil. So if I know something on which I can take action, I would like to take action as early as possible so that I can remain healthy for the lifetime. In place of after getting the disease and then figuring out what has happened to me, and by that time the situation has worsened. So yes, of course, as I said in my talk, considering all the factors of communication and also the cultural aspects and everything, we have to explain it to the people that whole genome sequencing guides you for lifetime. Hence, it is important to do it as early as possible and not, I mean, I'm the user of WES as well as WGS. Personally, I have seen the differences in the results and what I could get in WGS, which was not possible in WES, whether it is intronic or exonic reason. So yes, by giving my example, by telling case studies, by telling stories, and also by guiding them for the advantages, pros, cons, etc., taking care of their emotional and psychosocial concerns, we can help them in understanding that. Thank you, Doctor.
Thank you very much, Dr. Jyoti, for the quite elaborate explanation and how genetic counseling could transform the insights and care for the patient. My next question is back to Dr. Sridhar before we move out to the audience. See, today we have heard about WGS, and a lot of people have been asking, is WGS the first line of testing? Is the last line of testing? Is the last resort? Is it affordable for a lot of people to do it, etc., etc.? It indeed, at the moment, is exotic medical test, typically used at the end of the diagnostic OTC. But given the evidence in hand and given the throughput that is being improving over the many years, Dr. Sridhar, what do you think, or when do you think whole genome sequencing would transform from being an exotic medical test to a fundamental human right for every single child or every single individual? If Dr. Sridhar is not there, Dr. Bani, would you like to take that question?
No, I'm there. Can you hear me?
Yeah, please.
Yeah, sorry, my mic was switched off. Thank you for that. Thank you for the provocative question. The way I see it is that the whole genome sequencing is already getting implemented in certain focused medical disciplines. Like we have seen in neonatology, in cardiology, in certain diseases which are life-threatening, especially. So in two conditions, I can clearly see it's already being implemented. In life-threatening conditions, number one, where a very quick answer is required. We don't have time for multiple rounds of testing. Second is where the evidence is not, you have a multiple levels of clinical presentation and it confuses the clinician. The provisional diagnosis is not clear. In that case, again, WGS has become very useful in arriving at a decision point. But having said that, I think newborn screening applications or applications where we have life-threatening conditions in the baby are two very, very pertinent cases where I see it has to become a universally available tool, because today it does benefit the individual and the society. Second is people who have a family history of a genetic disease, which today can be treated or prevented or postponed. I think WGS has to become almost a right, because the tools are there. Not using them is criminal. However, I will also express a word of caution when it comes to whole population level deployment. We do need regulations around it. The European continent or the US continent have deployed very good examples of how the laws can be framed to protect such a deployment at a country level, or so I think the Middle East is almost there, and some small countries have been successful. I think it's time that we implement a regulation around the misuse of whole genomes, so that people could actively use this technology at a population scale. But that is just a matter of time, I think. It's just a matter of time before it happens, even in a country like us. So I'll stop there. And I think I'm only seeing a very bright future for this technology. Over to you.
Thank you very much. And I think now we can open it up for questions from the audience. So this question is from Hadisur Rahman. And the question is what is WGS the last resort? What about other omics layers? And what's the future of diagnostics beyond WGS? Dr. Bani, would you like to take that question?
Yes, sure. So WGS, I won't say it is the last resort. It should actually be the first line of diagnostic or therapeutic discovering option that should be provided to patients now. And now that I think through this very extensive session, we have tried to make everybody here understand that WGS is one single test that can not only reduce the diagnostic odyssey that many rare disease patients follow, but they also help. The single test also informs the treatment plan that will be best suited for the patient. Now, given that all of this is now presently possible, there are a lot of different things that can come up in the future as well. For example, going beyond just whole genome sequencing, there is a telomere to telomere whole genome sequencing that has also come up thanks to long read sequencing data that is now supported by machines like PacBio. So now this kind of sequencing not only sequences your entire genome, it also gives you an insight about what is the how it can basically expand this testing to the complete ends of your genome, which are the telomeres, which is something which has been till now a challenge to do so. So there are a lot of things that can come up in whole genome sequencing, which are not just limited by science, and now they are not even limited by the analysis part of it. So this is one test that has generated a lot of data from your DNA, and then this single test can thus be used to now find out a lot of different things from your genome. And that, coupled with other omics techniques such as transcriptomes, can help understand diseases and the basic biology of how we work and live in a better context, I feel.
Thank you, Dr. Bani. The next question is from Jitendra Naik. And he asks, what is the evidence threshold or policy shift that is required to justify the routine clinical implementation? Dr. Sridhar, would you like to take that question?
Sure. Very, very pertinent question, and we clearly do require a policy shift before the routine implementation happens for multiple reasons. One, how do you ethically use this data in clinical settings? That needs to be defined. We need to define how this data is used in regulations in terms of insurance or in healthcare schemes. So clearly, the implications of this go beyond just routine sequencing. So I really think that we should implement the equivalent of what people have done in other countries to protect common citizens, and that has to be through one of our regulatory bodies. And I'm aware that there are several discussions that are ongoing to implement it so that it doesn't get misused. Also, misused during transmission of information. This requires a larger deliberation, but clearly a country level policy is much warranted at this stage for this technology to be widely adopted. I'll stop there. Thank you.
Thank you very much. The other question is from Chandrika. The question is, what are the fastest growing career roles in WGS right now? Whether it's of a clinical geneticist, analyst, counselor, or a bioinformatician? I would pass this question on to Ramya as well as Dr. Jyoti. Ramya, you could go first.
Okay. It's a very great question. I think, especially if you are a budding student, I don't think I can tell you in terms of a bioinformatician, because from whatever experience I have had, it is game changing. And I think the best part is it is the most fast growing in terms of you get to do new things every day, you discover new things every day, and you also feel like you're giving back to society. Staying in a corporate role, that is the best part. So I think I cannot speak in terms of, say, a clinical geneticist or a counselor. I think Jyoti ma'am can give you more insight on that.
Sure. Thank you, Ramya. I agree with you because, as you said, being a bioinformatician and doing WGS, it's wonderful. I mean, you are solving the mysteries of life. So that is there. But yes, of course, those mysteries are to be spoken to the common people. Whose mysteries are these? Yes, genetic counseling role is very, very important. We have very few genetic counselors in our country. We do not have enough counselors for a country like us with huge population. So yes, genetic counseling has got a wonderful scope. If somebody becomes a genetic counselor, they are in demand nowadays, and I think this will keep increasing. So yes, people can opt for that also. Yeah.
Thank you very much, Dr. Jyoti. The next question is from Dr. Tanu Datta. Can polygenic risk score be also calculated from whole genome data? I can take that question. In fact, the polygenic risk scores can be calculated from individual WGS data. At this point in time, there is a guidance against using it for clinical care. It's largely for public health as well as evidence making on a cohort of individuals, but not to be advised at an individual level. I'm sure for the constraint of time, we could not take a lot more questions, and therefore we'll take just one last pending question. And this is from Arun Biju. And the question is, has there been a consideration on how it might impact the patient's health insurance, especially if some incidental finding comes up? Wouldn't be that an issue? Have you faced any cases such as this? Would Dr. Sridhar like to take that question?
Sure. So again, a very pertinent question that often comes to us from employers as well as employee on how do you handle this. So till now, any such question with an incidental finding or incidental finding that impacts health, okay, when it comes as a surprise, usually the insurance ends up spending a lot more than using it as a preventive measure to sort of mitigate it or address it very early on in life so that a planned approach could be taken. I think we have sort of communicated this in so many words or as a policy to many of our collaborators and insurance companies, and who are taking the right look at it in the manner that they're sort of leaning towards taking a planned approach, not a knee-jerk reaction towards such incidental finding. So I think it's only a matter of time, and the regulatory laws that I was referring to earlier are very important to protect such people from getting discriminated. Over to you, Vinod. Thank you.
Thank you very much for that quiet, elaborate panel discussion. Thanks to all the panelists for joining me and also providing your valuable insights. I think for lack of time, we'll need to close this fantastic session. And thanks to all the speakers in the session for providing their insights. I'd invite Harish to close
Thank you,
the
Vinod.
session.
Thank you, Vinod. Thank you, Vinod, and thank you, rest of my members.
Thank you, sir. Thanks a lot to every single one for attending the program. This is really wonderful. The kind of discussions that we had on the forum, the kind of knowledge transfers that happened across. It's no more about just me learning or just a few of my team members at Karkinos learning. It is about an entire community of people coming together and learning what is really important and what is actually making us the genome generation as a whole. Thanks a lot for being a part of this session. We would be also sharing a post-course questionnaire. So that would come with a set of tests and a lot of interesting questions which will help you to understand the subject much better. And post that, there will be certifications and certain key insights and a surprise gift in terms of whole genome sequencing along with it. So glad to have every one of you here. Thanks to each and every single one of the speakers of today's session. Thank you, Vinod sir. Thank you, Jyoti ma'am. Thank you, Bani. Thank you, Ramya. Thank you, Sridhar sir. Thanks to everyone who helped us organize this. And thank you all for attending. Thank you.
Thank you, Harish, and thank you, everyone.
Thank you, Harish. Thank you. Thank you. Wonderful.
Thank you, Harish, and have a good evening. Thank you very much.
Sure, sir. Thank you all. Bye-bye.